Is it? Voice is audible, everybody let me know through messages. Huh? It's not coming. Yes, okay. Voice is audible, right? Yeah. So can you see the screen? Okay, fine. Shall we start now? So today I'm here to, yeah, it's audible, yeah. Today I'm here to complete class 12 portion. Okay, and based on students' difficulty level, yesterday when I went for rounds in study hours and students asked me to start with evolution. So first I'll start evolution and later genetics. After that only based on the availability of time in sequence, I'll complete all the portion. I'll maximum try my level best to complete all the points required for your exams. Okay. And for this, what you have to do is open your textbook and keep it ready, and some rough notebook also, because any calculation part is there, then we'll solve questions in that only. So keep your rough notebook also ready with you. Okay, total NCERT based revision I'm going to do now. Okay, this is about weightage based on class 12 topics. So human reproduction, yeah, we are expecting 3 to 4 questions. Last time 5 questions they asked actually. Reproductive health, 2 to 3 questions. Genetics and HGP, DNA fingerprinting based, total 3 questions we are expecting this year also. Evolution, 3 to 4 questions. Human health and diseases, 3 to 4 questions. And biotechnology, its applications, zoology part, 1 question we may expect. And ecology, 3 chapters are there: organisms and populations, biodiversity, ecosystem. Yeah, weightage is good and easy to score also, and 6 to 8 questions we can expect total. Today now I am going to teach and we are going to discuss for 22 questions. Okay, so almost half of the paper. So be focused, children. Important thing is don't sleep in between. Okay. Okay. Evolution NCERT. Students complain about this topic is how many ever times they read the statements and all that, they are not able to remember, it seems. So we'll try to rectify that problem now and consider this as the final zoology revision, okay, based on NCERT, because again you don't find this much time to read every line from textbook. Only based on main points you have to give your revision, because after this revision you have to be totally ready for your grand test. Okay, so I'm here to help you for that. So shall we start, because already we are late, and I'll immediately start the chapter. Evolution first. Okay, evolutionary biology is the study of history of life forms on Earth. Okay, history of life forms, it seems. History of life forms means suppose I have to know about your history of life, then what I have to know first? I have to know about your origin, and I have to know about where you studied and what you are doing now, like that. So origin of life and evolution of life forms. So that speaks about like total history of life forms. So evolutionary biology is what? Study of history of life forms, which include origin of life and evolution of life forms. Okay, now coming to origin of life. Origin of life, first of all we have to know means, first we need to know about origin of universe and origin of Earth. Okay, let us see about origin of universe. So Big Bang theory explains about origin of universe. Origin of universe, so according to Big Bang theory, it's actually hypothetical. Nobody was there to notice anything. Just hypothetically scientists estimated it would have happened at that time when 20 billion years before actually. So when exactly universe was originated? 20 billion years ago. Okay, Big Bang theory says that universe initially was just like a fireball, very tiny, very dense structure, very hot. It is like a fireball, and that ball-like structure, the fireball-like structure expanded with huge explosion and also with the louder sound, which that sound also we can't even explain in our words, it seems. Okay, that much huge explosion with a lot of sound it happened, and now, even now, according to this theory, this universe is still undergoing expansion. It's not stopped. Even the expansion is ongoing even now also. It's a continuous process. Okay, so Big Bang theory explains about origin of universe. Universe is actually 20 billion years old. Okay, then they said that, like, now gradually, if you see, at that time when universe undergone for the explosion, and hydrogen helium first actually released from that particular structure. Later on, these chemicals, hydrogen, helium, and gradually undergone for the reactions, and gradually atoms became molecules, molecules became inorganic, and organic compounds like that, and gradually solar system also formed. The solar system formation was explained by nebular hypothesis. Okay, nebular hypothesis says that universe, Earth was originated 4.5 billion years back. When universe was originated 20 billion years ago. So now when Earth was originated 4.5 billion years ago, according to nebular hypothesis. Okay, so next here, when Earth was formed only, like almost sun, everything was formed at that time only. Then they believed that that time actually atmosphere was reducing atmosphere, meaning no molecular oxygen available in the atmosphere. So no molecular oxygen available means we term that atmosphere as the reducing atmosphere. Okay, so according to the scientists, initially, in the surrounding medium, water vapor, methane, carbon dioxide, ammonia were released from the molten mass. Okay, what are the compounds were there? Methane, carbon dioxide, ammonia, and water. And sun was also there, right? When Earth was formed only, sun was also formed. So in the sunlight, UV rays were there, and these UV rays split up the water molecule and broke it up to hydrogen and oxygen. And hydrogen was a light gas, so it used to escape, and oxygen reacted with ammonia, methane, and form water, carbon dioxide, and other substances. Okay, this oxygen, you may say that, ma'am, you said early atmosphere was reducing atmosphere, and now you are saying that oxygen is combined with ammonia and methane. Immediately it used to get reacted. The molecular oxygen used to get reacted immediately, and free molecular oxygen was not available. That's why we consider the early atmosphere as the reducing atmosphere. Okay, so this O2 combines with oxygen atom, and ozone layer was formed like that only to protect Earth from the UV radius effect. Okay, and gradually water vapor also, because temperature, when universe started expanding, temperature also cooled down. In the early atmosphere on Earth also, temperature was around 5,000 degrees centigrade. Gradually it came down. Even now, that's the that is supported to to support lot of chemical reactions. Okay, gradually on the Earth, life appeared 500 million years after the formation of the Earth. Okay, life appeared when exactly? 500 million years after the formation of the Earth. Okay, so you can consider 4 billion years back. Okay, and here in galaxies, actually a lot of stars, planets, rocky bodies, all these used to be there, and the distances between these stars, the heavenly bodies, anything, planets and all, called as stellar distances. We measure stellar distances in light years. Okay, stellar distances are measured in light years. Okay, now this paragraph is totally about origin of life, the theories. Many scientists came forward to explain about the theories, and first, actually, theory of special creation. So this paragraph you can mention theory of special creation, and one more is theory of catastrophism and theory of panspermia. They highlighted here, theory of panspermia, and one more is theory of spontaneous generation, and Louis Pasteur-based theory of biogenesis, and last we all accepted for theory of chemical evolution. Okay, so let me explain about all that. First one, theory of special creation. Don't get confused between special creation and spontaneous generation. Theory of special creation means it's all about God is the creator and God is only a destroyer. Like that, the people used to believe everything happens because of God only, like that. So here the postulations, 3 postulations related to theory of special creation were explained. Okay, so theory of special creation says that, first point, all living organisms that we see today were created as such, it seems. Then second point says the diversity was always the same since creation and will be the same in future also. Okay, third point says that the earth is just about 4,000 years old. It's wrong, actually. Don't believe this, it's wrong. So earth is just 4,000 years old only. So people were in this type of views, like everything happens due to God only, like that people used to think. So at that time, many scientists also proposed many theories. Next theory is here they mentioned theory of panspermia. So this one, Greeks, early Greek thinkers supported this theory of panspermia or cosmozoa theory. Theory of panspermia or cosmozoa theory. Okay, so Greek people supported this according to them. This is really funny for us to see, but actually according to them, panspermia or cosmozoa are small seed-like structure, spore-like structure, came from outer space, fallen on the earth and germinated like new life forms, it seems. Okay, and of course we don't believe, but a lot of fans are there for that theory also even now. Okay, so panspermia are like small seed-like structure, came from outer space, fallen on earth and germinated as life forms, new life forms. That's their opinion. No evidence actually, so many people rejected this particular theory. So here you remember in this paragraph, theory of panspermia supported by early Greek thinkers. Okay. Next, about theory of spontaneous generation. On top of that only, you can even mention this as theory of abiogenesis. But don't get confused with this abiogenesis term. Why? Because theory of chemical evolution also can be called as abiogenesis sometimes, because that is also from non-living molecules actually. Okay, you have to be careful to understand the that particular term. So theory of spontaneous generation or else theory of abiogenesis proposed by our favorite Aristotle, father of biology, and von Helmont, like this type of scientist, big scientists supported this particular theory. According to this, new life is originated from the dead and decaying organic matter. Okay, dead and decaying organic matter like straw, mud, like that. They believe from Nile river mud, no, lot of aquatic arthropods, okay, lot of rodents, everything originated from that mud only, it seems. Okay, like that they did Aristotle, this von Helmont and all did experiments and proved that actually. Don't know how it happened. And Louis Pasteur, Reddy Spelagini, these scientists, you remember them for theory of biogenesis. Theory of biogenesis. According to these scientists, whom Louis Pasteur, Reddy Spelagini. According to these scientists, new life originated from pre-existing life forms. New life originated from pre-existing life forms. Okay, so Louis Pasteur is famous for his swan neck flask actually experiment, swan neck shaped flask experiment. Okay, he did one experiment and proved that life existing, like new life is there, means definitely pre-existing life form should be there, but he couldn't give, and these 3 scientists couldn't prove that how that pre-existing life was originated. Okay, that's the reason. And even theory of biogenesis is valid to reject the theory of abiogenesis or spontaneous generation. So that point you remember properly. Who rejected, who disproved theory of spontaneous generation? Who disproved theory of spontaneous generation? Louis Pasteur. Okay, Louis Pasteur. Okay, next one. We'll go with the theory of chemical evolution. Theory of chemical evolution was proposed by A.I. Oparin. Okay, he's from Russia, and Halden Halden of England. Okay, JBS Halden from England. These two scientists, Oparin and Halden, these two scientists are popular for theory of chemical evolution. According to these scientists, life existed from, like new life forms existed from non-living organic molecules. That's the reason some people consider this theory of chemical evolution otherwise as abiogenesis. Now only I told theory of spontaneous generation also called as abiogenesis. So don't get confused. So what they are asking in the statement, like whether they are asking you to find out that the new life is formed from dead and decaying organic matter, non-living matter, that time you can go with theory of spontaneous generation. Okay, and for this chemical evolution they will definitely mention non-living organic molecules, they will mention. Okay, that time you understand, oh, this abiogenesis is related to chemical evolution, like that. Okay, so here according to this Oparin and JBS Halden, life is originated from non-living organic molecules. They believe that in the early atmosphere actually supported for a lot of chemical reactions. At that time, reducing atmosphere was there, so that was one of the favorable conditions for the reactions to happen. And volcanic eruptions were there, and high temperature was there. So because lightning, continuous lightning process was there, these acted as the energy sources to support lot of chemical reactions. Okay, so these chemical reactions produced, like first initially atoms, let us take, and atoms reacted with each other, and later they formed, first atoms reacted with each other and formed molecules. You can consider like initially inorganic, and gradually inorganic compounds reacted with each other to form organic, simple organic compounds, and later simple organic means like amino acids, monosaccharides, something like this, and they also reacted with each other and formed complex organic compounds like amino acids would have reacted with each other to finally form a protein, complex protein, and monosaccharides, and finally formed polysaccharides. Like that, complexity gradually increased. See, I am explaining about Oparin and Halden's view, theory of chemical evolution. So at that time, the conditions like energy sources like lightning, UV radiation, then reducing atmosphere, okay, volcanic eruptions could provide some energy and acted as the energy source for reactions to happen. Okay, so at that time, atoms became molecules, molecules to like even inorganic to organic, like that, gradually complexity increased. And some believed, this Oparin and all, they believed that initially, like before, like living forms were created, first protobionts were there, it seems. Remember this particular term, protobionts, primitive life forms. Primitive life forms. According to Oparin, that primitive life form is coacervate. Okay. And some other scientists also, microsphere, like that, they said that microsphere concept. Okay, Sidney Fox, so many scientists came forward here. So coacervate or microspheres, or some people believed that RNA concept, like RNA was the first protobiont, which later gradually evolved as living forms, like that. Okay, so because see, from chemicals suddenly life will not like life forms will not create. So some transitions definitely would have been there between totally non-living and the living form. So that transition we are speaking, we are considering that as the protobiont. Okay, this protobiont can be coacervate, microsphere, or RNA. They did not confirm, and this only has to be a protobiont, like that. They did not confirm, so we also no need to worry about it. Worry about that. In NCERT also 2-3 places they mentioned we are not sure in between chemicals and life forms what is there exactly. We are not sure, like that. In NCERT also they mentioned, but definitely transition stage would have been there. That is only called as protobiont. After these protobionts, what to say, first formed living structures are chemo autotrophs. No, before that also, first life was originated in water, that you remember. Okay, heterotrophs. So after protobionts, first heterotrophs would have been there. Heterotrophs. Then after these heterotrophs, okay, next I think you can continue, otherwise I'll write this side. After heterotrophs, I'm continuing this one this side. chemoautotrophs. Okay, chemoautotrophs. After these chemoautotrophs in the evolution gradually, and they evolved as anoxygenic photoautotrophs. Anoxygenic photoautotrophs. Okay. Then after that, oxygenic photoautotrophs. Oxygenic photoautotrophs. And after that, prokaryotes, after that, eukaryotes, unicellular, multicellular condition, like that gradually. I'll read it out if it is not clear for you. Atoms reacted with each other, formed molecules. Molecules to inorganic, organic compounds were evolved. Okay, aggregation of some of the organic compounds shown little motility, little growth, divisions, etc., etc. They consider this transition stages as protobions, and later on, gradually heterotrophs were originated in the water. Then after that, gradually in the evolution, chemoautotrophs were evolved. After that, anoxygenic photoautotrophs, and after that, oxygenic photoautotrophs, and prokaryotes, eukaryotes, and like that only unicellular became multicellular condition, and this much diversity today it is created. Okay, this is all about Oparin's views. So for this type of theory of chemical evolution, experimental evidence was given, because for all the other theories, if we came across a lot of theories, all these theories were lacking with evidence. That's why theory of biogenesis or abiogenesis, so because of that only they rejected. But for this chemical evolution, actually experimental evidence was given by Stanley L. Miller and Urey also, Harold Urey, one more scientist, Urey, Miller and Harold Urey. These two scientists actually gave experimental evidence for theory of chemical evolution. These two are American scientists. Actually in 1953 they simulated all the early atmosphere conditions in a laboratory, and they created this type of equipment, glassware also. This is Miller's glassware, and he set up, he simulated all the conditions in the laboratory just like early atmosphere. And here he had taken, see, this is the spark chamber, this is boiling water chamber, and here he has taken the ocean water, because at that time first life originated in the water only, na? So in water, all the organic compounds used to get dissolved in that water, so that type of water he took. This is called as primordial soup, not any other soup, tomato soup or like that. Primordial soup means the ocean water with dissolved a lot of organic chemicals over there. Okay, then he boiled this water, and here in this spark chamber he had taken the mixture of chemicals. In that mixture, CH4, NH3, H2O, H2. So this I'll keep for some time aside. CH4, NH3, H2. These three actually in 2:1 1:2 ratio. I repeat once again. CH4, NH3, H2. CH4, NH3, H2. Ratio is 2:1 2. 2:1 2. Okay, this chemical mixture he had taken, and water like in the form of vapors. Then he connected this spark chamber with two electrodes. So by using these two electrodes, 75,000 volts of electric spark he could produce. And he simulated 800 degrees centigrade temperature in the laboratory. 800 degrees centigrade temperature, 75,000 volts of electric spark was acting as energy source for the reaction to happen between the simple chemical substances. Okay, 18 days experiment continuously. So 18 days he performed this experiment, and all the products which produced were in gaseous condition, because 800 degrees centigrade, right? So he used a condenser to condense those gaseous products, and finally he could collect amino acids like alanine, aspartic acid, glycine. What were the amino acids he could produce? Alanine, aspartic acid, glycine. These were the amino acids finally he could collect from there. Okay, so this is about, and many scientists also came forward on by changing the energy source here, because in this setup energy source was not fixed. Any, like, you can change your energy source and you can change the chemical mixture like that. So many scientists came forward, changed their energy sources here, and some used UV radiation and all that, and instead of this HCN and all they have taken. At that time the products were different. Okay, when they used HCN they could produce nucleic acids, purines, pyrimidines and nucleic acids finally, and some could produce proteins, some could produce these polysaccharides and all that, like that, all the complex organic compounds they could produce. And they believed within some 2-3 weeks of time only they could by using simple substances they could produce complex organic molecules. So in the nature, in millions of billions of time, definitely evolution like this much diversity would have been created, like that they believed, and they supported theory of chemical evolution in that way. Okay, next, first non-cellular forms were originated. So I'll show you that line. First non-cellular forms originated 3 billion years back. First cellular forms originated 2 billion years ago. 3 billion, you can even consider that as 3,000 million years back. And first cellular forms 2,000 million years back. Okay, children. So simple correction here in the paragraph. Here theory of chemical evolution only he referred in this paragraph also. Okay, so he considered that theory of chemical evolution here as theory of abiogenesis. Okay, not spontaneous generation. Here you consider this as the chemical evolution based only. A should not be away from the term, it should be like combined abiogenesis. It's not abiogenesis, it's combined abiogenesis. Okay, that's the only correction here. And next, coming to evolution of life forms, a theory. Yeah, many feel difficulty to understand certain statements, but beautifully written. We have to appreciate NCERT people for this. Yeah, evolution chapter they wrote well, but here and there, no, like in your level to understand the essence of the statement will be tough. Father of evolution. Who is father of evolution? Charles Darwin. Okay, he is a British naturalist, and he had gone for 5 years of sea voyage by a ship that is called a HMS Beagle. Okay, so he traveled for years. Of course, most of his research happened 3 years almost. Among those 5 years, 3 years he stayed in Galapagos Islands. Okay, he researched so much and he worked almost on all animals, but Darwin finches were his that's a favorite research area which he did. Okay, and here in these lines they mentioned that he noticed the similarities between two organisms, and also he noticed about the differences, and those differences he termed as variations. And gradually he said, see, in the group, actually in the group of organisms, suppose some variations are there, like if among those variations any positive variation is there, definitely those organisms, those individuals with the positive variation will be definitely selected by nature. Obviously, right? So among us, suppose high IQ people are there, they always take the benefit. Okay, some extreme talented person is there, or extreme strong person is there, one or the other way they'll be taking the benefit. Like that, Darwin also noticed that in the group of organisms, whichever has positive variation definitely will be so comfortable in that particular area and will be winning, like in the competition or whatever, they'll be taking the benefit. Okay, in these lines he mentioned that only. So our organisms with positive variation is more fit to be in that particular geographical area. So that fitness, according to Darwin, refers ultimately and only to what? Reproductive fitness. Very, very important statement for exam. Okay, the fitness according to Darwin refers ultimately and only to what? Reproductive fitness. Those organisms being so comfortable in the geographical area can produce more number of offsprings, and their population sizes will increase. That particular species will dominate in the geographical area. That's what he understood. Okay, and he considered that selection of that positive area. You are with the one characteristic feature, you are so comfortable. That itself is considered as natural selection. Natural selection is not something different. Natural selection means by having a characteristic feature, you are selected and you are so comfortable in the geographical area. Okay, so by having that particular character, those organisms are surviving more better and reproducing more better, and that itself he considered as natural selection, and he mentioned that as the mechanism of evolution. What is like natural selection itself is like mechanism of evolution. Okay, and the same time, just like Darwin only, Darwin is famous for his Darwinism. Okay, same like this natural selection based explanation was given by another scientist. He is Alfred Russel Wallace. Okay, Alfred Russel Wallace. He is also a British naturalist. When Darwin worked in Galapagos Islands, Wallace did his research in Malay Archipelago Islands. Malay Archipelago Islands. Okay, and so remember this also important only, just similar to Darwin's research. Which scientist, which another scientist also did the same research means Alfred Russel Wallace. Where did he do? In Malay Archipelago Islands. Okay, and all the existing life forms share similarities and share common ancestors. Definitely, see, we show similarities with our the same other group individuals. Not only with the existing people, we also show similarities with the extincted ones also, like our ancestors and all. They may not be there on earth now, but definitely we show similarities with the ancestors. So like that he told. And he spoke mainly, he spoke about common ancestry, how variations are accumulated, getting inherited to the next generations and all that. Of course, again, Darwinism is there in next, after 2-3 pages. Again, I'll highlight his views under that. Okay, and this line also important for exams, for assertion, reason and all that. The geographical history of Earth closely correlated with the biological history of Earth. Okay, geological history of Earth, like any changes happening in the Earth, like in the initial stage when Earth was formed and all that. So those changes only created the difference, like which type of life forms now it has to be existed or extincted, because based on that only, like the conditions, environmental conditions are deciding which species in that area has to be dominated, which has to be suppressed, and which will survive better, which will extinct. All this will be decided. Okay, that's why first geological history, that will decide, that the geological history of Earth, that will decide what type of biological history, biological life forms have to be there on the Earth. Okay. And next, this is one of the important areas, the evidences. So first we discuss about paleontological evidences. Paleontological evidences. Paleontology means study of fossils. Study of fossils. Okay. Next, we will discuss about embryological evidences. We will discuss later about comparative anatomy and morphological evidences. After that, we discuss biochemical evidences and vestigial organs, atavistic organs, like that. Also, so many evidences are there, and we restrict to NCERT based concept only. And here, among whatever evidence, let them give in the exam. Yeah. First, they will ask which is the direct evidence in the evolutionary history, like that. Direct evidence is like paleontological evidences only. Fossil based evidences are direct evidences. That is important. What are fossils? Fossils are dead and what to say, remnants of the dead organisms. Remnants of dead organisms. Okay, so you can consider remnants are remaining parts of the dead organisms. So for plants, you can say sometimes, where did it go? So for plants, you can take some pine cones, and sometimes leaf prints will be remained, seeds will remain. Okay, and for animals, if you see bones, okay, then the entire skeleton also. Sometimes footprints also will remain. Okay, like that. So remnants of parts of the dead organisms, plants or animals, it can be called as fossils. Study of fossils is called as paleontology. Where do we find fossils? Which type of rocks? Sedimentary rocks. We like sources of fossils. It's sedimentary rocks only. We find these sedimentary rocks mostly near the oceans, actually. Okay, and 1 second, I'll open NCERT. Yeah. So Fossils are remains of hard parts of life forms found in rocks. So main sources for fossils, sedimentary rocks. Okay. So here, by seeing the fossils, we can understand the characteristic features of organisms, and we can estimate the age of the fossil also, and we can connect to that fossil to the existing species, and we can have the evolutionary history of that. So fossils help us to know more better about the ancestral history. Okay, and how we can estimate the age of these fossils. So one is like relative, the estimation of age of the fossils, radioactive dating, and one more is relative dating. Relative dating is very simple. The first, relative dating. See, generally how fossil is formed means whenever one organism dies, like imagine like one buffalo or something is walking at the bank of the river or by bank of the ocean or something, like maybe certain incidents actually died over there only, and the flesh part of the organism easily get decomposed. But maybe see at the bank of ocean, if you see, actually salt content is more. In that environment, decomposition process is delayed. Okay, now so mainly see fossils we find closer to ocean only because salt content is more. That prevents the decomposition and otherwise delays the decomposition. In the meanwhile, when the process is getting delayed, the what to say, our like chemicals and all that present in bones and all that will be replaced by, okay, calcium, whatever phosphates, whatever are there, that will be replaced by iron, then silica present in the sand, okay, some carbonates and all will replace the material. Actual living material is replaced by that. That's how preservation will happen. So this preservation of fossils is called as petrifaction or fossilization. Fossil formation is fossilization in simple, so that is only called as petrifaction. Okay, so in this petrifaction process, what happened exactly? Our flesh, all that will, other tissues will be easily decomposed, and the material present in the bones and all that is replaced by some other chemicals like silicates, iron, then carbonates, etc., etc. That's how preservation of the organism happened. That is only that preserved material only we are calling that as the fossil. Okay, so here in relative dating, how to estimate the age of the fossil? But of course, it's a lot of what to say, exact age we can't estimate based on this, but approximate age can be estimated. Suppose like this is the sand material where the organism died. Here the one organism died, okay, long ago. Okay, and some after some certain days, if you see the here some sand would have been deposited, the dead body would have been covered because of air or something. Daily some sand will fall on the dead body, so like that it would have been covered. After so many years, if you go and see also, you can't find that particular dead body because it would have properly buried. Okay, maybe accidentally then after so many years, the same spot again one more dead body is here. Okay, again even this also gradually will be what to say, covered by the sand, etc., etc., like this only, and this sand and all would get hardened up. Unlike that, the rock also would have been created. Generally at the bottom of the oceans with the sedimentary material, it will happen. Okay, so now like here also one more dead body is there, like that, let us assume. Okay, so like this in the same spot, but periods are different. Long ago it would have died, and after some years, and this is like more recent dead body, like that. So how to estimate the age of it? Like now if you see this particular rock strata, and which is more recent and which is oldest one, like that, like can you identify by seeing the position of it in the layers? Like which the dead body which we find in the upper strata definitely will be more recent. Okay, the dead body which we find at the bottom strata, definitely it's the oldest one. Okay, so based on this, actually relatively we can understand the age of the fossil, but not accurate. Accuracy will not be there in this method. That's why one more method is actually radioactive dating method. radioactive dating method. So here carbon 14 converted into N14, nitrogen 14, like based on the half-life period of certain isotopes, they estimate the age of the fossil. So carbon, radioactive carbon dating method and radioactive potassium dating method, uranium method like that. So potassium 40 also converted into argon 40. So like that, and uranium also converted into uranium 238 is converted into lead 270. So like this, based on certain isotopes, a half-life period will give us idea about the age of the fossil. Of course, like 70,000 years old fossil age can be estimated by using carbon dating method. Very old, million years old means, yeah, potassium they are using. Okay. Even this is also not most accurate, and most accurate one and easily the age of the fossil can be estimated by using electron spin resonance method. Electron spin resonance method, that's the most accurate method. Okay, so so this is about how to find out the age of this italics they mentioned. For this the answer is this one. So very radioactive dating method. So very clearly we can estimate the age of the fossil by using what is electron spin resonance method. Now DNA also they are using. So a lot of other technologies improved here. Next about embryological evidences. Embryological evidences first about Ernst Haeckel. Ernst Haeckel considered as father of embryology. Okay, Ernst Haeckel is father of embryology. One more scientist also mentioned here. He is Carl Ernst von Baer. So Carl Ernst von Baer is considered as father of modern embryology. See here, both of them are fathers only. Okay, Ernst Haeckel is father of embryology. Carl Ernst von Baer is father of modern embryology. Okay, the views are very interesting. First, Ernst Haeckel said that he proposed also one theory based on his research. The name of the theory is, let me write here. Ontogeny repeats phylogeny. Ontogeny repeats phylogeny. Otherwise, it is called recapitulation theory. Recapitulation theory. And one more is biogenetic law. Same only, biogenetic law. You can call the same thing as recapitulation theory or biogenetic law or ontogeny repeats phylogeny. Proposed by Ernst Haeckel. Proposed by Ernst Haeckel. Whatever they mentioned, we are giving a name for that particular hypothesis, that's all. It's not beyond NCERT. Okay, so Ernst Haeckel noticed the embryos of all vertebrates. Okay, fish embryo, frog embryo, reptile embryo, bird, and mammal embryo, everything he compared. He noticed, oh, everywhere zygote undergo cleavages and converted into morula, blastula, etc. like this only. He noticed a lot of similarities are there like that. And even when organs are created, see, for mammal embryo, let us take. We don't use actually gills for respiration. Okay, we actually use lungs only for respiration. Okay, but instead of lungs in our embryo, we show the presence of gills. Okay, gills are formed. Okay, that's why Ernst Haeckel said that. So gills actually those are the primary respiratory structures in fish embryo. And we are showing, like in our embryonic stage, we are showing the fish characteristic feature. So like that, he said, the ontogeny means our birth history. Ontogeny means birth history. Okay, repeats what? Phylogeny. Phylogeny means evolutionary history, ancestral history. So in our embryonic development, during our birth history, we are showing characteristic feature of fish, no? So like that, we are repeating the ancestral characteristic feature. Like that, he said. And not only that, heart formation, let us take. Ours is four-chambered heart, right? So, but instead of, in the beginning only, instead of formation of four-chambered heart, like we have shown first actually one tube. Later that forms two-chambered heart. That's the characteristic feature of fish. And later 3-chambered heart. That's the characteristic feature of frog, reptiles, like that. Okay, so like that gradually it is evolved into like 4-chambered heart as it has to be in the mammal. Okay, like that only every time, whenever our characteristic features we have to get, and before that we are showing the ancestral characteristic feature, it seems. Okay, that's why he confirmed, yes, ontogeny means our birth history repeats phylogeny, evolutionary history, like that he framed. Actually, it's valid. And some more examples also he considered. You know, right? The frog larval form is tadpole. Okay, tadpole actually, the larval form of fish, larval form of amphibian shows the characteristic features of fish, it seems. Its ancestor. Okay, and caterpillar, it's actually butterfly larva. Arthropod characters it has to show, but it has shown the characteristic features of annelida. Its ancestral characteristic feature. Are you all getting? I am saying tadpole being an amphibian larva, it is showing the characteristic features of fishes, and caterpillar being arthropodon showing the characteristic features of annelidans. Okay, so that's some larval forms also he considered, not only embryos, larval forms also he considered, and he told that ontogeny repeats phylogeny. Yeah, Van Baer said that how exactly these embryos, larval forms can show the characteristic features of adult. Now caterpillar, it's the larva, but showing the body appearance, everything like adult annelidon. So how can actually some notification, is there limited time offer? Okay, fine. I don't need it. Fine. He said that, Van Baer said that embryos, larval forms never pass through the adult stages of other animals, like that. Okay, so he noted that Van Baer said that embryos, larval forms, how can, if you are calling that as embryo, you are calling that as the larval form because it doesn't have advanced characteristic features like adult. And how can one embryo, one larval form show adult characteristic features, the previous adult characteristic features? If it is showing adult characteristic feature, call that as the adult only. Why are you calling that as the embryo or larval form? So like that he said that embryos or larval forms never pass through the adult stages of other animals, other previous ancestral animals. So whatever he framed is wrong. Like that he disproved biogenetic clara, ontogeny repeats phylogeny, which was explained by Ernest Haeckel. Van Baer rejected that, saying that the embryos and all will never show the previous ancestral history. Might be it's a coincidence. He said that it's a coincidence, like that. Okay. So next we'll move on to homologous organs and analogous organs and comparative anatomy and morphological evidences. And before that, I'll just give you brief information about the schematic representation because it's about fossils, no? Paleontological evidences related schematic representation it is. Okay, so here Brachiosaurus. See, actually, certain reptiles only evolved into birds, mammals, like this. You know that early reptiles only evolved into modern reptiles, birds and mammals. Okay, so in this chart about dinosaurs mainly they highlighted, see, Brachiosaurus you are seeing, Tyrannosaurus you are seeing, Stegosaurus, Triceratops, Pteranodon, P silent, so Pteranodon, Crocodilian, Archaeopteryx, like this. Among all these children, remember, among all these, only crocodilian group is existing. The remaining all extincted. Okay, and one more, among all these, except Archaeopteryx, the rest are like reptiles only. Archaeopteryx only bird it is. Okay. It is categorized under aves. Remember this. Being here, then don't consider Archaeopteryx as a reptile. It is Archaeopteryx is actually connecting link between reptiles and aves. Okay, connecting link or missing link also, it's because it's fossilized, no? So you can consider that missing link also. It is between missing link or connecting link between reptiles and aves. Okay, but it is classified under aves. Among these, that's the only bird with teeth. Okay, let me tell you some points about Brachiosaurus. Brachiosaurus, see, appearance wise, long neck is there, long tail is there, forelimbs are also longer than the hind limbs, and herbivorous dinosaur. Okay, from this type of dinosaurs, I'm not saying exactly Brachiosaurus only evolved. I'm not saying like that. This type of, like, lot of group of dinosaurs evolved into, like, with the variations accumulated and inherited next generation after so many years later, Tyrannosaurus, this type of dinosaurs are evolved. Okay, it's not, you should not understand the representation like directly Brachiosaurus only evolved into Tyrannosaurus. They did not say that. This type of dinosaurs evolved into Tyrannosaurus, this type of dinosaurs. Of course, Tyrannosaurus is the largest carnivorous dinosaur. Okay, and it has dagger-like teeth, 20 feet height, and very aggressive, very dangerous also. Okay, that is Tyrannosaurus rex. Now coming to Stegosaurus. Stegosaurus is also one of the herbivorous type of dinosaurs. Okay, you can identify Stegosaurus with its bony plates present on the dorsal side. Okay, and horny teeth, horny teeth are there, and also tail is also present. And based on this, you can identify this type of Stegosaurus type of dinosaurs. Not exactly Stegosaurus. This group dinosaurs evolved with the variations and finally evolved into Triceratops. Actually 3 keratops. Keratops means horns, okay, on the head portion. Like 3 horns are there. So that's why as 3 horns are there on the dinosaur, we consider this as the Triceratops. These 2 are herbivorous only. Okay, here I said this is herbivorous, but Tyrannosaurus is carnivorous, but Stegosaurus and Triceratops, both of them are herbivorous only. Okay, next, Pteranodon. Pteranodon actually, it's the flying reptile. Flying reptile, it's not dinosaur. Okay, flying reptile. In the statement, suppose if they give Pteranodon is a flying dinosaur, it is wrong. Okay, that much clearly I am saying. Flying, it's not flying dinosaur, it's flying reptile, Pteranodon. Okay, crocodilian is actually existing group. Archaeopteryx, I already told you, classified under aves, but actually considered as the missing link between reptiles and aves. Okay, children, and this is the bird with the teeth. Yeah. And next, yeah, important concept. Like earlier, they used to give definitely one question in the evolution means actually homologous organs and analogous organs. This concept definitely will be there in the final exam also. I hope this year also it will be there. Okay, let us recall. Very simple concept. Is it easy to score also in the final exam? Homologous organs. Homologous organs, see, like a set you remember, I'll just write here. Homologous organs. Divergent evolution. Common ancestry. This is one set. Okay, let me explain about it. Homologous organs have similar origin and similar structure, but have different functions. I repeat once again. Similar origin and similar structure, but have different functions. Okay, when it will happen means when organisms undergo divergent evolution, definitely those organisms show the presence of homologous organs. See, let us assume in this is one particular habitat. Okay, here like one organism has many descendants. Descendants means offsprings you can consider. Okay, these offsprings, maybe for food, they couldn't tolerate the competition for food in this environment. Otherwise, maybe environmental pressures, they couldn't tolerate the temperature over there. So the descendants of this ancestor, actually this organism, migrated to different geographical area. One offspring moved to a grassland, one offspring moved to a mountain. Let us assume one moved to some other environment, the deep, dense forest or something. Yeah, like that. 4 different habitats I created here. So from the descendants of one organism migrated to different geographical areas. Okay, it's like when a big family you imagine, like combined family you imagine. Okay, so all the offsprings, like maybe I have some 5 children or some 5 children. So one of my children moved to Australia for education purpose, and the second one moved to America for job purpose, and third one married someone and went to Australia, and someone settled in somewhere in Europe. Okay, like that, different places. Okay, like maybe like years passed, and they also grown up and settled there itself. They did not turn back. Okay, I did not go that side. After so many years, maybe not my generation, I also got expired, and after that maybe next generation, after like in a function or somewhere later, they all gathered. Means, will it be having different structure in them? No, right? Because they all actually belong to one family only. Definitely skin texture or the way they think and the way, like internal anatomically, nothing will be changed because genes are common. Okay, maybe their lifestyle is different. The food which they eat and food which they eat and the way they speak, okay, dressing style, all that may vary, but internal. I'll try. Yeah. So internally, the anatomical structures will not be changed. Only our behavior and all will be changed because you speak in different way, different slang of English if you settle in America, and the one in Australia speaks in different way. Like that, slang will vary. But the way they think and all will not change. The same here also, like from one particular organism, descendants actually migrated to different habitats, and when they settle over there and trying to get adopted to that particular geographical area, their feeding habits change, okay, and the living style change and competition pressures change. So because of all that, they evolved new characteristic feature, and they appear as different species. Okay, this is actually divergent evolution. Hope it is clear. Like so whenever divergent evolution is noticed in the organisms, definitely as ancestry is same, common ancestry is there, so definitely they show the presence of homologous organs. And see, that's why I said homologous organs, divergent evolution, common ancestry. These three, like a set you remember, like examples like development of mouth parts in insects. Insects mouth parts is one example for this. And here they had given forelimbs perform different functions in these animals. Like mammals you can take, they have similar anatomical structure, like whales, bats, cheetah, human. Okay, all these forelimbs have similarities, like anatomical similarity, like having humerus, radius, ulna, carpels, metacarpals, phalanges in their forelimbs. Okay, and heart and brain development also you can consider. Precursors are same for that. So heart and brain development also example for homologous organs. Okay, next we'll move on to analogous organs. Analogous organs, convergent evolution, different ancestry. Like I said, you mention it. Okay, analogous organs. Analogous organs. Convergent evolution. Different ancestry. These three you remember as a set. Okay, so this is analogous organs means they have similar functions. Organs perform similar functions, but origin and structure may not be similar because ancestry is different. Okay, it's like, see, one organism like from one habitat. Yeah, let me consider this as the grassland and mountains, forest, and some other habitat, let us take. Okay, mountains like that. And from here, some reptiles migrated to one geographical area. And from this one and some other mammals, actually elephant or something, migrated to this particular area. And here some human beings, and here some birds. So like that, birds, mammals, reptiles, human beings, different organisms. Okay, let us take our own campus now. So now currently I am in Penamaluru. So you might be in Poranki, you might be in Ganguru, or whichever campuses let us assume. Okay, the rodents, which were rats, which you are seeing in your rooms, are migrated from some other area. Their mother and father might be in some other place, and they would have migrated, expecting a lot of food will be available in your hostel rooms. Okay, and these crows and all, which you generally find in surroundings of the mess and all that, maybe their native place is different, migrated to, attracted to this particular campus for food or something, the noise or whatever, and they want to prepare for Neet exam, we don't know. And human beings like you all, like me and you, I came from some place and you came from some other place, your natives are different, but we all gathered in one particular habitat. Like that, now though reptile it is, or though insect or whatever, the finally all gathered in one particular habitat and facing the same environmental pressures. This much temperature in Vijayawada now we are all facing. And you don't think that only human beings are facing. A cockroach is facing, a lizard is facing, a rat is facing. Every organism living in that same environment is facing the same environmental pressures. At that time, no, we, like our ego may not accept, but actually we all are undergoing the evolutionary processes, and we get definitely some similarities to tolerate all this. That's why you are surviving here. However, you are surviving here, the same way rat is surviving, cockroach is surviving, the mosquitoes are surviving. So some similarities will be there. Okay, so this type of evolution is called as convergent evolution. We see from different habitats, organisms converged in one particular geographical area and trying to get adopted. Okay, so by having similar features, so this is only called as convergent evolution. Okay, here different ancestry may be there, but they have the similar feature to perform a same function. Function is same. Structure, origin, all are different. This is about analogous organs. Hope it is clear, children. Analogous organs, convergent evolution, and different ancestry they have to have. Okay, and see polar areas. That's the best example. I think you can understand. Polar bear is there. Some birds also used to live in polar areas. Wolf is there, and some other human beings are also can be there. So all definitely to bear that extreme cold environment, we all have to have it. Human beings and other organisms, though it is a reptile, though it is a bird, it has to have thick fur, a thick layer, outer layer to bear the extreme cold environment. That's a common similar characteristic feature it has got to bear that low temperature. Okay, this is about convergent evolution. And examples here: wings of butterfly and wings of birds. So complexity, structural complexity is different. Butterfly is very simple, and bird, like feathers and all, but they both perform for same function like flying only. And next, eye of octopus and eye of mammals, flippers of penguin and flippers of dolphin, and sweet potato, which is a root modification, potato which is a stem modification, and both are useful to store the food. So these are the examples of convergent evolution. Okay, children. So now, so generally, adaptive radiation concept also next we will discuss. Adaptive radiation in general sense, actually that is nothing but divergent evolution only. Okay, divergent evolution only. And but when you have to consider adaptive radiation as convergent evolution, I'll let you know after some time. Yeah. This paragraph is about biochemical evidences. So here they mentioned about proteins, genes and all in certain organisms. Like if you want to do research or something, like when you gather the information related to proteins, like my the blood, whatever proteins are there from my blood you extracted, and from rat I extracted, rabbit also, something else. Okay, so suppose similarity is more in these proteins, structure, function, more similarity, more genes are connected connected, like chimpanzees and human beings. 98.4% or some that much amount of genetic composition between human beings and chimpanzees, it is common. So like that, then we assume that the more similarities are there in like having proteins and genes between two individuals, two groups of organisms, then we assume that yeah, they will be having a closely related ancestor. Okay, so we link with the ancestral history. Yeah, more similarities with the proteins and genes, then we connect like more similarities of the ancestral history will be there, like that. That's how biochemical evidences also act as the evidence for the evolutionary history. Okay, this one they said about artificial selections. Actually, they want to say that yeah, in millions of years, definitely this much diversification would have created because of evolution, and artificially when man is doing it, but within few months or within few days, he could create a new species. Why can't nature create new species? Like that, to make us to understand about the evolutionary history, actually they highlighted this paragraph here. Artificial selection examples, like in the plant breeding, it is there, animal husbandry, you know, like the varieties of organisms, cattle and varieties of cows, buffaloes, and the other dog species also in other they are producing, and horticulture, agriculture, they are taking the, and even sports, security purpose also, they are taking the benefit of all these artificially produced organisms. Okay, and next, industrial melanization. I think hope you are comfortable with this. Before industrialization and after industrialization, the story explains about the peppered moth. Peppered moth means Biston betullaria. Scientific name of the peppered moth is Biston betullaria. Biston betullaria. And here two variants of the peppered moths were there: dark winged moths and white winged moths. Okay, and around 1850s, like that, no industries in that particular area, no pollution at all the tree barks were covered by lichens, light color. So that time, whenever white winged moths sit on top of it and the background matches, so birds, predatory birds, you were not able to trap them, actually capture them easily. When dark winged moths sit, then easily it used to appear, and the bird used to pick them up and kill them. So like that, at that time, before industrialization, like white winged moths number was more than the dark winged moths. And after industrialization, after 1920s, if you see, dark winged moths were more than the white winged moths. That's because of increased pollution. Okay, because after that, industries increased in that area, a lot of smoke and the pollution actually deposited on top of the bark. Lichens died because lichens are very sensitive to pollution. That's why lichens were not there, and the bark was dark, and that background supported the dark winged moths but not the white winged moths. So this story you remember as the example of natural selection. That's very, very important. Industrial melanization is an example of natural selection. Okay, let us be very clear. Remember this as the example of directional natural selection. And specifically, okay, natural selection in general, but which type of natural selection means directional. Okay, and not only that, dear, see here some more examples are there. Herbicide resistance, having the resistance of pesticides, antimicrobes, having antibiotic resistance, okay, DDT resistance, all these examples you can consider as the examples of natural selection, and directional only mostly you consider. Okay, and when situation they create a story or something, that might be like different term we have to apply. Otherwise, in general, in your level, they focus like directional natural selection only for this. Okay, and man is involved. Like industries are constructed by man. Because of that only, the direction of evolution. White-winged moths were like enjoying nicely in the habitat. That time, who expected that industries will be constructed and pollution increase in that particular area, that will kill them more. So like that, no one will think that after a certain period again dark winged moths will be favored. Okay, So actually indirectly, like human beings are involved, so that's how evolutionary direction is changed to some other species. So that's why all these examples are considered as based on the anthropogenic actions. Anthropogenic actions means where man is involved. Everything like industrial melanization, herbicide, pesticide, antibiotic resistance, DDT resistance, all the examples are anthropogenic actions only, natural selection examples. And based on the given story, they summarize the point here that evolution is not a directed process in the sense of determinism. Evolution is not a directed process in the sense of determinism. It is a stochastic process, and stochastic process means like it's not confirmed. Like you can't, evolution is not a directed process. Like you can't say that yeah, definitely another 100 years these white winged moths only will be there. You can't say. Okay, maybe whenever you see something is dominating in the area, you may say that another 100 years definitely it will only dominate like that, but we don't know tomorrow what happens. Okay, so you can't predict anything in the evolution. It is not a directed process, and it's chance based, like based on the events happen in the nature, based on the mutations happen in the organism, definitely the direction of the evolution will be changed. That's the meaning of stochastic process. Anything, the event which happen in nature or mutation happens in the organism can change the direction of the evolution. Children, okay? So shall we move on to adaptive radiation? Yeah, many children like suffer to identify whether this example is like divergent evolution, or what to say example is for convergent, like that. Let me give you clarity about it. Darwin actually 3 years of his research he did in Galapagos Islands only. Okay, so he noticed small black birds, very small, tiny black birds. He noticed 18 species of Darwin finches. Okay, 18 species he noticed. But you know, all are from like they have common ancestor only, and that common ancestor also originally seed-eating bird. Okay, but they migrated from one particular area. Earlier I told you this story, like from one particular area, organisms migrated to, this is the area, this ancestor bird which is seed eating, this bird is here, and the descendants, the many descendants migrated to different geographical area. One went to desert, one went to mountain, one went to grassland, one went to forest. Okay, different geographical areas they entered, and after entering, like the same whatever mother bird used to give, that food only will not be found. Now might be in your home you might be eating some breakfast, but after coming to Gosalites you have to eat idli only. Okay, so like that, the once after the descendant migrated to different areas, definitely the feeding habits vary. Okay, accordingly, so over a time period, as their feeding habits are changed, even the beak sizes also accordingly changed. Okay, shape of the beak is changed and feeding habits are changed in these birds over a period of time. This is how different species are formed. This is the best explanation about adaptive radiation. See, process of evolution of different species. Once in weekend exam I gave a question keeping the similar species here. Okay, it's not similar species, it's a different species in a geographical area, in a given geographical area, starting from a point, means starting from one geographical area, literally radiating means divergence, radiating to different geographical areas, and after going to that geographical area, what is happening? Evolved into different species. This is called as adaptive radiation. Actually, most of the cases adaptive radiation is actually divergent evolution. The best example for adaptive radiation and divergent evolution is Darwin finches. Okay, Darwin finches. Anything dear, like see, hostile marsupials picture is given here. One group, if they are explaining, like see, now common ancestor is there and migrated to different geographical areas and evolved into different organisms. Wombat is there, kangaroo, marsupial rat, banded anteater, tiger cat, Tasmanian wolf, sugar glider, okay, marsupial mole, koala. So like this, so many organisms, bandicoot and all that. So one ancestor only, common ancestry is there. Common ancestry is there, and different species are evolved based on the migrations, means that is like divergent evolution. So let them give like a single example of Darwin finches, otherwise Australian marsupials, otherwise placental mammals. Single example if they give, That's like adaptive divergence only, means adaptive radiation and divergent evolution. Hope it is clear. Suppose like my question is like Darwin finches evolved into 18 species. So which type of evolution it is example for? Divergent evolution, adaptive radiation only. Okay, same like in place of Darwin finches, let me keep Australian marsupials. Let me keep placental mammals. That time also it is all adaptive divergence only. When you have to consider this as the convergent evolution, means when more than one adaptive radiation is happening in the same geographical area. Okay, now see, we are there in this campus, and definitely evolution is not like it doesn't have full stop. It is a continuous process. It is happening. Okay, you are undergoing evolution, and parallelly with us only, like in this campus, rats are there. Rats are also undergoing evolution. Don't think that only human beings undergo evolution. It's not like that. Okay, so nature is not partial. Okay, so rats are also undergoing evolution. Cockroach is undergoing evolution. Bird is undergoing evolution. Snake is undergoing evolution. So evolution is happening. Like yours is, if you consider ours as adaptive radiation, you came from your native. Okay, migrated to one area and settled here. So this ours is one adaptive radiation, bird related one adaptive radiation, snake related one adaptive radiation. So more than one adaptive radiation is happening in the same geographical area. Then you have to consider that as the convergent evolution. See here. See here, that's why they compared, like in Australia, in certain geographical areas, placental mammals and Australian marsupials live in the same geographical area. See, they are very closely related. Both are mammals only actually, but placental mammals are more advanced. Na? So placenta is there to nourish the young one, to take care of the young one and all. And Australian marsupials, marsupium is the pouch actually, pouch-like structure. The placenta is not, the little lower to the placental mammals. These are not so developed. Okay, and see, these are living in one type of geographical area in Australia. So undergoing the evolution. Okay, this is considered as more adaptations are happening in one area, right? So this comparison will be the example of the convergent evolution. Hope everybody is clear. So one single example is given, like in the geographical area, like only Darwin finches or only Australian marsupials, only placental mammals. Then you can consider that as the adaptive radiation as divergent evolution. When many species, like many adaptive radiations are happening in the same geographical area, that time like in comparison, or human beings and birds, and human beings and snakes, otherwise like these two, like that, if the comparison is given, then you consider that as the convergent evolution. No confusion here, right? Yeah. So this tabular column is like memory based. Even I'm telling you, these names, all these names also you have to remember. Like suppose if I say Australian marsupials, then you have to list out every organism. Okay, sometimes we used to give the questions like based on by giving this picture also, like identify the picture, and wombat you need to identify, and you need to identify that as the Australian marsupial also. Okay, like that, the memory based video. So 2-3 revisions you have to have, and this is all for short-term memory. So before final exam also, a day before, you have to refer definitely. Otherwise, no, humanly it's impossible to remember this for anybody, even for me also. Then next comparison, and placental mammals and Australian marsupials, like mole of the placental mammal, because they are living in the same environment, same environmental pressures, same competitors, etc. So over a period of time, as both are mammals only, this placental mole, appeared like marsupial mole. Similarities are there. Anteater to the numbat, mouse to the marsupial mouse, lemur, spotted cuscus, flying squirrel, flying phalanger, bobcat, Tasmanian tiger cat, then wolf, Tasmanian wolf. Like that, you have to remember. So lemur, as per my knowledge, lemur mostly they focus. Lemur, placental mammal. You need to remember all this. Lemur means immediately you have to recall, oh, this is a placental mammal. Okay, immediately it is compared with what in convergent evolution? Spotted cuscus. Okay, anteater, numbat. Okay, bobcat, Tasmanian tiger cat, wolf, Tasmanian wolf. Tasmanian is there, then go with the Australian marsupial, something like this. Okay, so I think revise today itself, today afternoon again, so it will be clear for you. Okay, this is about convergent evolution. Yeah, here this in concept also for certain people it will be like khichdi only. So a lot of statements are there, but very beautifully written. I have to appreciate these people. Yeah, here they highlighted Darwin's theory. Actually, the essence of Darwinian theory about evolution is natural selection. The rate of appearance of new forms is linked to the life cycle or the lifespan. The rate of appearance of new forms is linked to the life cycle or lifespan. Suppose organism's life cycle is so simple, lifespan is so short, like insect lifespan or something, then they definitely like will be having the next generations faster, so variations will be inherited faster, and evolution you can see like new species formation, you can see faster. So the organisms with a simple life cycle and shorter lifespan, in them actually the faster rate of evolution is noticed than the organisms with longer lifespan like us. Okay, so here one example about microbes they had given, like to say that which when you can consider the organism is more fit to be in the geographical area like that. Suppose recently I saw in the Ganguri campus that because of high temperature, when ACs are not working, like they couldn't sit outside actually. So very few actually, only 1 day, 2 days, they couldn't bear, and some sensitive children will be there, no, they went to home actually. So who tolerated the temperature stayed in hostel, we have to say they are more fit. Otherwise, who couldn't bear the condition and escaped out, so they are more fit. Whom we have to consider, whoever tolerated the adverse condition and stayed over there can be considered as the more fittest organisms actually. So even in evolution also they say that, so nature selects for fitness. Okay, so nature selects for fitness. How that tolerable characteristic feature they would have got based on the characteristic feature. That fitness you have, it's because of your characters. Okay, how you have got that character? Because of genes. How you that genes like inherited. So they finally concluded saying this the characteristic feature which is giving you more fitness to be in the geographical area is inherited. It is inherited means definitely it has got genetic basis. Okay, so they concluded saying that adaptive ability is inherited. Adaptive ability is inherited. Children, this type of statements only they ask in the exam. So be more focused and underline that particular statement. Adaptive ability is inherited. It has a genetic basis. Fitness is the end result of the ability to adapt and get selected by nature. So when you are getting adopted more better way, then that is only called as you are fit to be in the geographical area, and that because you that characteristic feature you have and able to tolerate it means that is only actually like the natural selection also. And Darwin actually gave priority always for two key concepts. According to Darwin, what are those two key concepts? Branching descent and natural selection. Branching descent and natural selection. Okay, branching descent, here you can mention, it speaks about common ancestry. It speaks about common ancestry means from one ancestor or descendants will receive certain variations, like again from descendants to the next generation, if you see all the variations are accumulated and get inherited. So like that only over a period of time, like a new species also can be created. Okay, so that is about branching descent, and if positive characteristic features are there, that will be selected by nature and they get the comfort in that particular area. Reproductive fitness also he spoke. No? Then next about Lamarckism. Compared to this, we consider Darwin as the father of evolution, right? Actually, Darwin, exactly, he is the father of evolution. He wrote a book also, Origin of Species, and popular for his Darwinism, this natural selection based explanation and all. But before 100 years, before like 100 years before to this Darwin, Lamarck actually the most senior person like who entered into this research. Okay, and he's a French naturalist. Lamarck is a French naturalist. He wrote a book also, Philosophy Zoologic. The name of the book is Philosophy Zoologic. This in Lamarckism, actually two postulations mainly you remember. One is like use and disuse theory. Use and disuse theory. Use theory. So here you can see, right? Yeah. First about use theory. It's for us to listen, it will be little funny, but he noticed in that way. Use theory says that if we use any organ like more and more, then that organ will be more developed, it seems. Like currently the brain or whatever, if you use again and again more and more, then it will be developed. Suppose if you don't use, according to Lamarck, not according to me, okay, according to Lamarck, if you don't use any organ, then that will get disappeared for the next generations, it seems. Okay, so like that, that's why be careful, use it proper way. Okay, then so use theory example according to Lamarck is like the elongation of neck in giraffe. Giraffe earlier was like goat, dog sized only, and that much short neck only. Like as that time competition was more for grass, and it was not able to get sufficient food, and started stretching its legs and also neck to pluck the leaves from big trees and all that. Then that helped them, as it is using this neck, legs and all that, elongation happen in those areas. Okay, so use theory, example, elongation of giraffe neck. Disuse theory, like if you don't use organs, it will be lost, I said, na? So disuse theory example is like loss of limbs in snakes. Snakes are tetrapods, means limbs are there, 2 pairs of limbs are there in snakes also. But actually, if you see the snakes' limbs, they were not using them for the crawling locomotion. That's why the limbs are lost. Okay, and one more postulation, he said, okay, use and disuse theory some people accepted, but more rejection Lamarck had got for inheritance of acquired characteristic features. Inheritance of acquired characteristic features. He said that whatever characteristic feature you get in your lifetime, that gets inherited to the next generation, it seems. Okay, suppose like we met with an accident or loss of some forearm or something, then the next generation, like your son or daughter, like are born without hand. Do you accept this? No, right? So that's why Lamarck has got more rejections for this particular theory only. Here it is mentioned inheritance of acquired characteristic feature, because he noticed whatever examples he had taken for use and disuse theory, like snakes losing the limbs, giraffe elongation of neck, it inherited to the next generation. So in that sense, he said that yeah, every characteristic feature, whatever they acquire, will be inherited like that. But actually, August Weismann opposed this very strongly, and he proposed, he's famous for his germplasm theory. Who is that? August Weismann. August Weismann. Okay, he is popular, this scientist is popular for germplasm theory. Germplasm theory. He said that any change happens in the germplasm, germ cells and all that, so that will get inherited to the next generation, not like every characteristic feature. Like what if change is happening in the somatic cells, so that will never get inherited, and if change would have happened in the germ cell, that can be inherited to the next generation. Okay, children, that's about Lamarckism and the inheritance of acquired character. This characteristic feature of this postulation of the Lamarck was strongly opposed by August Weismann, and who proposed germplasm theory. He's famous for his one experiment, like his 17 generations, he cut the tail of rats actually, and he noticed that the appearance of the tail in the offsprings. Okay, next. Darwin was influenced by one, I said, na, Alfred Russel Wallace. Wallace, same like Darwin, only Wallace also did research in Malay Archipelago Islands. And one more person, like Darwin was influenced by, is Thomas Malthus. Malthus is popular for his population-based, articles. And he said that, the Malthus said that resources will never increase, but population can increase exponentially. Okay, like that he said, and that point he considered, and how much our population is exponentially increasing during the favorable seasons. Okay, but definitely limit will be there by certain environmental pressures and competitions and all that. Okay, so next, here this paragraph is about Darwinism. What happened? Ma'am, sugar glider, flying phalange are same. Yeah, consider same only, dear, because in the chart also they had given the same, right? No problem. Yeah, so Darwinism. Hey. You are asking this one, right? Sugar glider and Flying, same only because they had mentioned like that, and we can't take the deviation from NCERT, na? So consider same only. Again, squirrel, if they mention, go with placental mammal, okay? If they consider the phalange or sugar glider, consider same only, Australian marsupials only. Okay, dear. Okay, I was explaining about this paragraph. Children, if you read this paragraph, it takes time, okay? So the summarized version of this particular paragraph is Darwinism points, postulations in sequence. So first you consider overproduction. Darwin first understood that if conditions are favorable, every organism utilizes maximum reproduction and produce the more number of offsprings. And second point is how much our population sizes increase. Definitely, what to say, due to accidents or diseases or sudden competitions, heavy competitions and all. Finally, again, population sizes will be limited. Constancy in population size. Population size. Population size. Okay, third one, struggle for existence. Struggle for existence. Fourth point is about variations, like positive variations, like that, selected by nature. So fifth one is about natural selection. See, this you remember in sequence only. Sequence should not be jumbled up, okay? Overproduction, constants in population sizes, struggle for existence. This struggle also Darwin explained within the species, intra-struggle and inter-struggle, inter-specific, like between two different species also struggle may happen for food and all, habitat, etc. And if organisms have positive variation, and definitely that type of species will take the benefit and win over the other species. That is about natural selection. And last point, postulation, is about origin of new species. Origin of new species. Okay, so this is about the Darwinism, overproduction, if situation is favorable, then constancy, like because of competition or accident, diseases and all, again limited population sizes are maintained, and they fight for the food and all that, so struggle will be there within the species, intra-specific struggle, and between two species, so inter-specific struggle, and variations, if they have the variation, positive variation, definitely they take the benefit out by taking the benefit with that particular variation. That's about natural selection. And finally, that leads to origin of new species. This is about Darwinism, and the same meaning is there. If you read that paragraph also, it's applicable for these short terms only. Okay, sequence is important in PYQs also once they ask this. And coming to mechanism of evolution. Here they compared the versions, two versions of Hugo de Vries and Darwin. Okay, we are not here to judge who is right or who is wrong, and both the views are right only. Okay, we respect their views, that's all. Like Hugo de Vries, he's popular for his mutation theory. First mutation term, it was termed by Hugo de Vries only in 1901. He did his research on Enothera lamarckiana. Enothera lamarckiana. Ma'am, which originate first, monocots or dicots in NCERT diagram? Yeah, dicots only, no? Origination first dicots only. Later on only the recent and more advanced ones are monocots. Anyways, I'll come to that concept here. And currently Hugo de Vries, yeah, Hugo de Vries actually proposed mutation theory. According to Hugo de Vries, mutations are random and directionless. Random and directionless. He believed always in the large number of mutations which are inherited suddenly, like saltation, single step large mutations. So de Vries believed in single step large mutations. He did not believe in transitional stages and all that as Darwin believed. Okay, no transition stages, just direct. If mutation happens, if that is inherited, that's all, evolution happens. Like that, Darwin said, yeah, it is also right. We are not rejecting that. It's correct actually. So, but Darwin said, always he believed, he doesn't know the term of variation. See, Darwinism, this much he did right, but he failed. Darwin's feedback also you have to remember. he said about survival of the fittest by considering the variations and natural selection and all, but he failed. Darwin is failed to explain about the arrival of the fittest. He spoke about survival of the fittest, but he couldn't explain about the arrival of the fittest, means the cause behind the variation, how this variation appeared, because he doesn't know the genic composition and undergoing the changes and all. The term only they don't know. What is gene, what is DNA, they don't know anything. Okay, so that's the reason Darwin explained about survival of the fittest, but he failed to explain about the arrival of the fittest, means reason behind the variation he doesn't know. And always he focused on small number of variations only, and he always said that definitely it will be inherited next generation to next generation, so directional. So these variations are small in number and they are directional. So always with the help of these variations, the slow and gradually evolution happens. Okay, but according to De Vries, evolution is sudden. Okay, with the help of saltations, means single step large mutations, but according to Darwin, what slow and gradual evolution occurs. Okay, children, so now Hardy-Weinberg equilibrium, right? Where did it go again? What's the time? 45. Hardy-Weinberg equilibrium. Yeah, many children are waiting for this concept. It's a mystery for them. Yeah. In a large population, allelic frequencies remain stable and constant. Okay, see, certain conditions are there for the Hardy-Weinberg equilibrium to follow. Okay, they consider always large population only for it. It's not applicable for smaller population here. The conditions which are applied for Hardy-Weinberg equilibrium to follow. So first one is population has to be larger only. Larger population. Large population. And random matings have to be there. For this Hardy-Weinberg equilibrium to maintain, random matings have to be there. Random matings. Otherwise called as panmictic matings. Panmictic matings. Random matings only should be there, not specific. And no evolutionary forces. Based on this only you can expect questions. No evolutionary forces applied. Like no evolutionary forces applied means no natural selection, no mutation, no migration, no genetic drift, no gene flow, etc. Okay, all these factors should not be operated, then only Hardy-Weinberg equilibrium can be maintained. This is very, very important. Based on these questions will be asked. Okay, it has to be larger population and random matings have to be there and no evolutionary forces should be operated in that particular geographical area. So now coming to the principle. The principle says that allelic frequencies in a population are stable and constant from one generation to next generation. So at that time, the allelic frequencies are constant, definitely gene frequencies are also constant. Okay, if gene frequencies are constant, then gene pool is constant. Gene pool is constant. Gene pool means total number of genes and their alleles in a population. Definition is important. What is gene pool? Total number of genes and and their alleles in a population, not in individual. Okay, so that is called a gene pool. This is called a genetic equilibrium, and at that time when genetic equilibrium, everything is constant, alleles frequency, gene pool of the population is constant. At that time they said that Hardy and Weinberg, two different scientists, they said that sum total of all the allelic frequencies is equal to 1. They said sum total of allelic frequencies is equal to 1, means P plus Q is equal to 1. Let me explain about this. Suppose randomly one gene, like maybe it is coding for the characteristic feature like your skin color, your eye pigmentation, something like that. Okay, any randomly one gene you take. For that particular gene, for any gene, Consider two alleles, like dominant allele is represented with capital letter and recessive allele is represented with small letter. Okay, capital A, small a. Okay, this is what dominant allele I am saying. Dominant allele. Small, like this one, small letter A is actually recessive allele. Recessive allele. Children, see here. And how many out of 1,000 members in a population, how many members, have this dominant allele, this particular characteristic feature? If you could identify, that gives the frequency of that particular dominant allele. Like see here, you are saying about the dominant allele and recessive allele. How many members in a population have that particular allele? That is about the frequency, their frequency. So now I am mentioning capital P as the dominant allelic frequency. Dominant allelic frequency. Q, small q as the recessive allelic frequency. Recessive allelic frequency. Okay, so now they said that sum total of the all allelic frequency is equal to 1 when gene pool is maintained constant. Means P plus Q, dominant allelic frequency plus recessive allelic frequency is equal to 1. Okay, so now this P plus Q is equal to 1. Can we write this as P plus Q whole square is equal to 1? Same only, right? Doesn't change. And binomial expansion of this equation is P square plus 2PQ plus Q square is equal to 1. What is P square here? P square is actually homozygous dominant. Now you can't refer this as allele because P square, two alleles are there. That's why it is a homozygous dominant genic frequency. Genic frequency. Then Q square, homozygous recessive genic frequency. Okay. Next, 2PQ, heterozygous, 2PQ based calculations also they ask. So, heterozygous dominant genic frequency. Children, is that fine? So P square is homozygous dominant genic frequency, Q square is homozygous recessive genic frequency, 2PQ is heterozygous dominant genic frequency. Okay, this is the equation which if P square plus 2PQ plus Q square is equal to 1, this is only Hardy-Weinberg equilibrium. Okay, when no evolutionary forces operated and population is large and all are undergoing random matings only and same age group individuals are there, same type of reproductive method is followed. At that time, according to them, gene pool is constant and genetic equilibrium is maintained at that time in that particular population. Okay, and we'll solve certain calculations also based on it. I'll show you that. And first we'll discuss about the factors which deviate Hardy-Weinberg equilibrium. Before that, 2-3 lines I'll show here, which you face very tough to understand. When frequency measured differs from expected values, the difference indicates the extent of evolutionary change. Means suppose if it is not P plus Q is equal to 1, any different value is there, then you can understand the equilibrium is not there and in that area evolution is happening. Okay, and the same thing again they wrote, disturbance in genetic equilibrium or Hardy-Weinberg equilibrium, like a change of frequency of alleles in a population would then be interpreted as resulting in evolution. Okay, remember that. Then there are 5 factors which disturb the Hardy-Weinberg equilibrium. Those are gene migration or gene flow, genetic drift, mutation, genetic recombination and natural selection. These are the 5 factors which deviate or which disturb Hardy-Weinberg equilibrium. Listen carefully, children. The writing every line, this is a big chapters, no? So it takes time. Gene migration or gene flow means the exchange of genes between 2 different populations Those two different populations belong to same species, like human beings to human beings, but maybe present in different areas, like that. Okay, the exchange of genes between two different populations, okay, belong to same species. That is called as gene migration or gene flow. When this the movement of genes or exchange of genes, this will happen during immigration, emigration, and sometimes like marriages time. Okay, like that you can consider immigration, emigration. Okay, better, like we can understand it and we can connect it to those terms very easily. Okay, so gene migration or gene flow means movement of genes between two different populations belong to same species. It may have immigration will add the genes to the population, and emigration will remove the genes from the population, like that they explained here. And one more line. If gene flow, like if there would be a gene flow, if this gene migration happens multiple times, gene migration number of times if it is happening, that is termed as gene flow. In that gene migration, gene flow, only little variation he created. Like suppose Tirumala type of pilgrimage centers, if you see, like continuously lakhs of people will be entering to that particular area and moving out also. Like repeatedly people will be going and coming out, going and coming out. So repeatedly, if this movement of genes between two different populations happen, and that gene migration repeatedly if it happens, then that itself is termed as gene flow. Okay. Next, genetic drift. Yes, interesting topic, genetic drift. Here only I'll mention. Yeah. Actually, genetic drift, it's a non-directional selection. One scientist, Sewell-Wright, he explained about it. Sewell-Wright hypothesis it is. Okay, genetic drift, non-directional selection, and it is two types. One is about founder's effect, and another one is about bottleneck effect. Bottleneck effect. Okay, first about founder's effect. This is there in the syllabus, founder's effect. Okay, so here one large combined family, let us take large family, let us take. Okay, and from here maybe because of their work or something, some individuals, very few individuals came out of that particular population. I consider this as the large population. Few individuals, very few individuals moved out of the large population. These people are called as founders. So what do we call like founders? Who are founders here? The few individuals who moved out of large population are called as founders. These founders, suppose they enter into a new area. Okay, they enter into a new area. Let me mention here. So now over a time period, if you see in this particular new area, their characteristic features, the founders' characteristic features only would have been inherited in their offsprings and all that in their next generations. But you can't see their grandparents' characteristic features. It's maybe in the beginning, in one or two generations, maybe grandparents' characteristic features also we can notice in the population. But over a time period, if you see, stabilization happens for the founders' characteristic features only, it seems. And this is proved, and a factual story also it is there mentioned about the Red Indians population. Actually, we always consider founders effect, no? That now, even now also, if you go that area, you can notice that. And Red Indians population, when they were in all the Red Indians in the larger population, that time the blood groups alleles are IA, IB, IO. Okay, means you can see the individuals with A blood group, B blood group, and O blood group when they were in the large population. Randomly, some people came out. Okay, those founders occupied a new area. Now, if you go to that island and see, all these people are with O positive blood group only, it seems now. A is not there, B is not there. Entire area is with O positive blood group only. Okay, so diversity is reduced. See, in the weekend papers in the revision session they asked, no? So in the genetic drift, actually in the founders effect or bottleneck effect and all that, the variations may be reduced, diversity is reduced compared to larger population, like when 3 alleles were there in the large population. But in the founders effect, in the founders population, diversity is reduced or it is increased. IO allele only is there. Now one allele is only there. Here 3 alleles, but when it comes to this, only one allele is there. So like that, the diversity may be reduced, but their characteristic features only we are seeing here. This is founder's effect. So if this type of allelic frequencies are there also, then Hardy-Weinberg equilibrium can't be maintained. Okay, and bottleneck effect says that bottlenecks are, it's here, it's not mentioned, but okay, we can understand. Bottlenecks are nothing but natural calamities, earthquakes, floods, tsunamis, like that. Okay, so whenever this type of natural calamities occur in the area, massive deaths will happen. Majority will die. Very few lucky survivors will be there, and their characteristic, if they marry, okay, their characteristic features only will be inherited to the next generation. Okay, like that, the allelic frequencies will change at that time also. Okay, this is about genetic drift. Hope it is clear, children. And you remember the definition of genetic drift is very, very important, and sometimes in entrances also they asked. In a small population, allelic frequencies are changed by chance. Okay, I repeat once again. In a small population, allelic frequencies are changed by chance. It is called as genetic drift. Okay, by chance. This is the key term to identify the definition. It can be founder's effect or what to say, bottleneck effect. Next, mutations. Here, these 2-3 lines are about mutations. Mutations, see, you know, this alternation of DNA sequences, suddenly it happens. Yeah, that may create the change in the genotype and later to the phenotype. That's how variations appear, and that creates a change in the allele sequence. Okay, that's how Hardy-Weinberg equilibrium will be disturbed. So these mutations, like antibiotic resistance you take or any other resistance, it is actually at that situation only it will not be created. So mutations already like pre-adoptive, means some before the situation only, like you would have got that particular resistance power or something like that. So they had given microbial experiment shows that pre-existing advantageous mutations when selected will result in observation of new phenotypes. Means because of that particular pre-existing mutation only, one resistance power or some characteristic feature you will get, and that will help you to survive better. Suppose you are traveling and you sat next to a chicken pox suffering person, okay, some other COVID person, and now nobody worries about COVID and all that, but even then, okay, we'll consider that. And after that, like in your discussion at that time only, after some time you came to know, after you exposed to that particular person, sometime you came to know that you are sitting with that particular person only, who is suffering from so and so disease. After going home also, you think about a lot, like you may also like get that particular disease, but a week is over, 10 days over, you did not get that infection. Understood that you are resistant to that particular disease. When did you get this resistance? When you sat next to that patient, otherwise already it was there in your body. Already it was there in your body, right? But because of that situation, you came to know that, you realized that you are resistant to so on infection. Okay, that's what I am saying here. Mutation, this resistance power or any mutation based character, already it would have been existed in your body, but because of the situation you realize at that time. Okay, so that because of mutations, characteristic features change, so alleles, because of that Hardy-Weinberg equilibrium will be disturbed. Okay, now coming to natural selection and recombinations. You know, genetic recombination during sexual reproduction, actually it happens during crossing over between maternal and paternal chromosomes. Yeah, that also disturbs. So 5 factors: gene migration, gene flow disturbs the Hardy-Weinberg equilibrium, and genetic drift, mutations, genetic recombinations, and natural selection. Very, very important. Natural selection, 3 types of natural selections are there. What are those? Stabilizing selection, centripetal selection, otherwise called stabilizing, otherwise called as centripetal, and this one is directional selection. This one, the third one, is disruptive selection. Disruptive selection is centrifugal selection. Centrifugal selection. Okay, coming to stabilizing selection. Stabilizing selection, whenever, this is all a natural selection only, in the geographical area, when environment is more stable. No much change, nothing. Environment is stable. Like today, tomorrow, and after some days, temperature is not much variable over there, and rain, humidity, air pattern, everything is almost the same only. So that time, stabilizing natural selection is operated in the geographical area in which the medium trait is favored, but the extreme characteristic features that will be eliminated. Meaning, say, let me be more basic here. For any characteristic feature, three variations are there. Like body height you take, body weight you take, or skin color you take, anything, any characteristic feature, majorly three variations. Two extremes and one intermediate. Two extremes and one body height. Two extremes is extreme tall and extreme short. Okay, and intermediate height also can be there, and weight also extreme obese, extreme thin, and medium weight. For any character, that these are the three variations. In this stabilizing natural selection, who is favored, like which trait is favored? Intermediate trait people are more in number because situations are favorable for that particular individual only with the medium trait. And the extreme characteristic feature individuals are eliminated. Maybe body height, let us take the example, or body weight, you take the example. In 2020 or some paper, they asked a question in a hospital in Singapore or somewhere. Newborn children, actually, if they see, some are with the 4.5 kg weight, like that. Newborn children are with 4.5 kg weight, like that. Okay, and some are with 2.5 kg, something like that, and some are with very low weight. Okay, and they said in that question, this is neat question actually, they mentioned that this overweight newborn offsprings and less weight, underweight people also died, it seems, but more survival rate is with the intermediate weight only. So which type of natural selection is operated in that hospital? So intermediate rate is favored, means stabilizing selection is operated over there. Children, okay? So example, body height, body weight. So here also extreme tall, extreme short people are eliminated from the geographical area, or you can consider they are less in number, but peak is for the mean trait or intermediate trait only. That's what that is about stabilizing selection. And next one is about directional selection. Here directional selection is like any one trait is favored at a time, only one trait at a time. That is not intermediate, definitely. It's only for extremes. Let me write over there. And only one extreme here. Only one extreme at a time. Only one extreme at a time is favored. What about stabilizing selection, dear? Mean character or intermediate trait. Mean trait is favored in the stabilizing selection. That's over. Body height, body weight are the examples. And directional selection, see the arrow mark. This arrow mark you need to notice. Okay. So any one extreme at a time is favored, but never for the intermediate trait. Suppose industrial melanization you take. Once white-winged moths were more in number, then after pollution, after industries were connected, after pollution, dark-winged moths were more. Okay, when white-winged moths were more, dark-winged moths were less. When dark-winged moths were more, white-winged moths were less. Like that only one characteristic feature, one extreme characteristic feature is favored. In which direct natural selection? Directional selection. Okay, examples. List out the examples. This is important. Industrial melanization. NCERT exemplar point also this is, and DDT resistance, antibiotic resistance. Okay, next pesticide resistance, herbicide resistance. All these are the examples for directional selection. Next, disruptive selection. See here in the graph you can see this is one extreme trait and this is one extreme trait. So both the extremes are favored, but not the intermediate trait. Okay, intermediate trait is not favored and both extremes are favored. It's exactly opposite to stabilizing selection. Stabilizing selection, intermediate trait is favored, but both extremes are less. But coming to disruptive selection, both extremes are favored, but intermediate is not favored. Okay, example, any adaptive radiation, Darwin finches you can consider. Darwin finches you can consider. In this type of environmental area, you know, geographical area, always environmental factors are fluctuating. Today one type of temperature, rain pattern and all, and tomorrow another way. What to say, the disturbances are very frequent. Okay, at that time this disruptive selection can be operated in the geographical area, and of course among the three this leads to evolution faster. This leads to evolution faster. Children, okay? So this is about disruptive, opposite to stabilizing. Centrifugal, see the arrow mark? Favoring the extreme peripheral characteristic features but not the intermediate one. Okay, example, adaptive radiation, Darwin finches I said. Yes. Some stories also they create, like while framing the question to ask the stabilizing, directional, disruptive, and like I think in one paper 2019 I think they asked the cows, like artificial selection based the breeding of cow. Okay, actually farmer selects, like farmer is doing artificial selection on the cow, it seems. So his low milk yielding capacity cow, because of this artificial selection, gradually became like highest milk yielding capacity related cow. Okay, so one extreme gradually shifted to another extreme. So what, only one extreme is favored over there. Earlier his cow was not giving much milk. After artificial selection, gradually milk production increased. So at a time only one extreme is favored means which selection? Directional selection. Okay. Children, shall we start this? A brief account of evolution. Yeah, the first life forms, cellular forms appeared on Earth 2 billion years ago. It's like 2000 million years ago. Okay, gradually the mechanism of how non-cellular aggregates of giant macromolecules could evolve into cells with membranous envelope is not known. That's what I said. The protobiont, the transition stage and all, they did not mention clearly. They said we are not known like that. Then some of these cells, they believed that after the cellular forms appeared first, okay, at that time they started releasing oxygen, and here you can mention that the first the cellular form which released oxygen in the early atmosphere was like cyanobacteria. They considered over there, and they thought that maybe some light reaction, like which generally happens in the photosynthesis, would have happened in those primitive cells, and that's how oxygen was released like that. And slowly the multicellular condition created organisms, animals started evolved. Okay, 500 million years ago invertebrates were originated. 350 million years ago jawless fish was originated. This is memory based, children. Okay, 500 million years ago invertebrates were originated, and jawless fish evolved around 350 million years ago. Seaweeds, few plants 320 million years ago, 320 million years ago. And always, like you consider, like first life originated in water only, and first land occupying organisms like plants. First plants were there, later only animals occupied the land. And fishes with the stout and strong fins could move on land and go back to water. And maybe continuously they were living in water. They want to expose something green on land. Let us eat so much over there. Maybe they got attracted and they came to land, but they couldn't survive, and they went back. So that also happened 350 million years ago only. Okay, and in 1938, a fish caught in South Africa happened to be a coelacanth, Latimerian fish. It is Latimerian, lobed fin fish. Okay, that is coelacanth, and this coelacanth they captured in 1938. Otherwise, they thought that that would have got extincted and all, but they understood that no, it did not get extinct. It is still existing like that, and they considered this coelacanth fish only the ancestor for amphibians. Old amphibians originated from it, and from these old amphibians only modern amphibians originated. Okay, old amphibians got extincted. From these amphibians, modern amphibians like frogs, salamanders and all, then reptiles also originated. And compared to amphibians, survival rate for reptiles is increased. Why? Because they had cladoic eggs. Cladoic eggs means thick shelled eggs. Reptiles had thick shelled eggs that protected, they gave more survival rate for the egg actually. It was not getting dried up with the sunlight and all, and more protection was the predators also. Oh, something is hard, I don't want, like that, the less eating of those eggs, it happened. So that's why more number of reptiles could survive. So turtles, tortoise, crocodiles and all, and the next 200 million years reptiles only dominated the earth. Same time when reptiles were dominating the earth, giant fawns or pteridophytes, they were dominating, and because of them only the dead plants buried, and finally coal deposits were created, and certain reptiles like what to say, they went back from actually first true organisms occupying the land were reptiles only, right, among the animals. So some reptiles from land went back to water, appeared as fish-like reptiles. Fish-like reptile is ichthyosaurus. Okay, yes, saurus, this suffix is there, so don't consider that as the dinosaur. It's just fish-like reptile only, it's not dinosaur. Okay, and ichthyosaurus, then it happened 200 million years ago. The land reptiles, among them the Tyrannosaurus rex is the largest dinosaur. Okay, and 20 feet height with had huge jaws, dagger-like teeth, and carnivorous. About 65 million years ago, people say it is a Cretaceous period, actually Mesozoic era, Cretaceous period. 65 million years ago, dinosaurs suddenly disappeared from the earth due to climatic conditions or lack of food. We don't know, the reason might be different. Okay, some say most of them evolved into birds. The truth may live in between, but small size reptiles of that era still exist even today. And the first mammals were like shrews. Okay, because even evidences are there, like fossil evidences are there for that. And mammals, as they are viviparous, and the young one is properly taken care by the mother inside the body. So that's why protection was more, and IQ was more, developed nervous system. Because of all these benefits, you know, survival rate gradually increased for mammals after reptiles. Okay, and at that time only continental drift also, shifting of the Pangea, means the soil pieces and all happened. So that scattered the organisms and organism, plants, animals and all that. That time South America flora fauna joined with North America. So these animals like which were appearing like hippopotamus, bear, rabbit and all that, highly competed by North American fauna. But luckily, these pouched mammals like kangaroo and all, the pouched mammals of Australia survived more successfully because of lack of competition in Australia. Okay, this was asked in PYQs, actually, previous year questions. Whales, dolphins, seals, sea cows are the examples for aquatic mammals. Okay, these, you are all waiting for these charts. First, I'll tell you about the geological time scale, and later I'll come to this. It's memory based. That's why you have to have a number of more number of revisions. Yeah. The previous year only, 2024, one match the following is asked based on it. First, actually in textbook they did not mention, but in the match the following this was there. Proterozoic or Precambrian, like these are the eras. First, actually consider the long time scale is considered era. Era is divided into periods. Periods are divided into epochs. I repeat, the bigger one is era. That is divided into periods. Each period is again subdivided into epoch. The smaller segment is epoch. Okay, like that. Based on the evolutionary history only, this division of the periods is given. And first about one era, the oldest one. Azoic, that time life forms only not there on earth. Archaeozoic means prokaryotic cells only were formed, but these two no need to remember, children. So now we'll go to Proterozoic era or Precambrian. Here slowly these protobionts evolved into some primitive life forms. Eukaryotic cell formation or multicellular condition created in this Proterozoic. This was asked in the match the following in 2024 Neet paper. Proterozoic or Precambrian era, you can see the multicellular eukaryotic forms. Okay, that you remember. Now from Paleozoic era, actually in textbook it is given in plant evolution, that schematic representation you can see there. Paleozoic era onwards they had given. The chart is given. Only thing, you add these two, Cambrian and Ordovician periods, you have to add to that particular chart down. Okay, Paleozoic era, in our textbook, they mentioned from Silurian. Just check once. Silurian, Devonian, Carboniferous, Permian is given na. So now you add these two also. The oldest is in the Paleozoic era, oldest period is Cambrian. After that, Ordovician. After that, Silurian. Later, Devonian, Carboniferous, Permian. Children, okay? Now let me explain about this. How to remember the sequence? See, I advise you to go, like you have to learn the periods, the era and periods names you have to remember, at least whatever is given in the textbook, that chart. So here, I have a blind mark. I don't know how much it will work out with you. Then here COSDCP, like that it is there, no? So in a camp office, senior DCP is present or something like that. You remember camp office, senior DCP, Deputy Commissioner of Police, like that will say, no? Like that you remember. In camp office, senior DCP is present. Like that. So in camp office, like C for Cambrian period, Ordovician, next Silurian. No, later I'll take. So Cambrian, Ordovician, Silurian, Devonian, Carboniferous, Permian. So this is the sequence of periods. Okay, children. Can you recall? Okay. So about Cambrian. Cambrian here, origin of most invertebrate phyla. So all the invertebrate phyla, you know, right, like Porifera onwards. So that evolution happened. And Ordovician, that's the golden period for invertebrate. That much only you remember, not everything, like very briefly, because already we are human beings and this much content we can't remember. Already this is shorter memory based concept. So Ordovician, you remember only one word, that's the golden period for invertebrates, that much. Yeah, Silurian. So here actually jawless fishes, all these were evolved, okay, and origin of vascular plants also happened in Silurian. Okay, forget about it. Coming to Devonian. Devonian is the golden age of fishes. Golden age of fishes. Carboniferous is the golden age of amphibians. Okay, that much one word only you remember, nothing, okay, nothing more than this. Then I repeat once again. Paleozoic era, the sequence of the periods is Silurian, Ordovician, next, Cambrian, Ordovician, Silurian, Devonian, Carboniferous, Permian. Okay, so once again, Cambrian, Ordovician, Silurian, Devonian, Carboniferous, Permian. So Cambrian first. So invertebrate phyla evolution happened. Ordovician, like jawless fish, that these and all also originated. Silurian, and Silurian here, the evolution of diversification of vascular plants, evolution of jawless fishes also. And Devonian, age of fishes, that one word you remember, age of fishes. Carboniferous, age of amphibians, and Permian. So here radiation of reptiles, origin of mammal-like reptiles, most modern orders of insects, extinction of many marine invertebrates. This much Ramayana you can't remember. Based on the chart, I will tell you what has to be highlighted over there. Okay, and reptiles diversification you can consider, that much you can say. Okay, and Mesozoic era, it's considered as golden age of reptiles. Golden age of reptiles. And here three periods: Triassic, Jurassic, Cretaceous. Three periods: Triassic, Jurassic, Cretaceous. Okay, sequence. And Triassic. Gymnosperms dominated the land. Gymnosperms dominated the land. And first dinosaurs and first mammals appeared. Okay, first dinosaurs, mammals appeared in Triassic. 3 points. Gymnosperms dominated. First mammals and dinosaurs appeared. And next Jurassic, yes, domination of dinosaurs. This is the, see, be very careful. If era is asked for the domination of dinosaurs, go with Mesozoic. Period is asked, go with Jurassic. Okay, and gymnosperms continued its dominance, and dinosaurs also dominated in Jurassic period, and first primitive birds originated at that time. Cretaceous, actually dinosaurs got extincted. I said no, 65 million years ago dinosaurs got extincted. That is in Cretaceous period. Okay, so dinosaurs got extincted, first modern birds appeared, angiosperms also appeared. Okay, next Cenozoic. Cenozoic era is that is actually for domination of mammals, and that's the existing era also, that is the golden age of mammal. This was asked in that match the following. Once you refer that 2024 match the following question, and Cenozoic is for golden age of mammals, and here two periods are there, tertiary and quaternary. These are all epochs level, you no need to break your head. Okay, no need to bother about it. And tertiary, in tertiary period in Cenozoic era, mostly diversification of angiosperms and diversification of mammals happened. Okay, and quaternary, and mostly like human evolution happened. Most of the human evolution happened in quaternary only. Okay, children. So this much you remember. And now back to our textbook schematic representation. Yeah, this one. Here only I asked you to add Cambrian, Ordovician, like Camp Office, Senior, DCP. Okay, DCP, Devonian, Carboniferous, Permian. Okay. Chlorophyte ancestors. Chlorophyte ancestors actually, time is up, you are saying. Okay, chlorophyte ancestors, actually these are aquatic green algae. Chlorophyte ancestors are aquatic green algae and non-vascular, not so developed also. And these are the ancestors for the rest all the plant groups. And these chlorophyte ancestors evolved into tracheophyte ancestors, like land living and vascular plants. Like this type of primitive avascular aquatic green algae evolved into vascular plants, tracheophyte. In between, you can see bryophytes, amphibious plants. So origin you can consider from in the Paleozoic era beginning onwards, but diversification of bryophytes happened from mid-Carboniferous. Okay, so bryophytes diversification from mid-Carboniferous you can consider. Okay, next after this tracheophyte ancestor, these are all vascular plants I said, right? So xylem, phloem, all these are developed, and three genera are there. The genus 1 is Zostrophyllum for the tracheophyte ancestors. Zostrophyllum is one genus, all vascular plants are there. Second genus is Rhynia. The next genus is Xylophyton. So all these are tracheophyte ancestors only. Zostrophyllum, Rhynia, Xylophyton. Okay. So tracheophyte ancestors with certain set of variations may be evolved into Rhynia directly, but in between the transitions will be there. No, those transitions they are called as Zostrophyllum. Zostrophyllum evolved into arborescent lycopods and herbaceous lycopods. Arborescent lycopods got extincted by the end of the Paleozoic era, and herbaceous lycopods are still existing. These are the oldest vascular plants still existing. Okay, then Rhynia, totally with the variations accumulated, maybe 100 variations or something, the totally like replaced by Xylophyton type of ancestors. This Xylophyton group is this ancestor group is the the source for pteridophytes, gymnosperms, angiosperms. From Xylophytons only spinopsids are originated, ferns, ginkgos, conifers, needles, like that. And see, you know, ginkgos are the connecting links between pteridophytes and gymnosperms. Okay, and these are actually oldest gymnosperms only, but actually considered as connecting links between pteridophyte and gymnosperms. And Xylophyton evolved as progymnosperms, and later seed ferns. After that, one lineage is towards cycads, another different lineage is towards the angiosperm formation. First formed angiosperms were like dicots only. After that only, with the more and more variations accumulated, inherited, and that converted into monocotyledons. Children, here some question is there. Let me see here. Ma'am, Xylophyton belong to which era? Let me tell you, dear. Let us see textbook based only. See, no need to break our heads for anything else. Xylophyton, like late Silurian, and you can see like it is fallen into like Devonian. So it's actually in between Silurian and Devonian. Okay. When you have to mark only one option, dear, go with Devonian. But origin, if you see Silurian only. For Xylophyton, children, student is asking about where exactly we have to place Xylophyton. Origin, if they ask, go with Silurian only, late Silurian, means last period of the, like last days of the Silurian. And Devonian also it occupied. Children, okay? Next, any doubts based on the chart? Ma'am, hence having secondary growth is primitive character as monocots were the latest, isn't it, ma'am? Yeah. Yes, monocots are the most evolved ones, more advanced one, more what to say, the among the recent group, actually most diversified one is monocot only. Okay. And see, I don't think you can focus on the origin diversification and all. I don't know that much memory will support and all. I advise you to go with the sequence only. Learn the sequence thoroughly. Like say chlorophyte, tracheophyte, then rhinia, xylophyton. Okay, in between chlorophyte, tracheophyte, bryophytes. Then in between tracheophyte and rhinia, then zostrophyllum, arborescent lycopods, herbaceous lycopods. Like the sequence you focus, like xylophyton to angiosperms to connect, progymnosperms, seed ferns. Next, dicots or monocots, like that. Okay, so like that sequence you focus. But if you have the curiosity and you are like 180 on 180, definitely we have to get one word also we should not miss from the textbook. Yes, I'll encourage for that. And see, diversification of progymnosperms happened between Devonian and Carboniferous. Yes, our aim is to get 180 only for everybody. You all have to get 180 and 180 in zoology. That's why we have to learn everything. Diversification of progymnosperms happened between Devonian and Carboniferous, and seed ferns diversification happened between Carboniferous and Permian. Okay, and cycads origin, suppose if you ask me, see, need to consider this beginning, dear. Permian you have to consider. Origin here. Diversification, if you ask me, then I'll say that it's in Jurassic. Wherever it is bulged, no, that is diversification. Wherever this line starts, no, that is the origin. This much clarity I can give you. Okay, and see here dicots also. Origin you can consider here. Maybe diversification happened in end of the Mesozoic, almost in the Cretaceous. No, Cretaceous it happened. Dicots. Then monocots are currently most evolved ones. Hope everybody is clear about it. Shall we move on to animal evolution? Schematic representation. Yeah. Early reptiles are into two groups of ancestors, sauropsids and synapsids. Sauropsids and synapsids. Synapsids, first you see very clearly, synapsids, these ancestors with the variations accumulated appeared first as Pelycosaurus. Okay, Pelycosaurus, so this type of ancestor with the variations accumulated changed into Therapsid. Therapsid is the recent ancestor for mammals. See the sequence? Early reptile ancestors, like take the group as synapsid. Synapsid ancestors evolved as Pelycosaurus, Pelycosaurus to Therapsid, Therapsid to mammals. Over. This is the sequence. Focus on that only, and question may be asked, who is the recent ancestor for the mammal, like that? Go with Therapsid. Again, if they ask like Therapsid, like ancestor, exact direct ancestor for Therapsid, then Pelycosaurus. Like which type of reptile ancestor this Pelycosaurus is? Synapsid. That's all. Okay, next, sauropsids. Sauropsids here, directly from sauropsid ancestors with the variations, certain set of variations created the evolution to form turtles, lizards, snakes, tartarus, like this. And with the variations, it appeared, some of the sauropsids appeared like first thecodons. These thecodons are the ancestors for crocodiles, dinosaurs, birds. Crocodiles, dinosaurs, birds. Okay, children? That's about the Thecodont ancestral origin. Okay, so Thecodont is nothing but sauropsid only. From Thecodont, what are all originated? Crocodiles, dinosaurs, birds. Is that fine? Next, human evolution. Wow, important only. Human evolution also we can't ignore. Dryopithecus and Ramapithecus were existing 15 million years ago. Dryopithecus and Ramapithecus were existing 15 million years ago. Among these, Dryopithecus was like more ape-like and Ramapithecus more man-like. Okay, and mostly human evolution happened in Africa. Okay, in that also Ethiopia and Tanzania. In those places only human evolution happened. Okay, and hominoid terms, let me tell you, hominid means more like a resemblance with the modern man, human appearance. Hominoid means distantly appeared, like ape-like appearance, gorilla-like appearance, like not much connected to modern species appearance, like that. Okay, then 3 to 4 million years ago. See, first Dryopithecus, next Ramapithecus, after that Australopithecus. 3 to 4 million years ago, man-like primates walked in Eastern Africa. They were probably not taller than 4 feet, but walked upright. 2 million years ago, Australopithecines. See, among Australopithecus, 3 to 4 million is also about Australopithecus only. This 2 million years ago, Australopithecines also about that only. But among like Australopithecus, almost 5 fossils they discovered. The recent one is Australopithecines. Tuang baby, like that we'll say. Okay, Australopithecines 2 million years ago probably lived in East African grasslands. Okay, savannas we say. Evidence shows they hunted with stone weapons but essentially ate fruits. Okay, some of the bones among the bones discovered were different. This creature was called the first human-like being, the hominid, and was called Homo habilis. Same time when they discovered Australopithecines in the same place 2 million years ago only. Some more bones also, different bones they discovered, and that is like first human-like being, the hominid, Homo habilis. So for him also you consider 2 million years ago only. Brain size 650 to 800 cc. And they probably did not eat meat. Fossils discovered in Java in 1891 revealed the next stage like Homo erectus, Dryopithecus, Ramapithecus, Australopithecus. After that, Homo habilis, and now Homo erectus about 1.5 million years ago. And the brain size was around 900 cc. And this person only first ate meat. Who ate meat first? Homo erectus. After that, Neanderthal man, Homo erectus only migrated and became like Homo sapiens. So among Homo sapiens, the earlier one is Homo sapiens neanderthalensis. Homo sapiens neanderthalensis, Neanderthal man. With a brain size of 1400 cc, lived in Near East and Central Asia between 1 lakh to 40,000 years back. He is the one, most of the times in exams also they ask this point only. Neanderthal man brain size, you remember, important, 1400 cc, and they used the hides to protect their body and buried their dead ones. Homo sapiens arose in Africa, moved to the different continents and evolved in different races. Okay, and Cro-Magnon men also remember here. That's also our actually direct ancestor, modern man direct ancestor is Cro-Magnon man only. And here they mentioned about the cave, the prehistoric human, like that. This is about Cro-Magnon men only. Okay, most of the details are not given, but consider. And Cro-Magnon man is the with high IQ, 1650 brain size actually. Okay, and highly talented, and he started this gifting also, and ornaments used to make with the elephant tusk bones and all that. Okay, and during Ice Age between 75,000 to 10,000 years ago, Homo sapiens arose, and prehistoric cave paint by Cro-Magnon man is popular. Even now also we can see in Bhimbetka rock shelter. It's a cave actually in Raisin district of Madhya Pradesh. Agriculture came around 10,000 years back. Prehistoric cave paint 18,000 years ago. Okay, and this is all about evolution and the calculation. I said, na, at least some models I'll show. For evolution only this much time I have taken. When will I complete all the other chapters? A particular locus frequency of A allele is 0.8 and that of small a is 0.2. What would be the heterozygous frequency in a randomly mating population at an equilibrium? What's the answer? What do you have to find out? Heterozygous frequency, 2pq you have to find out, children. So dominant allelic frequency and recessive allelic frequency is given, 2pq you have to find out. See, in a randomly mating population means Hardy-Weinberg only. Hardy-Weinberg, according to Hardy-Weinberg equilibrium, the heterozygous condition is 2pq always, not only pq. What's the answer here? 0. Mention in message, nobody is active. Where is the pen? So 2 into 0.8 into 0.2. What's the answer? 0.32. Okay, this is one model and most preferably we expect this model only will be asked. Okay, next. Actually in 2019 one question was asked. That also I'll show you. First, let us solve this. In a population of 5,000 individuals, 1,750 belong to homozygous dominant, 2,500 heterozygous, and 750 homozygous recessive. Based on this data, the frequency of capital A means we have dominant allelic frequency. They are asking you to find out. What's the formula? P is equal to, P means here the dominant allelic frequency. So A only you can mention as they mention here. So this dominant allelic frequency is equal to 2 into homozygous dominant, capital A plus heterozygous condition by 2 into total. 2 into total. So here you can take 2 into this 1750 plus capital A small a 2500 by 2 into total, 2 into 5000. Okay, now whatever value you get, that's the answer. Okay, children, thank you. 0.32 now they answered. This also you can say, what's the answer for it? Okay, shall I show another model? We'll wind up after showing the previous question, 1 or 2. Genetics, many children asked me yesterday when I went for rounds, actually many children asked me to teach genetics also. Yeah, this was this was the model given in 2019 Neet paper. Hope you would have solved it, and many times we also gave in weekend papers. Okay, now 2-3 times we repeated the same question because so that you will be familiar with the. So I'm just showing you the model. They can ask you this type of questions. Okay, how will you find out dominant allelic frequency is given, and how to find out the individual frequency? Capital A 0.4 into 0.4, so 0.16, and homozygous recessive small a. So whenever the dominant allelic frequency is 0.4, the recessive allelic frequency will be 0.6, because remember always P plus Q is equal to 1. So like that. So 0.6 into 0.6, 0.36. Small a 0.36 it will be. And heterozygous condition 0.2 into 0.4 into 0.6. So 4.8 will be. I think this is the answer. Okay. With the same question, yeah. Well, last question for evolution chapter. After this, we'll close evolution. Yeah, solve this. In a Hardy-Weinberg population, frequency of autosomal dominant disease that is 60%. Then what would be the frequency of the affected person in the population? 60% means actually they are giving like capital P. Dominant allelic frequency, capital P is equal to 0.6. That's the meaning. Okay. So Q is equal to how much? 0.4. They are asking you about 2PQ condition. Okay. So that much only na. What would be the frequency of the affected persons? Affected persons means affected persons means the autosomal dominant. This is affected only, and this one also affected only. We need to find out that. Okay, so I represented capital P means A, and this is like this. Okay, so now capital A is equal to capital A is equal to P square into total. Okay, so 0.36 into total 100 only. When they did not give anything, 100 you can take. So it's like 36, and this one capital A small a is 2PQ into total. Children, are you getting 2PQ how much? 0.48 into total 100. So it's like 48. So total how much? 48 plus 36? 84. Yeah, 84. Okay. So that's what they asked. The autosomal dominant affected means homozygous dominant you have to take, and heterozygous also you have to take both. Okay. That's the answer. Yeah. Shall we close evolution and move on to genetics? Okay. What's the time? Yeah. Take a break for 5 minutes and come back faster. Yeah. I'll be back just 2-3 minutes. Someone is asking for DNA fingerprinting, active, passive. Definitely, dear, I want to complete everything actually. I don't know, I am not greedy. Make things will be positive and I'll complete everything. I want to help you maximum because this is the last session in Marathon Gold also. And I don't have, again, the opportunity to teach also. Okay, fine. So definitely, I'll complete genetics first, and many children asked for it actually yesterday. And DNA fingerprinting will be included in this only. I'll cover, and later on, yeah, I'll take up health and diseases or human reproduction. Based weightage wise, I have to give priority. Yeah. Sex determination. Generally, sex determination in humans happens whenever zygote is formed. Okay, during fertilization, when zygote is formed only, sex is determined. So, based on the development of gonads, which type of gonads are developed in the individual, okay, that will determine the sex actually. Okay, type of gonads only determine the sex in the individual. So, first, remember here in this paragraph, Henking, this is PYQ also, Henking discovered X body in 1891. I repeat once again, Henking in 1891 discovered X body. In where actually in squash bugs, when he was doing experiments on squash bugs, like few insects they mentioned, no, that name of the insect is squash bug. Okay, when he was doing experiments on spermatogenesis, he noticed like half of the sperms are with X-shaped structure and half of the sperms are devoid of that. Only autosomes are there. Okay, so A plus X, A plus 0. This combination among the sperms he noticed. Okay, so next one, McClung, one scientist actually, he termed that X body as X chromosome. Okay, McClung noticed that X body as X chromosome. McClung is popular for research in grasshopper spermatogenesis. Okay, Stevens and Wilsons actually they discovered Y chromosome. It's not required. Henking only, you remember, in 1891 he discovered X body. Yeah, coming to the types of sex determination. Actually, if I speak detail about the sex determination, in lower organisms or in some other organisms, different methods are there. Just based on the egg size only in certain organisms, sex is determined. Okay, larger egg size means female and smaller egg size means male, like that. And in certain reptiles and all that, incubation temperature, like while mother animal is sitting on the egg and hatching it, at that time based on the incubation temperature also sex is determined. But now we are not discussing about all that type of sex determination methods. We are discussing about the chromosomal sex determination methods, okay, which generally noticed in the developed organisms. Okay, so coming to the sex determination types, XX and XX and XY method, XX and XY method, XX and X0 method, and ZW, ZZ. This is what is given in syllabus. And one more also let us consider, Z0, ZZ method. Okay, so here XX, XY. Generally, this type of sex determination method we see in humans and Drosophila. Humans and Drosophila. XX, X0, and this is like grasshoppers. Most of the insects fall into this category. Squash bugs, I said, right? Squash bugs. Next, you can say ascaris, cockroach. All these are the examples for XX, X0 method. And ZW, ZZ, you can consider birds, reptiles, fishes. Fishes also you can consider. Z0, ZZ, butterflies. Fumia. Fumia is a small moth. Okay. See, whenever you say this XX, XY, XX is like female condition, XY, along with autosomes, this will be there. Like if I have to represent this condition, females like AA plus XX condition, like a full set chromosomes, and male is like AA, autosomes plus XY. So whenever gametes are produced in males, two different types of gametes are produced.
Okay, half of the sperms are with A plus X condition, half of the sperms with A plus Y condition. But if you take female, how many ever ova in her life she produces, all the ova are with the same chromosomal condition, that is A plus X. So like here in this condition, I am just introducing the term. If one individual is producing same type of gametes, we can consider that is homogametic condition. When one individual is producing different types of gametes, then we can consider that as heterogametic condition. So this one can be taken as an example for male heterogamety. Male heterogamety, XXXY condition is the example. Then XXX0 condition, here also same thing. And here the sperms condition, if you see A plus X is half of the sperms and A plus 0, half of the sperms will be there. Two conditions. So this is also example of male heterogamety, because here the females will be having all in the ova A plus X condition only. Children, hope you are following. A plus X condition in all ova you can see here. But in the male insects like cockroach male, grasshopper male, grasshopper, like that, if you see, half of the sperms will be with A plus X and half of the sperms only with A, autosomes, no sex chromosome. Okay, this is also example of male heterogamety, these two. And here if you see, this is for female heterogamety. Here female individual is producing two different types of ova, but males are homogametic here. Okay, so here the ova condition, if you see, A plus Z, one ovum, and another type of ovum is like A plus W, like that. And here half of the ova can be with A plus Z and half of the ova will be with only autosomes, no sex chromosome. Okay, now one more point I'll highlight here. So now you understood, na? These two are the examples of male heterogamety. These two are the examples of female heterogamety. Yes, and now coming to here. Here, whenever organisms have this type of chromosomal condition, chromosomal number in male and female is same. Number is same. Okay, so but only what is determining the sex in this condition in humans and Drosophila? What is determining the sex? Y chromosome is determining the sex. Y chromosome is determining the sex here. Okay, that is also important point only. And coming to grasshoppers, cockroaches and all, females have, remember this statement, very, very important. And females have one chromosome extra, one sex chromosome extra than males. Okay, suppose if you take a female cockroach has 24 chromosomes, how many chromosomes are there in male cockroach? 23. Okay, here 24, here 23. So this condition is actually example of male heterogamety only, but mostly noticed in the insects. Okay. And next one, ZW, ZZ. So here, W chromosome is determining the sex. Here also, chromosomal number in the male, female is same only. But type of chromosome, which type? W chromosome is determining the female sex. So ovum is determining the sex here. Okay, and that best example, birds, reptiles. Okay, then here absence of sex chromosome in the ova, half of the ova determines the sex. Okay, the next we'll move on to the next haplodiploidy condition in honeybees. One more method is haplodiploidy condition. Haplodiploidy condition, best example, honeybees. Honeybees. Like generally we say the group organisms belong to the group Hymenoptera, ants, wasp, honeybees, all these follow this method only. And according to this, the set of chromosomes, not the type of chromosome or number of chromosomes. See, if you take this type of condition, here type of chromosome is determining the sex. If you take this type, this one and all, number of chromosomes is determining the sex. But in case of this, set of chromosomes is determining the sex. Okay, 2n condition actually, 2n condition, diploid condition determines actually female sex in honeybees, and haploid set of chromosomes, haploid set of chromosomes determine male sex. Okay, so 2n condition 32 and haploid set 16. Okay, so generally this is about fertilized eggs, and haploid set you can see in unfertilized eggs. Directly converted to drones or males. Remember about this. See, haploid condition, like 16 chromosomes are present in drones or males, and they are directly developed from mother's ova or unfertilized eggs itself. This condition is called archenotoky, parthenogenesis. Parthenogenesis. In parthenogenesis, in this type of asexual reproduction, males are produced. So parthenogenesis, which is able to produce males, this is called as archenotoky. It's called archenotoky. Okay, clear? And coming to this 2n condition, generally females, like these are developed from fertilized eggs. Fertilized eggs converted into females. That's why diploid condition is a sexual reproduction, result of sexual reproduction. So sexual reproduction occurs here. And after that, among these females, two categories are there, queen and worker. Okay, worker bee and queen. Only one queen bee, many worker bees are there. So remember the statements here. Consider worker bees as sterile diploid females, okay? And queen bee as the only fertile female. Queen bee as only fertile female with diploid set of chromosomes. Coming to drones are males, fertile or sterile, that will confuse you in the long run. And you consider haploid is there, maybe sterile like that, but actually these are fertile males. Drones are fertile males. Okay, though they have haploid set, they are fertile males. They can produce sperms. Okay, this is the important information. And one more point also I have to highlight, and that I can show in textbook also. Actually, during spermatogenesis, males produce gametes only by mitosis. Okay, suppose if I take drone with 16 chromosomes, okay, here only mitosis is required to produce sperms. Okay, then suppose if you take fertile female queen bee with diploid condition, and here meiosis is required to produce gametes. In oogenesis, meiosis is required because haploid set finally has to be converted into diploid condition finally has to be converted into haploid ova. No? So gametes have to have haploid. So certain ova immediately get fertilized by the sperms, and finally they produce zygote. These zygotes actually converted into females. This is the story, whatever till now we came across. Females. And unfertilized eggs will be there, no, which are not fertilized, and that will get converted into drones, males. For these males, which are with the haploid set of chromosomes, there is no father. Okay, because only through mother's eggs only they are developed. So as such, father is not there. Okay, but this even this drone can't produce sons also. They don't have father, they can't produce sons, but they have mother's father is there, no? So they have grandfather and they have grandsons. Okay, that statement also you remember. And this is about haplodiploidy condition. Now I'll show mutations. Where is that copy? Yeah. Who proposed mutation theory? Hugo de Vries. Hugo de Vries proposed mutation theory. He is the one who introduced the term mutation. Like, what is mutation? And one more point I will tell you. Here, Y chromosome is the smallest, like in XY type, if you see in human beings and all. Y is the smallest chromosome in human body. And on top of Y, SRY gene is it. 3 capital letters, SRY. SRY gene is present on Y chromosome, which codes for TDF factor, TDF. SRY gene codes for TDF, capitals. Testis determining factor. Testis determining factor, that's the reason in males, testis formation occurs. Yeah, coming to here, what are mutations? So this is about alteration of DNA sequence. Okay, sudden changes in the DNA sequences can be termed as mutations. Mutations happen in the in gene level, or may happen in chromosomes also. Based on that, gene mutations and genetic mutations, like chromosomal mutations, like that we can segregate them. Okay, coming to gene mutations. Suppose any ATC became AAC due to mutation. Okay, at that time, first genotype will be changed. After that, this affects the phenotype also. Our characteristic features are changed. Okay, chromosomes based, if you see, like chromosomes structurally change may happen, structural mutation may be there. Otherwise, number of chromosomes also, it can be added or it can be deleted. Like that, addition or deletion of chromosomes also may create a change in the body. Okay, so chromosomal mutations include change in the number or maybe change in the structure. Structural change is considered as chromosomal aberration particularly. Chromosomal aberration means structural change in the chromosome may lead to, and mainly these aberrations are only lead to cancer. Okay, so that's the main reason for cancer here it's given, and will be more clear. I'll be clear about aneuploidy, polyploidy, euploidy, all that. And simple version here, they had explained about point mutation, gene mutations. These are point mutation and frame shift mutation is given in this paragraph. And point mutation means change in a single base pair of DNA. Change in single base pair of DNA. Best example for this is sickle cell anemia. Sickle cell anemia. Frame shift definition is deletion or insertion of some base pairs of DNA can change. See, somewhere some base pairs are added or some are deleted. From that particular point, the entire frame of the DNA will be changed. Okay, that's why the name is given as frame shift mutation. Okay, so that is thalassemia, like that we'll consider for that. Okay, and these mutations are caused because of mutagens, certain agents, physical, chemical, biological mutagens are there which can create mutations. Okay, physical, you can consider radiations, you can take like ionizing radiation or non-ionizing radiation, like that you can consider. Chemicals like the certain carcinogenic substances like that will be there, they can create the mutation. And biological, some viruses and all may cause it. Fine, shall I move on to the mutations? Here I have to highlight. Yeah, gene disorders, first I'll tell you. Mendelian inheritance pattern is followed for gene disorders, like inheritance pattern is followed. Okay, generally in society, we have single gene mutations only, that's very common. And see, based on their inheritance, like through autosomal gene linked it is getting inherited, or sex chromosomal linked it is inherited. Based on that only we consider the gene diseases as autosomal or sex chromosome or X linked, Y linked genes, like that. And when we consider something is recessive and something is dominant, means the dominant ones are like in heterozygous condition, mainly in the heterozygous condition, if a characteristic feature or some trait is expressed, then that time it is considered as dominant. Okay, homozygous, like homozygous recessive also expressed, homozygous dominant also expressed. So mainly which filters some characteristic feature is recessive or dominant is based on the heterozygous condition. In heterozygous condition, suppose it is like this, capital A, small a. At that time, which character you are seeing? This characteristic feature or this allelic characteristic feature? This will decide which is recessive or dominant trait. Based on that only, now we have in the society the list of recessive dominant diseases based on the expression of the trait in the heterozygous condition. Okay. Next, mainly when it comes to autosomal linked, the inheritance happens through autosomes. Like among our 23 pairs of chromosomes, except one pair, XX or XY, the rest, all the 22 pairs are autosomes only. Okay, so autosomal related Mendelian inheritance diseases, if you see, autosomal recessive are there, autosomal dominant, okay, like that. See, autosomal recessive, let me mention both side by side. Autosomal recessive, autosomal dominant. Suppose any gene will be having two alleles, na, will represent like dominant like with capital letter and recessive as with the small letter. So capital A, capital A, capital A, small a, small a, small a, like this.
And for this also in society, for expression of the alleles will be in this way only. I will show you the difference. Like generally, autosomal recessive, when I mention here, my view is like recessive, mutation is recessive here. Mutation is recessive. Mutation is represented with small letter. Children, many students have the difficulty in writing this genotype only. So consider like you are terming some disease as autosomal recessive or some characteristic feature as autosomal recessive. There you are representing that mutation, the character with small letter. Okay, this one. Okay, now this is mutation. So here autosomal recessive is expressed when two mutations are there. Homozygous recessive condition is expressed condition. Homozygous recessive. Homozygous recessive. This is the expressed condition, so they are affected or something like that. And this is carrier. In recessive condition, like one dominant and recessive allele is there, then we consider them as carriers. And this is totally normal. They don't have any mutation. They are normal totally. Okay, this is affected condition. Affected or trait is present, something like that you can consider. Let me apply it for autosomal dominant. Dominant mutation is dominant and we are representing with capital letter. So two mutations are there. Definitely the person is affected or some trait is present, like that you can understand. Heterozygous, like this is mutation, this is normal condition. So at that time in dominant condition, this person is affected even in the heterozygous. There are no carriers in the dominant condition. Remember that there are no carriers for the dominant inheritances. Okay, now there is no mutation, capital letter only not there. Okay, both are small letters only, A. So this is totally normal condition in this particular person. For solving pedigree charts, if you can write this one, then it will be easy, children. Okay, nothing is there in pedigree charts. We have phobia that, oh, those charts are very tough and all that. If you are able to write these genic expressions, you know, solving the chart is very, very easy. Okay, many have like immediately it will not click in your mind that, oh, autosomal dominant affected condition, what shall I take, like that. So immediately you can go dominant means capital letter will express the mutation. So both are like both alleles are mutant, then definitely the person is affected. One dominant mutation is there, also person is affected. No carriers for this. Always carrier term you will hear for recessive condition only. Okay. And next X-linked recessive. X-linked recessive. Hemophilia, color blindness. For this we have to recall this combination. See, X-linked, any X-linked dominant or X-linked recessive, criss-cross inheritance is followed here. Criss-cross inheritance is followed. Okay, let me be more clear. Criss-cross inheritance means criss-cross inheritance. The mutation from father it goes to daughter, from mother it goes to sons. This is called as criss-cross inheritance. Okay, so this is there even in X-linked dominant also criss-cross inheritance is there, but that we can't be specific in autosomal condition. Okay, and here how to represent, suppose examples I told you, examples are color blindness. In our syllabus, X-linked recessive, color blindness, hemophilia. And one more also, if you want to write, you can write DMD. Duchenne's muscular dystrophy, DMD. Okay, so all these are the examples for X-linked recessive. For this, the expression for female, it has to be for female, no mutation. XX is normal, obviously. Okay, and color blindness, suppose if I take recessive mutation, a small c I will take on top of 1x. Okay, small c, small letter c. Then this is carrier, and homozygous recessive is expressed condition here, and she is color blind. XCXC is color blind, and she is carrier, and this person is normal. Yeah. Okay, for male, XY is normal, XY is normal, XCY is colorblind. XCY is colorblind. Yes, children, if hemophilia is there, instead of small c, I'll keep small h. Okay, like that, you have to change. And coming to X-linked dominant, shall I move? X-linked dominant.
X-linked dominant here. X is normal for female. See, no mutation. We did not represent here any mutation. Suppose incontinentia pigmenti, one disease is there where the mosaic patches are appeared. Then XI, heterozygous and homozygous dominant is considered as affected. This is the problem. So compared to X-linked recessive, whenever females have two mutations on the X chromosome, then only they suffer. That's why X-linked recessive is very common in males. Okay, not for females. But X-linked dominant, mostly females are affected. Okay, however, males are affected, but compared to males also, mostly females are affected in X-linked dominant. Okay, because in two probabilities, females are getting affected, heterozygous and homozygous condition. So this is also affected condition only. For males, same two probabilities only. Na? XY is normal. This is normal condition for male. And XIY, this is affected. Children, is that fine? So we'll recall about all the points related to autosomal recessive inheritances, and we'll move on with the condition. Shall I go to the textbook? Quickly, I'll complete. We need to solve one worksheet also. Yeah, I'll come back to pedigree charts. Yeah. First, X-linked, I already told you about the genic expressions, and color blindness. Color blindness here, whenever mutation is there on X chromosome, yeah, cone cells pigments are not formed correctly. So the color identification, green, red color differentiation is the problem for these people. Okay, and in society, if you see, I told already, X-linked recessive disorders follow crisscross inheritance, and compared to females, males are very commonly affected. Okay, and here in society, if you see case study based, 8% of males are affected means only about 0.4% of females are affected. This statement also you remember. Okay, and next we will solve and find out, like here people if they can't differentiate between red and green color, that color blindness is called as protanopia. If they are not able to differentiate all the green color shades, then that is called a deuteranopia. So protanopia, deuteranopia, green color blindness or red color blindness are the examples for X-linked recessive condition only. Okay. They may ask, in a worksheet actually I gave some questions like this, but even then just like one model I'll show. Suppose if you take carrier female, carrier, she's carrier for colorblind, and normal male. What's the probability of their male children to be affected? Normal male. Carrier, what is the genotypic expression? XXC. She married a normal male. Now find out how many of their male children affected. This is parental generation. What are the gametes for her? And sperms with this. Now, this type of sperms fertilized by the sperm XX. You can go with Punnett square also if you are comfortable. XCY. So how many of their male children only I asked, not total children. How many of their male children are affected means 2 boys are there. See here, boy 1 and boy 2. So among 2 boys, 1 boy is affected. So you will tell the answer as 50% of males. 50% of males are affected. That's the probability. Okay. Any of the females are affected? No, 0%. Females are not affected here. Okay. And suppose the question, last line is important, children. In this type of questions, last line you have to focus. Like how many of their children, they generalize. How many of their children are affected? At that time, 4 children you will take and find out the probability. Okay. Among 4, only 1 is affected. So that time, 25% is the answer. Okay, how many male children if they specify, then only 50%. Yeah, someone answered. Yeah, very good, Shruti. You answered correctly. Yeah. Yeah, next one is about hemophilia. Hemophilia, it's a bleeder's disease, otherwise called as Royal's disease. Queen Victoria is the carrier for hemophilia. So they will ask in the exam, what's the genotypic expression of Queen Victoria? Can you write? XXH. Okay, that's the genotypic expression of Queen Victoria, and somewhere in old papers it is there. And Queen Victoria did not get inherited the mutation from the father. For her, it is a spontaneous mutation. Suddenly in her life only she got it, spontaneous mutation. Now because of her, what to say, next daughters became like carriers. Nah? They married royal family prince. Okay, like that many royal families now are with hemophilia only. Okay, because of the mutation present on the X chromosome related to hemophilia, there's some clotting factors which are mainly involved in blood clotting are not produced. So because of the lack of clotting factors, bleeding occurs and blood clotting is delayed, I can say. And in severe cases, blood doesn't clot, and any small injury also make them to lose lot of blood and they will die. Okay, that type of condition. And Queen Victoria suffered from here. Actually, two hemophilias can be considered. Hemophilia A is very, very common type. 80% of hemophilia cases are hemophilia A type only. Okay. Hemophilia B is actually 20% cases I can consider, and she suffered from hemophilia B actually. Okay, and what are the differences between this? Both are hemophilia bleeding disorder, blood clotting disorders only. And here hemophilia A is caused because of the absence of anti-hemophilic factor, like 8th factor. It is clotting factor 8. Okay, so hemophilia A, lack of clotting factor 8 is the reason behind it. Okay, when mutation is there on X chromosome, clotting factor 9 is not produced. That leads to PTC, actually plasma thromboplastin component, Christmas factor otherwise called as. So that leads to hemophilia B. I repeat once again, hemophilia B is actually because of the lack of Christmas factor, means factor 9, clotting factor 9. Okay, so that's about hemophilia. Same, the probabilities we will solve later in the worksheet. I'll move on to the autosomal recessive disorders. And one more point I have to tell you here. In case of females, general version I'm saying here, not with respect to any disease or something. See, XX in females, if you say, as we have two same chromosomes, like homozygous, heterozygous condition is noticed. But in males, we can't hear this type of terms. Homozygous, heterozygous, in males it is not applicable. Okay, it is like a hemizygous condition. In males it is like hemizygous because both the chromosomes are different. X, Y are different. So we have to term like hemizygous condition over there. Okay, so for males, homozygous, heterozygous term is not applicable. One more. The genes present on Y chromosome are called as Hollandric genes. Okay, genes present on Y chromosome are called as Hollandric genes. Okay, next. Autosomal recessive condition examples. Autosomal recessive condition examples are sickle cell anemia, very, very important, and mainly in main exams like Neet exams, they are asking questions from this. And one more, thalassemia, phenylketonuria, cystic fibrosis, okay, albinism, okay, alkaptonuria, all these can be considered. Okay, so first about sickle cell anemia. I think if I take the concept to board only, it will be good, but time will not be sufficient, I think. Yeah, sickle cell anemia. So it is because of the mutation on chromosome 11. Where is the mutation? On chromosome 11. So due to mutation, generally in these people, sickle-shaped RBCs are formed under oxygen deficiency. Okay, whenever low oxygen tension is present, at that time their RBCs get converted to sickle shaped. Then these sickle shaped RBCs can't transport much amount of oxygen, and then they block the blood vessels. Then the nutrients also will not be circulated properly when the blood vessels are blocked, and like that, lot of visceral organs are also damaged. That's why sickle cell anemia is also considered an example of pleiotropy. Like defect is there with respect to one particular gene on chromosome 11, but multiple effects are there. That's why it is also an example of, like, generally phenylketonuria you consider, na? So consider even sickle cell anemia also
for this pleiotropy. Whenever this phenylpyruvic acid is accumulated in the cerebrospinal fluid in the brain, and people will become mentally retarded. Okay, they become mentally retarded, and kidneys absorb all these like very poorly. So that's why the urine, all these acetic acid, pyruvic acid, excess of phenylalanine, ketone bodies, everything can be excreted through urine also. That is only phenylketonuria. That's why term is also given like that. And because amino acid effect, right? It is related to amino acid effect because of the lack of enzyme. That's why skin pigmentation, hair pigmentation is also affected. That will be reduced. And this is the best example for pleiotropy. Okay. And eczema also, skin rashes and all also appear here. Okay, this is about phenylketonuria. Coming to thalassemia. Thalassemia, here two types of thalassemias are there, alpha thalassemia and beta thalassemia. Okay, alpha thalassemia, if you see alpha thalassemia, here problem is with chromosome 16 related to two closely linked genes, HBA1 and HBA2. Children, remember, two genes are there means, see, one gene, two alleles. So here four alleles are involved, two genes no, so four alleles are involved here. Related to each parent four alleles are involved. Okay. So generally, if mutation is not there, your HbA1, HbA2 are normal on chromosome 16. Like your in your body, hemoglobin alpha chains are produced in normal number. So because of mutation, alpha chain synthesis is affected. Okay, because of this mutation, alpha chain synthesis is affected. Okay, see, I'll just show, I'll give one example here. I'm speaking about alpha thalassemia. So where the problem is with chromosome 16 and HbA1, HbA2. At that time I am saying, like actually this is hemoglobin structure, like 4 polypeptide chains totally. Among them any two, like you can, like let me take this one as alpha 1, alpha 2, this one as beta 1, beta 2. Due to mutation on chromosome 16 with respect to these genes, alpha chain synthesis is affected, I said. Okay, let me say, sorry. Let me say this one is not produced. Okay, that is one problem. One of the among two, in one hemoglobin, one alpha chain is not synthesized. Quantity is reduced. Then now, additionally, in this place, it is not empty. Again, excess of beta can be produced. Beta 3 I can mention. Okay, loss of alpha is one problem, addition of excess of beta is another problem here. So sometimes question will be asked, in case of alpha thalassemia patients, alpha chain is less, but beta chains are more. Okay, and this is nowhere like considered as normal hemoglobin. Now this is abnormal. This type of abnormal tetramer will not exist. So unstable hemoglobins will be easily destroyed. That's why persons will suffer from anemia here. So alpha thalassemia patients have more number of beta chains, less number of alpha chains. Okay. And beta thalassemia compared to alpha, beta thalassemia is very dangerous. Why? Because the mutation is with respect to one gene only. HBB gene present on chromosome 11. HBB gene means only two alleles are involved. So if one allele is mutated also, symptoms will appear. Here 4 alleles are involved. So when 2 alleles maximum, like 2-3 alleles are mutated, then only he shows like more symptoms. Otherwise, one allele mutation doesn't affect the person much. Mild symptoms will be there. Thalassemia minor only will be there for them. Okay, hope you are getting. So beta thalassemia is caused because of the mutation present on HBB gene present on chromosome 11. Okay, due to this mutation, beta chain synthesis is reduced, but alpha chain synthesis is increased. That's why this is a quantitative problem. Whereas sickle cell anemia is considered as qualitative problem. Okay, that's what you have to recall. Palm is like broad and palm crease is the unique variety of lines are present. Physical, psychomotor, mental development is retarded. Okay, physical, psychomotor, mental development is retarded. You can see here, these are also like all heart abnormalities. Okay, all the some visceral organs also, gonads also not developed properly. This can be sterile. And main reason for it, main reason I'm saying, spermatogenesis also may be the reason. Like main problem is late pregnancies, 40 plus, like that women undergo pregnancy. Like oogenesis is the issue. During oogenesis, ovum formation time, anaphase 1 or anaphase 2, any time you can take, the separation of chromosomes, the pulling will not be proper, so abnormally it will be pulled, and 21st chromosome, trisomy, 21st chromosome will be there in one gamete. If that is fused, and this Down syndrome is caused. So oogenesis is the main problem, even spermatogenesis also. As it is like related to problem is with the trisomy 21, sex chromosomes are nowhere affected. So Downs can be males or females. Downs can be set of chromosomes. So 47 chromosomes are there. 47 chromosomes are there. So 47, XXY is the karyotype for Klinefelter syndrome. Okay, normally to write 44 autosomes are normal, XXY condition. These are males. Klinefelters are only males with the addition of extra X chromosome. Non-disjunction is the main reason, and mainly oogenesis issue here also, mainly oogenesis issue. Males with addition of X chromosome, so they have some female characteristic features also. These males are very tall statured, then breast development, gynecomastia is there, and these males are sterile, gonads are not developed, they are sterile, and feminine characteristic features, voice and all that also, the feminine pattern of hair growth, all that can be noticed in these people. Okay, and next coming to Turner syndrome. Turner's example of monosomy, allosomal related. Absence of one of X chromosomes in female leads to Turner syndrome, monosomy, allosomal monosomy. So 2n minus 1. So 2n minus 1, so 45 chromosomes are there, and 45 comma X0 can be written. Otherwise, 44 autosomes plus X0. 44 autosomes is mentioned, plus has to be there. 45 is mentioned, comma has to be there. It should not be written as 45 plus X0, okay? So it has to be written as 45 comma X0, otherwise 44 plus X0, children, okay? And these females, absence of one of the X chromosomes, right? That's why secondary sexual characteristic features are not developed much. So that's why these are sterile, ovaries are rudimentary, okay, and breast development is not proper, uterus also not grown properly, and webbed neck is present, and some people suffer from they become deaf and dumb also, it's in, they can't hear, hearing disability will be there. Okay, this is about Turners are also shorter, Downs also shorter. Here in the given syllabus, Klinefelter is only very tall statured male. Okay, Turners are only females. Klinefelters are only males. Downs autosomal related, so it doesn't affect sex of the individual. So they can be Downs males, babies are baby girls or baby boys. Okay, is that fine? Pedigree chart. See, for Mendelian, whichever actually follows Mendelian inheritance pattern, for those diseases only pedigree charts are useful. Okay, and yeah, I'll first introduce the symbols. Chromosomal disorders are not inherited for analyzing the inheritance pattern. Okay, they are not inherited. So you can't use pedigree charts for chromosomal disorders. Only for the disease like autosomal, now I said, X-linked dominant. So this type of disorders, Mendelian disorders, we can apply pedigree chart analysis. Okay. So here two charts are there. Let us see, like this one also. Till now in your entrance test, only these symbols are asked. They did not ask any calculations. But for practice, no, we in class and in weekend papers we are giving to make you more stronger in the concept. Yeah, the empty symbols, this one for male and this one for female. This symbol for sex unspecified, when we don't know about the gender, then we can write like that. And if it is shaded, then solid symbols represent affected individuals, and male and female with the horizontal line connecting to that, and it is representing the marriages or mating like that. Then two horizontal lines are there, then it is about consanguineous mating. Okay, then relatives, like mating, marriages between close relatives is represented like that. Then this is the first generation, first child, second child, like that. Then this is like parents have one affected male boy, like this one is, if the number, many times they asked in the entrance, if it is written like this, 5 unaffected offsprings. Okay, same thing will be asked if they have to ask this in the main exam. Yeah, coming to these pedigree charts. Yes. Whenever in the exam, this is like phobia. Actually, this is very simple topic, but children know in your level you will be having little phobia. Oh my God, this is like so different and all. Actually, for uneducated people to understand the inheritance pattern easily, they use the symbols. Though you learn so much, you are educated and having a lot of biology background, you guys have phobia to understand this inheritance. Okay, fine. When the charts are given like this, parents are shown unaffected, means they don't have symptoms, it seems. At that time, when parents are shown unaffected, children are shown affected, that time you can consider this as the recessive inheritance. Okay, so this is the blind mark. Parents are shown affected. Okay, sometimes they are shown affected also. Yeah, we can, but based on the calculation, we can find out. Like always, it's not the generalization that the recessive inheritance pattern has to be given like this only. Just based on the given pictures here, blind mark I am saying. Okay. So this is the recessive inheritance. That time you have a problem that, you ma'am, you said recessive only. Whether it is autosomal or X-linked, how to find out like that? Then we have to apply like this only. Suppose I will tell you to follow first X-linked inheritance pattern, you apply on the picture. Because why I am saying like that is crisscross inheritance clearly can be seen in X-linked recessive. Then one more is, I told you, crisscross inheritance is the mutation from male goes to daughter, from female it goes to son. Like more clearly, definition will be in detail in different way, but this is like simple version for you to understand. Yeah. So here, X-linked why to follow means X-linked inheritances are sex specific. See here, male is suffering with the disease. Then here, female. So you can clearly understand in the X-linked inheritances whether male has to be affected or female has to be affected. Okay, that's why finding out the probability based on X-linked inheritance is easy for you. Like more clarity will be there. So now I'll see. Here, parents are shown unaffected. In X-linked recessive, like you can take color blindness or hemophilia. Male is shown like this. Healthy person means XY only I have to give. I can't take any mutation on the X. Okay, because he is shown normal. For female, I can take XX or XXC. Carrier condition I can take. I can't take XCXY because she is not shown affected. Okay, she is shown healthy. So might be single mutation she might be carrying. Okay, XXC we have to take. Okay, at that time, is there any possibility that female will be affected? Just see. Is there any possibility like here father also has XC, then mother also has one of the mutations she carries, then XC combination make her daughter like suffer from it. Like from where like this XC is not there, no. So it's not showing, this picture is not showing X-linked inheritance. Like that we are avoiding, like one confusion we are avoiding, like by solving it, saying that it's not X-linked recessive. 100% it is autosomal recessive only. Understood? It is autosomal recessive. So next, autosomal recessive. Already I gave you the genotypic expressions earlier. So parents are shown healthy, but children are shown affected. So they might be having a single mutation, means carriers. Okay. So at that time for the affected people, both the mutations you give, autosomal recessive. And for this person, you can take homozygous or you can take a heterozygous condition here. Then this one also, she is affected, and this person you can consider either this or this. And this person definitely, for her, you have to take a heterozygous only. Why? The boy is affected. Boy is affected means she has to carry a mutation. Okay. So at that time, affected, and you can take homozygous or heterozygous for this. Okay. This is the probability. When we solve new questions, now you will have more clarity. Okay. Shall I move on to this picture? Yeah. Quickly, I'll move on to this. This is about, see, in this picture when you see, you can see clearly this one. Don't see the first one, and next if you see this one. Parents are unaffected, children are also shown unaffected. When parents are unaffected, children are
unaffected, then it is actually dominant inheritance. It is actually dominant inheritance. See, autosomal dominant, I gave you no mutation at all. So no capital letter. Here you have to take both the alleles as small letters only. Small a is the healthy condition in autosomal dominant. So any probability that children gets the mutation here? Everybody is like small a only. Na? There is no possibility that children will be affected, right? So that's why based on this blind mark, what is the blind mark? A blind mark are okay, we can rely on this. It's not exactly blind. We can rely on this. It is saying that if parents are unaffected, children are also shown unaffected, definitely it will be dominant inheritance. Yeah, we need to confirm whether it is autosomal or X-linked. X-linked only I prefer always to solve it, and he is shown normal, so XY for him, and she is shown affected. So we can take both mutations, X-linked dominant, homozygous or heterozygous. If I take like this, then all her children have to carry the mutation and get affected. But here some are shown normal, so that's why I have to take heterozygous, XXI. One mutation only I have to take, because so some people have to be healthy. Okay, so for healthier female, XX will work out, and for healthier male, XY you take, and for this affected man, XIY you take, and for this female, XXI. Okay, and all these are unaffected, like you can mention easily. And now for her, XXI you can give, and for the man, XY. And one more male has to be affected actually, but here it is given XY only. There is no problem, like maybe next delivery also should have delivered the healthier male. It is also, this probability is also showing actually inheritance of X-linked dominance. Maybe they did not give any example related to that. That's why in our syllabus they had taken the autosomal dominant. Autosomal dominant example is myotonic dystrophy. Myotonic dystrophy. Okay, myotonic dystrophy. Here chromosome 19 has the mutation. Okay, so due to mutation present on chromosome 19, one abnormal protein kinase enzyme is produced, and due to that signaling process for the skeletal muscle will be affected, and degeneration of skeletal muscle can be noticed. Okay, children. So let me give the genotypic expression for it. For healthier condition in autosomal dominant, what I have to take? Small a, in these three probabilities you tell. In these three probabilities, what I have to take for the healthier one in autosomal dominant? Capital letter should not be there. So small a is the healthier condition. For her, I should not take homozygous dominant because some children are healthy. So I can take only heterozygous for her. Okay. Remember it. So next, I think you can mark for this, wherever healthy condition is there, take only small a, wherever healthy condition is there. Wherever affected is homozygous dominant is not possible at all. So it is heterozygous dominant only. Okay, we'll solve some questions related to it. Okay, some 10 questions for increasing your confidence. Time is, no. I'll take later, Amma. Ayyo, so many topics are there. I have energy. I don't know how much patience you have. Hey, come on. Yeah. See this properly? The represented below is the inheritance pattern of a certain type of traits in human. Which one of the following condition could be an example of this pattern? So from mutation from male is going to daughter and also to the son. Mutation from female is going to daughter and also to the son. Which one it is? Options you see first. Options you see? Hope it's visible for everybody. Hemophilia, thalassemia, color blindness, hypertrichosis. Hypertrichosis is like Y-linked actually. So which one you can consider? Crisscross inheritance you came across. What is that? So mutation from male to daughter, mutation from mother to son it will go. But here from both to both it is there. So definitely it will be something related to autosomal inheritance. In case of autosomal inheritance, sex is not specified. Sex is not specified. So you can consider any, what is the autosomal condition here? Thalassemia. Thalassemia you can consider. That's the example. Okay. Because you would have solved in weekend papers, color blindness based picture you would have seen. So little different. So that's why I have shown this. And now solve it and tell me the answer. A man and women each carry allele for phenylketonuria, an inborn error of metabolism, while neither of them has disease themselves. And what is the percentage of their child will have the disease? Phenylketonuria, first identify what type of disease it is. What type of disease? Autosomal recessive. Okay, autosomal recessive. And then, while neither of them have disease, parents did not have any disease. So, heterozygous condition, I already told you. Parents are heterozygous. Then what's the probability of their children to be suffered from the disease? 25%. 25%. Very simple question and remember thoroughly. And next. Ayyo. Next, pedigree. Wait, it is stuck. Oh, yo, people are hungry. 12 o'clock. Oh my God. So many chapters are there. Yo. Human reproduction, reproductive health, so much weightage is there. My God, health and diseases, at least some topics everywhere. No hurry. Yeah. Have patience, okay? And if you are hungry, go immediately eat something within 5 minutes and run and come. Array, totally lost. Situations will be like this. Ayyo. When we are in hurry only, time will not cooperate. Huh? Testing. Mm-hmm. Yeah. Solve it. See, this is the picture. And now whenever, see, they had given mother, father, you have to identify. The genotype of mother and father you have to identify. Quickly draw on your rough notebook. Just picture is given and options are shown like this. Mother and father, autosomal only, they have given like this. So you can identify whenever the options are given like this. Better to apply the options based information only on top of it, because whenever you see picture, multiple, like so many ideas will come. Like unnecessarily if you solve based on your ideas, time will be wasted. That's why see the option and check whether it is correct or not. Okay, now father is shown affected, mother is shown unaffected. You know autosomal recessive, affected one, what we have to mention? Small a. Unaffected one can be homozygous or heterozygous. Okay, let us see. Father, autosomal recessive means father has to be small a. So anywhere small a is there? Yes. Small a is there, then mother has to be capital A means nobody should be affected. But here affected ones are there. So you can't take that. For at that time autosomal dominant you have to consider. Autosomal dominant when you consider, okay, affected one is like unaffected one is shown small a. Okay, small a for the mother, unaffected. Okay, unaffected mother, small a and heterozygous father, affected. So that's right only, na? So fourth option. Next. Why this tick mark is not going? Okay, which of the following inheritance is not possible in the given pedigree? See here, not possible they asked. Autosomal recessive, autosomal dominant, X-linked recessive, X-linked dominant. See, one simple thing you can see here. Crisscross also you can see. Father has mutation, goes to the daughter. Crisscross you can see clearly. So X-linked recessive is possible. Okay, and our autosomal recessive, autosomal dominant, mostly it will be, probability will work out here. There are no need to solve also, sometimes it will work out. Okay, so this like it is possible, and X-linked recessive also possible. X-linked dominant doesn't work on this. Okay, X-linked dominant, like most of the, when father is affected, see I'll show why it is not affect, when it is not applicable means father is having dominant mutation, XIY, okay, father has mutation on X, okay, and she is XX only, let us take. So at that time, his mutation goes to daughters. The daughters definitely has to be affected. She has to get XXI, no, and XXI. Dominant mutation, heterozygous condition also affected. But females are shown normal. That's why it's not showing the inheritance pattern of X-linked dominant. See, how much ever we solve, like we'll be having that phobia. This is easy. This question. Regarding to phenylketonuria, which of the following statement is wrong? Phenylpyruvate deposited in heart, liver, kidney, because it is deposited in CSF, brain. So mental retarded condition is possible. And this one you tell me. And here the part of beta chain of hemoglobin of a person suffering from sickle cell anemia is given. The blank circle represents. So the mutation is there here, right? So valin. So same as 1. Your favorite. So parents are shown unaffected, children are also unaffected, so it is dominant inheritance. So apply that. Dominant inheritance, dominant affected condition is maybe homozygous or heterozygous. Unaffected is small a, small a. So father has to be small a. And mother has to be heterozygous, capital A, small a. Okay, so third option. Next. Known one, textbook one only. Parents are heterozygous, children are affected. Okay, so autosomal recessive, both are heterozygous. This one I think you easily can solve. One at least you solve, and we'll move on to DNA fingerprinting. A female affected with color blindness marries a hemophilic man. What percentage of male children of this couple will be colorblind? A female affected with color blindness marries a hemophilic person. She did not marry a colorblind person, hemophilic person, different disease. All the sadism is there in their family only. Married whom? Hemophilic man. Hemophilic man. Okay. Now, among their children, one will be with this, and one will be here like this. Tell me now, what percentage of male children of this couple will be colorblind? 100%. 100%. Okay. Next. Yeah, so sad. Next, what is the probability of that son of a woman who is a carrier for hemophilia and a normal man will be hemophilic? Actually, this I asked in weekend paper also. Okay, write and solve it later. Then I will move on to DNA fingerprinting. Fine. DNA fingerprinting, your favorite topic. One question from HGP, DNA fingerprinting, one question we can expect. Quickly. All big chapters actually. Biotechnology is also there. Chikungunya. Okay, no problem. Human Genome Project. What is genome? Haploid set of chromosomes present in a gamete is called as genome. Haploid set of chromosomes present in a gamete called as genome. Okay, actually, scientists have got the curiosity to find out the reason behind the differences between two individuals, and that made them to go ahead with a genome project. They wanted to know why two individuals are different. What's the reason? So they understood. Oh, first let us know about the sequence of each gene and the functions. Then only we understand about the differences like that. Okay, and here they sequenced all the genes present in the human gamete actually. Okay, so here it is considered as a mega project. Why? Because it has taken a lot of time. The project was launched in the year 1990 and it ended up in 2003. 13-year project. That's why it is called as mega project. Not only that, it is very expensive. See, human genome actually approximately they estimated that it might be having 3 into 10 power 9 base pairs, 3 into 10 power 9 base pairs. So to sequence each base pair, they estimated that $3 US dollars are required, and to sequence the entire genome actually 9 billion US dollars they have to spend. It's very tell him. Oh, yo, video is stuck. This is the problem, dear. Online 55% males are hemocrit children, 50 children. Answer there. Say how sincere my children are. No, it is stuck, it seems. Children are mentioning. Okay, I think for classrooms it is delivered. No? Classrooms it will be. Classrooms only it is stuck. Okay, video is stuck, he is saying. Huh? Adagama. Okay. Okay, I think in other Ganguru, Porangi, everywhere, it's all okay, fine. Okay, what I was saying that a human genome project is considered as mega project. One thing is like it's very expensive and it is like 13-year project, taking a lot of time, and a lot of books are also required to store the data. So in this project, actually, they had to discover the devices where they can store all the data actually. So that's why they had to work so hard to discover high-speed computers like this type of devices to store the data. Okay, goals of HGP means before taking up the project, they thought that at least we have to identify all the approximately 20,000 to 25,000 genes in human DNA. Okay, and they wanted to identify 3 billion chemical base pairs. Okay, and they wanted to store this data properly and compare the data with other organisms genome data. Then many departments many fields have to be taking the benefit of this information. Like that they estimated so much. Okay, and this human genome project first was initiated by US Department of Energy and the National Institute of Health. Okay, later it was so expensive, right? They requested other countries also to contribute so that they can share the information also. On that deal, Wellcome Trust from UK became the major partner, and small later contributions also came from Japan, France, Germany, China, and other countries. Okay, so India was not part of it at that time, and they took the benefit. Like now, because of genome project, the mainly it helped in medical field to treat gene disorders. Now Mendelian disorders were there, so these mutations to rectify this project was very useful. Okay, not only that, in agriculture, energy production, environmental remediation and all, even non-human models also the genome was sequenced actually, and they compared it and all. Here maybe they may ask in the exam, the Caenorhabditis elegans is there, no? That's the example of free-living non-pathogenic nematode. Okay, and plant side, it's only rice and Arabidopsis. In these plants, they sequence the genome. That you remember. Then methodologies, yeah. From past 3-4 years, every time from this paragraph, one question they are asking. Like either EST, like express sequence tags or sequence annotation, vectors base, or something they are asking from this. Okay, easy, very easy topic. So suppose we are doing the research. At that time, first what we require? What we require first? DNA. So isolation of DNA is the first step. From any living cell, isolation of DNA is the first step. After you isolate the DNA, we don't have the device to sequence everything at a time. So fragmentation is important. Okay. So second step after isolation, second step is fragmentation. After that, one fragment, see now I am the leader for the project or something, you are in the team. Okay, so I distributed the fragments to each and everybody. So if I give one fragment to you, like you continue with the experiment, what if it is damaged on that day itself? So again, entire procedure we have to do from beginning. That's the reason cloning is required. Like same fragment, whatever I gave you, that you can, by using certain technologies, you can amplify it. So cloning is required. For that, some vectors, some host we require. Generally, a host can be bacteria and yeast, and vectors can be bacterial artificial chromosome and yeast artificial chromosome, like that. Okay, so this is the procedure: isolation, fragmentation, cloning by using these vectors and host. And two methods. Now fragments are with you. Now, some people will think that, yeah, let me read everything, like I don't know which is a coding sequence, which is non-coding sequence. If I go randomly, then I may miss some. Okay, like that, they may have a lot of confusion. So blindly, they have sequenced everything. Why to take the risk? Like that, they read from top to bottom, everything. This is a blind approach. That is about sequence annotation. So it's a blind approach, and they sequenced everything. After that, based on their functions, separated that as the coding and non-coding. Children understood what is sequence annotation? And one more is expressed sequence tags. Express sequence tags means these people are very wise, and they saved the money also. They did not waste money also, and within short time only more accurate research they had submitted. Okay, who followed this method? Expressed sequence tags means they identified, first they allowed the DNA fragment to get transcribed and converted into RNA. Later, from that RNA, they got again the reverse transcription, again DNA. So in this process, they identified which sequence of the DNA is getting transcribed. Okay, so whichever gene sequence is expressed as RNA, only that they sequenced. So unnecessary ones they did not even focus. So time was saved, money was also saved. This is the best method actually. Expressed sequence tags means wherever gene is expressed as RNA only is sequenced. Understood? Next, actually, this principle, they got it from the device used by Frederick Sanger. Okay, Frederick Sanger was popular for sequencing amino acids in complex proteins actually, and same method they applied for sequencing genes also in DNA. Okay, and the sequence of chromosome 1 was completed only in May 2006. That was the last one, and most of the genes are present on chromosome 1. Maybe that's why the project would have been little delayed. And totally in this project, they sequenced 24 chromosomes, 22 autosomes, 1 X chromosome, 1 Y chromosome. Okay, next. Next, all the fragments, like now you completed the research. Now we have to set it back to the original condition. Humanly, it is not possible. So for that also devices are required. Bioinformatics field is totally about this only, if you take up that. Okay, so for that, humanly it's not possible. Devices are only required. So based on the repetitive sequences, based on restriction endonuclease enzymes, okay, they try to arrange based on the overlapping regions. They try to arrange the sequences like the original condition. Okay, see that time, no, the very challenging task is like maintaining the genetic and physical maps, means distance between like ATC and CGA, something is there. The distance between ATC and CGA, that is also very important. Distance between ATC, the next to next, that one also, A and T, T and C, that is also important only. So that was very challenging for them. Okay, finally, after the project, what they discovered, this is very, very important here. 2-3 times you read, no problem. Okay, human genome contains 3,164.7 million base pairs. Next, average gene consists of how many bases? 3,000 bases. So here, dystrophin gene is considered as the largest gene. write it down that dystrophin gene is present on X chromosome. X chromosome, that is not there, but it's important. This gene, largest gene, dystrophin, is present on X chromosome. And how many base pairs are present? With 2.4 million base pairs you mentioned. Certain textbooks, see here also, basis is given, but please mention base pairs. In certain textbooks, base pairs is only written, the revised version. So that is the correction here. Everybody write for this like base pairs only. Next, total number of genes is estimated at 30,000. Yeah, earlier they like different teams that they will estimate this much, but little lesser than that. The functions are unknown for more than 50% of the discovered genes, and less than 2% of genome only codes for protein. This much genome is there, less than 2% only codes for proteins, it seems. Okay, most of our genome is formed by repeated sequences only, and sometimes sequences can be repeated 100 to 1000 times also. And chromosome 1 has the most number of genes, 2968. Less number of genes are present on Y chromosome, 231. Scientists have identified about 1.4 million locations, actually SNPs, in human beings. SNPs means with respect to one nucleotide only differences will be there. Suppose CTC became like CAC, T became A, na? So one nucleotide base only changed. So that type of mutations are SNPs. So they identified that type of 1.4 million locations they identified with SNPs. Okay, and this information will be very much useful for so many fields: agriculture, then healthcare field, then evolution. Okay, for all these actually it's very important. Now, shall we start DNA fingerprinting? I can't take break also because time is not much. Yeah. So DNA fingerprinting, actually, this DNA fingerprinting process is useful for forensic department to solve crime cases and parental disputes, to hand over the baby to biological mother, father, like that, and evolutionary history to trace out, to find out our ancestors, okay, the sequence and all that. And whenever massive deaths happen, like a train accident or plane crash or something, dead bodies, to hand over the dead body to the relatives, that time to check the DNA bands pattern and all that. And this DNA fingerprinting technology is very much useful. And this was discovered by Alec Jeffries. Alec Jeffries in 1984, he produced the first DNA fingerprinting. Okay, he produced first DNA fingerprint in 1984. Okay, so coming to the concept, and here you all know that 99.9% of DNA is same in all individuals. The variation is lying only with 0.1%. So I am different from you, you are different from your friend. This is all because of that 0.1% of difference only. And this technology, DNA fingerprinting, not related to these fingerprints, dear. Okay, so it's not related to these fingerprints, it's related to DNA, that 0.1% which is different between you and me. So this they have to highlight. Okay, they want to separate that 0.1% of differences only. Okay, so DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repetitive DNA. And one more, they assumed us to, like they asked us to assume that the 0.1% may be like 3 into 10 power 6 base pair. So they want to compare like my 0.1% of DNA with yours, okay, to know the variations and all that. So focus is only on that. Focus is not on 99.9%. How can we separate? Like you are the scientist. You know that 0.1% of DNA is different. Now you also know, like how, like you want to go ahead with the, to solve a crime case or murder case or something. You want to go ahead with the procedure. Then how to separate 99.9 and 0.1% from one single DNA? Like we'll follow for separation of this 99.9%, 0.1%, we follow cesium chloride density gradient centrifugation method. Okay, cesium chloride density gradient centrifugation method. Like
First, take one live cell, living cell. From that, isolate the DNA. Okay. Then put it in the test tube with the solution. I put it, keep it in. I think you would have seen like one drum-like structure. Like we switch on, like rotations will be there. So if you that is a centrifugation machine actually. If you keep it in that the test tube in that machine, like if you switch on after so many rotations, if you remove the test tube from the machine, two bundles will be there. One is like larger in size, one is like very tiny. Based on the density, the DNA will be separated into two ball-like structures. One is considered as bulk DNA, genomic DNA. The other one is like satellite DNA. See, now another term we introduced, satellite DNA. Nothing but repetitive DNA only with high degree of polymorphism. Variations are more, that's all. Don't like these terms only make you feel like the confidence will go low because of these terms only. Otherwise, technology is easy only. Like, what is this repetitive DNA? What is this satellite DNA? What is mini satellite, microsatellite? Totally your confidence will go down because of it. Nothing is a simple concept. Okay, so here repetitive DNA, that 0.1% I said na. So in that the repeatedly sequences are there. So that DNA is called a repetitive DNA. Now by using the centrifugation density gradient method, we are separating the bulk DNA and satellite DNA separately. That satellite DNA is nothing but repetitive DNA with high degree of polymorphism. Okay, and it is non-coding. Remember this, repetitive DNA or satellite DNA is non-coding, doesn't code for any proteins. Okay, if you plot a graph also, major peak is like bulk DNA gives a major peak, and what to say, the satellite DNA will give a minor peak. If you plot a graph like this it will appear. The amount wise and all that, this is the bulk DNA. Okay, bulk DNA, and this will be like satellite DNA. Satellite DNA, remember, satellite DNA is repetitive DNA with high degree of polymorphism. Okay, and it is having non-coding sequences, does not code for any proteins. Okay, mainly this satellite DNA in the chromosome centromere part heterochromatin type actually. In centromere part, most like large amount will be there in satellite. Okay, in centromere the satellite DNA is more. And based on the repetitive sequence, like whether which type of AT is more or GC is more. So based on that and based on the number of repetitive sequences, satellite DNA is divided into microsatellite DNA and mini satellite DNA. See, microsatellite here we don't use only. For this DNA fingerprinting technology, mini satellite DNA is very important. Like any question, if they give, first identify the chapter, okay, or concept. Sometimes if you don't have any clue related to the topic, you pick up the option. Okay, see, microsatellite, mini satellite, something and all, it is given SSR, STR, something like that. At that time, okay, known term is like mini satellite. Go with mini satellite. Okay, like that. Known terms you select. So here among the satellite DNA, like mini satellite DNA is used for this technology DNA fingerprinting. Differences I'll tell you. Microsatellites are shorter units, usually less than 10 base pairs, you know, shorter segments, 10 base pairs only will be AT, CC, GT, something like that. It is repeated. That much stretch, again repeated. That much stretch, again repeated. 10, okay? 10 base pairs, this much length, again and again it is repeated. Okay, mini satellite DNA is bit longer. Micro, it is shorter. Mini is little longer. So mini satellite, like among 10, more than 10, even till 100 base pairs are repeatedly arranged. I can say A, T, T, C, C, G, G, something like that. So much big stretch I will take. That is again repeated, again repeated, again repeated. Like that, mini satellite DNA is a longer stretch with more than 10 to 100 base pairs repetitive sequences. Then microsatellite DNA has like more A and T sequence, and mini satellites actually useful for our technology. This mini satellite is GC rich. Okay, GC are more in mini satellite DNAs. Clear? Then next. See,
From that time, I am saying satellite DNA is having high degree of polymorphism. Polymorphism means what? Variation at genetic level. So identification of these variations in the gene level is important for DNA fingerprinting and also genetic mapping. To because of that only, if you don't trace out the variations, how can you identify the criminal here? So for DNA fingerprinting technology and for arranging the DNA fragments in the sequence, this variation only will be useful. Okay, so these polymorphisms, main reason for the polymorphism is mutation. Okay, mutations are the main reason for the polymorphisms. Okay, so because of these polymorphisms, because of these mutations, polymorphism may happen in somatic cells, may happen in the germ cells. Children, are you following? So suppose polymorphism happens in somatic cell, like in the skin cells or something like that, and these polymorphisms will not get inherited to the next generation but show impact on our body functions. We'll be suffering lifelong, we'll be having the impact of it. Okay, suppose if these polymorphisms happen in the germ cells, like ovum or sperm, like that, like you will not have any impact of it, but it can get inherited from this generation to next generation. Okay, so germ cell, these two lines, no, they wrote it like khichdi only, but meaning is simple. Germ cell mutations will get inherited to the next generation without showing any impact on your characteristic features. Okay, so next one, they said whenever this polymorphism, the mutation, okay, in our terminology, is inherited to the next generation in the population, when it comes to population, like 1,000 members, the standard number for the population is like 1,000 individuals. So when it will be stabilized, that mutation you can see in the population, this generation, next generation, like that, means whenever frequency of that mutation is more than 0.01, this line also will create a difficulty in understanding for you. Meaning of this one is, like, out of 1,000, minimum, like at least minimum, more than 10 members show one mutation, like extreme IQ or extreme strength. Otherwise, any other talent you can consider there, the variation. At least in the population, more than 10 members have the same type of mutation. Chance is there for next generation it can get inherited, and you can see that talent in some people at least. Okay, less than 10 members have that mutation, it will be easily degenerated. Like it can get eradicated because less than some 5 members show one talent or some polymorphism. They may marry or they may not marry. If they marry also, they may produce children or they may not. Infertility or sterility will be there. So we don't know, accidentally they may die suddenly. So like that, the mutation can get eradicated from the population. So that's why what they are saying, that at least more than 10 members show the mutation, and that will be stabilized in the population. So the frequency of the polymorphism has to be greater than 0.01. Okay, next, polymorphism. This one also, remember, like you can see in the coding sequences, these mutations can happen in the coding sequence or can happen in the non-coding sequences. Okay, so polymorphism in the non-coding sequences will not undergo any change. It will not produce any protein. So it will be accumulated in your body and get inherited to the next generation. They're also again accumulated, get inherited to their next generation. So polymorphisms in non-coding sequences are more useful and for the evolutionary history to trace out. Because coding sequence based polymorphism, children, because to write everything and explain, time is not there. So understand clearly, any coding sequence, coding DNA sequence has the polymorphism. Okay, immediately it will show, like it will be expressed as a protein or something. Instead of normal condition, like mutation is there, abnormal protein will be produced. Because of that abnormal protein, you suffer from a disorder or something, some abnormal character, and you will die also with a disease. So coding sequence based mutation is killing the person. So from that population, that person, the mutation is eradicated. But non-coding sequence polymorphism, what is happening? It is getting inherited, accumulated. So non-coding sequences based polymorphism is important for evolution. Understood? Which polymorphism is important? Non-coding sequence polymorphism is important for evolutionary history. Coming to the procedure of DNA fingerprinting, Alec Jeffries, he's the founder for this and father of DNA fingerprinting. Actually, he used the probe as VNTR. I'll come to this. Actually, I told you already for this technology, mini satellite is considered. In that mini satellite, VNTR is the example. VNTR probe is traced out. How? First, isolation of DNA. Suppose in any area some murder happened and police people immediately go and search for the samples if they can get, other than the dead body sample, any other samples if they get. Okay, they collect those samples that they'll send to laboratory. Hyderabad also one center is there like that. So forensic department, they will send that. Then the lab technician there isolate the DNA from the sample, like blood sample or saliva, anything, the semen or something like that. So isolation of DNA. After that, devices are not there to read entire DNA. So definitely fragmentation is important by using restriction endonucleases. After that, by following agrogel electrophoresis, they separate the DNA fragments. You know, in this technology, shorter fragments move farthest and they go to the lower side faster compared to the longer ones. So by keeping the fourth step is like one tissue paper type, you can understand. So they keep nitrocellulose or nylon sheath just below that device. Easily it will come and stuck to that paper. So that is only Southern blotting. Southern is the name of the scientist who invented this transfer of DNA fragments. Blotting means transfer. Okay, blotting means transferring the DNA fragments to the nylon or nitrocellulose sheath. So isolation of DNA, fragmentation. Third step, separation of DNA fragments. Fourth step, Southern blotting technology. So after this, what happens? So now just imagine, like now in the lab technician separated the DNA fragments and Southern blotting also over. So now from that, he will search for the repetitive sequences. Okay, in that he will see the unique sequence, like that unique sequence, that polymorphism should be in such a way that it should not match with anybody in the world. Of course, this technology is failed on identical twins. So we accept that identical twin, the rest, all the individuals, we can apply this. Okay, so here, the one sequence, like he will trace out that this ATCC, some sequences, ATCC. This sequence is very unique and it's not matching with anybody. So he identifies, oh, so much difference is there. Okay, and he will consider that as the probe. Probe is, definition is important, single-stranded DNA or RNA tagged with a radioactive element. The role of probe is to identify, what's the role of probe? To identify complementary strand. To identify complementary strand. Complementary strand. Okay, this is the role of probe. So he separated. Lab technician has this probe ready with him. Now, in the meanwhile, police people will inquire like how this murder happened and where exactly this murder happened. Suspected 1, suspected 2, wife killed, son killed, or security guy killed. So like that, he will the police people get the information and their samples also. Suspected 1 sample, suspected 2 sample. Everybody's sample will be kept here. There also like DNAs are like VNTR. This is only na probe is called as VNTR now with high degree of polymorphism. Okay, the technician kept ready, right? Now VNTR, suppose it is extracted from chromosome 5, because chromosome to chromosome VNTR length varies, sequence also varies. Person to person also it varies. It's not same. In your body only, chromosome to chromosome, this VNTR sequence varies. Okay. So this this probe, he will leave in the suspected 1 sample, suspected 2 sample, like that. This probe identifies complementary strand. So wherever its complementary strand is there, it will go and bind and form the bands actually. Exactly, the band will appear. The last step is like autoradiography. Here by using the X-ray film, we can identify. I repeat once again these 2 steps.
So till here, hope everybody understood, like DNA fragments are separated. So one among the DNA fragment, which is like with a unique sequence considered as a probe, VNT are variable number of tandem repeats. Okay, so now they will use this probe to identify the complementary strand. Once it identifies the complementary strand, it will form the hybrid. Okay, that technology only, hybridization, like probe along with the complementary strand is like this it will be creating. Okay, so that is hybridization that we can identify by using the X-ray film, that is autoradiography. Understood? So whenever probes are single strand is left in the samples of suspected people, same chromosome we have to take, that time the probe will go bind, attach. Suppose this is one chromosome, so like this it will be there. No probes I have left, some thousands of probes I left, some thousands of probes. Chromosome means segment of the DNA or something. If you take, when you leave the probe, that will go wherever that complementary strand is there, it will form the double strand. So that appears like a band. When you see on the X-ray film, you can see like a band. See this one, textbook picture. Whenever you are separating fragments, if required, PCR technology can be used to amplify the DNA, and VNTR length also can be varied from 0.1 to 20 kilobases. Okay, so this is the length of VNTR and mini satellite it is. Then see here, this is the what to say, band pattern. These are repetitive sequences. 11 repeats are there like that. Okay, so here DNA from crime scene, and it is more matching with, this is suspected person 1, suspected person 2, and it is more matching with the B individual, not with A. I have some more pictures too. I think you will laugh at this picture. If I look at the watch, hey, how come time is so? So okay, fine. So here, this is the, I don't know how much it is visible for you. So this is the crime scene sample, and it is more matching with whom? Second person. Okay, like this only autoradiography. When they see in the X-ray film, it will be. So next, which chapter shall I take? DNA fingerprinting is over. Human reproduction and reproductive health, I'll take it up. Someone is asking me this active immunity and passive immunity in health and diseases. Let's see. If you are hungry, go have food and come faster. Okay, no problem. Huh? Okay. No problem. We are human beings. Which one, children? Which one? Reproduction. It is. Yeah. At least main topics I want to, weightage is more. Reproduction and reproductive health I can't ignore. Yeah. I'll try to complete faster. Okay, human reproduction. So in this paragraph, focus on the sequence of the steps. Okay, reproductive events which happen in the individual, in human beings. Gametogenesis first, followed by insemination. Third step, fertilization. Fourth one, implantation. Fifth one, gestation. And last one is parturition. Every step is like hormonal influenced. Okay, and gametogenesis and all, if you see, lot of difference between the male and female also. Okay. Gametogenesis mainly happen in the primary reproductive organs in males in testes and in female in ovaries. And spermatogenesis is the process of gametogenesis happen in males, and this begins in puberty and continues in the till death. Okay, in female, it begins in the fetal life itself in the embryonic stage, continues till 50 years of age. Insemination is about transfer of gametes, male gametes to the female genital tract. Fertilization happens at ampulla region in female body. Okay, it's a fusion of male and female gamete to form the zygote. Okay, and formation of blastocyst and its attachment to the endometrium is called as implantation. And gestation is about the total embryonic development in human beings. The gestation period lasts about 9 months. 9 months. Okay. And it's about 38 weeks from fertilization day. If you take it, 266 days. How many? 266 days. Gestation, I'm saying. Gestation period also varies between all the animals. Elephant has longest gestation period, 641 days actually. 607 to 641 days of gestation period is there in elephant. Okay, and delivery of the baby is called as parturition. I will highlight the hormones also here. Next, male reproductive system includes a pair of testes, accessory ducts, glands, and external genitalia. And testis actually situated outside the abdominal cavity in the scrotum. It's because to maintain the temperature, actually 2 to 2.5 degrees centigrade is lower than the body temperature, so that is useful for spermatogenesis. Okay, and it's not like, suppose statement is given like in all mammals testis is situated outside the abdominal cavity, means it's wrong, because a lot of variations are in elephant and all that. Always it is intra-abdominal only. Okay, in certain organisms seasonally it is coming outside and placed in the scrotum, suppose for spermatogenesis. After that, if seasonally they are not active for the reproduction, it goes inside. So like that, a lot of variations are there. In humans it is situated outside. Okay, and length is 4 to 5 centimeters and width is 2 to 3 centimeters. And actually in each testis, if you see, 250 compartments are there. 250 compartments in each. All these are called as testicular lobules. In each testicular lobule, 1 to 3 highly coiled seminiferous tubules are there. 1 to 3 seminiferous tubules are there. So totally in each testis, if you see, 250 compartments, each one has maximum 3 seminiferous tubules. Total one side, how many will be there? 750 seminiferous tubules are there. Both together 1500 in the individual. Okay, and seminiferous tubule, one tubule if you imagine, then the cross section shows that presence of 2 types of cells over there. One type is spermatogonia, sperm mother cells, which are very important for spermatogenesis. The other type of cells are Sertoli cells. Remember Sertoli cells otherwise as nurse cells or sustentacular cells. Okay, they provide the nourishment for the developing sperms actually. And one more is the main point, PYQ, Sertoli cells in male can produce inhibin to check the production of excess FSH. So the negative feedback mechanism is there on FSH. So Sertoli cells can produce inhibin. Inhibin actually can be produced in female and also male. Okay, next one. These and ABP, certain factors are produced by Sertoli cells which are actually useful for efficiency, to increase the efficiency of testosterone and all that. Okay, next one, spermatogonia undergo actually both mitosis and also meiosis. Let me focus on the spermatogenesis process later on. Between seminiferous tubules, Leydig cells are interstitial cells are present. ICSH, interstitial cell stimulating hormone or LH, acts upon these cells and make them to produce androgens, which are important for maturation and development of accessory ducts and all that. Okay, sequence of accessory ducts is important. So first, sperms produced in seminiferous tubules comes out. Accessory ducts begin from rete testis. First enters in rete testis, later goes to vasa efferentia. After that goes to tightly coiled structure. What is that? Epididymis. Epididymis, after that vas deferens. After that ejaculatory duct. After that urethra. Once again, rete testis, vasa efferentia, epididymis, vas deferens, ejaculatory duct, urethra.
The opening of urethra is urethral meatus. One more highlighting point here. Epididymis is very important for maturation of sperms, okay, and also to increase the motility of the sperms, epididymis plays very important role, okay. The next one, yeah, external genitalia in male reproductive system are penis and scrotum. You think that only penis is the external genital organ, but actually even scrotum also you can include in this list. And glans penis, I think terminology you are aware. And male accessory glands, paired ones are seminal vesicles, unpaired one is prostate, and these bulbourethral glands are also paired only. Bulbourethral glands are otherwise called as Cowper's glands, and sperms along with the secretions of all these, and also the epididymis onwards, the tubular secretions also all together form semen. Semen pH is like slightly alkaline, 7.2 to 7.4 like that, okay. That supports the viability of sperm. So that's why in male body, sperm's viability is like for many days. But when it is introduced into female body, the lifespan of the sperm is reduced to 72 hours only. Okay, 3 days, and most efficiently like 2 days actually, but still it is alive one more day also, 72 hours of lifespan for the sperm. Okay, next, bulbourethral glands, Cowper's glands secretions mainly are useful for lubrication of penis. Okay, female reproductive system has a pair of ovaries and oviducts, uterus, cervix, vagina, external genitalia, and accessory glands are present. And these ovaries, testes are example for heterocrine glands also. They can produce, and primary structures here. So they can produce gametes and they can produce sex hormones. Okay, and fallopian tube length also you remember. Fallopian tube division is like fimbriae, finger-like structures receive ovum and all. Then what funnel shaped structure is infundibulum, and the widest region is ampulla, and later the narrow tube is like isthmus, and isthmus opens into uterus. Uterus is inverted pear-shaped sac-like structure, okay, and it is what to say, uterus has 3 divisions: fundus, carpus, body, and the posterior part is called as cervix. The cavity in cervix is cervical canal. Cervical canal actually with vagina forms birth canal. So for delivery and all it will be useful. Okay, and the wall, uterine wall has 3 layers: perimetrium, myometrium, endometrium. Okay, the nature of these 3 layers also you have to go through, children. Nature means, yeah, this one. Perimetrium is thin membranous, thick smooth muscle is present in myometrium. Myometrium plays a role in parturition, and inner glandular layer is endometrium for during implantation time and even menstrual time and all that, that is only involved. Okay, next, external genitalia includes mons pubis, labia majora, labia minora, hymen, clitoris. This is the list. Here clitoris is tiny finger-like structure which lies at the upper junction of what? To labia minora, important, and PYQ. And next one, hymen, this statement also you can go through. A pair of mammary glands are there. Mammary glands are modified sweat glands actually, okay, breast we say, and situated on pectoralis, major muscle, and made up of fibrous tissue, what to say, glandular tissue, adipose tissue, and all that. And in each mammary gland, the structural and functional units are alveoli, which can secrete milk and can store the milk also. And each mammary gland is with 15 to 20 mammary lobes. Okay, 15 to each mammary gland, not both. Okay, 15 to 20 mammary lobes. Like first, milk produced in alveoli. See, one more, I will make you recall here. Milk formation is under the control of prolactin. Milk ejection is under the control of oxytocin. One more, mammary gland growth, development, everything, estrogen, progesterone. The first one priority, like majorly estrogen contribution, even along with progesterone, it can be developed. Okay, and tubular sequence. Alveoli, the milk from alveoli enters into Mammary tubules. Mammary tubules. Mammary tubules join to form what? Mammary ducts. Mammary ducts join to form mammary ampulla. Mammary ampulla joins to form lactiferous duct. From lactiferous duct, milk and through nipple it is ejected. Okay, this sequence also 2-3 times in final exam they asked. So don't ignore. Once again, alveoli, mammary tubules, mammary ducts, mammary ampullae, lactiferous duct, and this one opens at nipple and milk is ejected because of oxytocin. Okay, next important topics: spermatogenesis and oogenesis. Oogenesis actually, main points I'll. And in one PYQ children, they asked this one also. Yeah, what are the components present in the sperm like that? Yeah, sperm. Middle piece mainly, remember. So actually fine. Total sperm, if you see head, neck, middle piece, tail. Okay, in head, elongated nucleus is present, and the anterior to the head actually cap-like structure, acrosome is present. Acrome is modified Golgi complex of spermatid. So in spermatid, Golgi complex is there, that is modified into acrosome, which is useful to penetrate into secondary oocyte during fertilization. Okay, because a lot of enzymes are stored over there. Then in the neck portion, remember, in the neck portion of sperm, only 2 centrioles are present, proximal and distal centriole, that's all. Then in the middle piece, mitochondria, spirally coiled mitochondria, nebenkern, we call this as nebenkern. This mitochondria is present in the middle piece of the sperm. And not only that, and so axial filament is there, cytoplasm is there, okay, in the middle piece. Then tail portion, total sperm is enclosed by plasma membrane. You know, at a time, like per ejaculation, 2.5 to 5 ml of semen is released actually. In that, 200 to 300 million sperms are there. Okay, among these 200 to 300, 60% of them are like with normal shape, size and all. Okay, 60%. First you find out the 60% of 200 or 300 something. Whatever you got, like the normal shaped ones you got, from them 40% of them are having vigorous motility, faster movement. Okay, so 40% of the sperms, like don't consider that among 260% of them with shape, proper shape, and 40% will be vigorous motility. It's not correct. Okay, what you need to consider? Among 200 to 300 million sperms, 60% of them are normal. Again, in that normal sperms only 40% of them are having vigorous motility. Okay, shall we start spermatogenesis? Yeah, some children have doubt here also. Spermatogenesis is easier actually. First here multiplication phase. Multiplication phase, growth phase, maturation phase, like the 3 phases are there. And first the precursor for spermatogenesis. This is spermatogenesis, formation of sperms in seminiferous tubules. Okay, so the precursor cell is what? Spermatogonium. Spermatogonium. So mother cell, male germ cell. So 2n condition, 46 chromosomes are there. Okay, and first it undergoes mitosis division. Mitosis, so many cells are produced. So multiplication, so many cells are produced, daughter cells are produced of same size only. Just mitosis occurs here. Little differentiation like spermatogonium A, spermatogonium B, like that it will be differentiated. So next, this one spermatogonium, no need to consider the remaining, because always we need to consider the concept with one particular spermatogonium only. Each spermatogonium undergoes growth phase and increase in the size. I am showing the concept with one spermatogonium. This is also spermatogonium, children. Don't get confused. Okay. So in textbook, actually, blue cells you notice. 2 blue cells are given. It's wrong actually. 1 blue cell only they have to give, because one spermatogonium can produce one primary spermatocyte. No doubt in that. Okay. Growth phase. So this is primary spermatocyte. Suppose 100 spermatogonia are there. How many primary spermatocytes are produced? 100 only. So only growth phase, na, right? That's why here also 2n condition, 46 chromosomes are there. Hope everybody is clear. So one spermatogonium, first spermatogonium undergoes multiplication phase, mean mitosis, and increase the number. That's over. That keep that aside. Now one spermatogonium which are produced, that one structure increase in the size and become like primary spermatocyte. Okay, this primary spermatocyte now begins maturation phase. Maturation phase means meiosis 1 and meiosis 2. Meiosis 1 produce two equal sized cells actually. This is equational only, and these are called secondary spermatocytes. Secondary spermatocytes. So meiosis 1 is actually reductional division, where the ploidy is reduced to half exactly, and so the first in this entire process, the first formed haploid cells are secondary spermatocytes. Okay, then after that, this is what phase it is called as maturation phase. Which cell first starts maturation phase means primary spermatocyte. Okay, secondary spermatocyte continues its division. So this undergoes meiosis 2. Here ploidy will not be changed, same only will be maintained, and this secondary spermatocyte also continues meiosis 2 like this. The products are called here spermatid. The daughter cells are called as spermatids. So this is also with haploid set of chromosomes. And each spermatid now undergoes one more process important, that is transformation of spermatid into sperm. That is called as spermiogenesis. What is it called as? Spermiogenesis. It is otherwise called differentiation process. Differentiation process. Okay, so one spermatid is changed in this process to like one spermatozoon. Okay, so like this. Suppose 100 spermatids are there, 100 sperms are produced. Okay, so these sperms are also finally, these are also spermatozoon I can say, or sperm I can say, haploid set of chromosomes are only present. Let me make you clear now that from one spermatogonium, how many primary spermatocytes are produced? From one spermatogonium, one primary spermatocyte is only produced. So hope it is little visible for you. Yeah. So 100 spermatogonia are there, 100 primary spermatocytes can be produced. That's over. Now one primary spermatocyte let us take. Now how many secondary spermatocyte each one is producing? 2 secondary spermatocytes. Suppose we have 100 primary spermatocytes. How many secondary spermatocytes can be produced? 200. So here 100 are there, 200. Is that over? Now each secondary spermatocyte, if you take, each one can produce 2 spermatids. Each one can produce 2 spermatids. So here if I take 100 secondary spermatocytes, how many spermatids are produced? 200 spermatids are produced. And now one primary spermatocyte, if you see here, how many secondary spermatocytes it is producing? 2. So the result finally after meiosis 2, from one primary spermatocyte or one spermatogonium, anything you can take, and finally 4 spermatids are produced. And if I take 100 here, and finally 400 spermatids are produced. No confusion, very important topic it is. Okay, and these spermatids again undergo, like for this Sertoli cells based certain factors are also important. That's why spermiogenesis, transformation of spermatid into sperm, that is called as differentiation. At that time only Golgi complex of spermatid is changed to acrosome, nucleus is elongated, a centrosome is divided into centrioles. All these type of changes happen in this differentiation process only. And once after sperms are produced, immediately they go attached to the Sertoli cells are there, right? So they go and attach to the Sertoli cells. So like this they attach, like this, all the sperms are attached to Sertoli cells. So with the effect of these hormones and all, that FSH hormone and all, because FSH only act upon the Sertoli cells. So because of the influence of LH LH acts upon Leydig cells for production of androgens, FSH act upon Sertoli cells, okay, and for this spermiogenesis and all it will be useful. So because of the influence of FSH and detachment of detachment of sperm head from Sertoli cell happen, fall into the lumen of seminiferous tubule, and that time it will move out of the seminiferous tubule. I repeat once again because this is the assertion reason in PYQs 1 year back, okay. So spermiation process, actually it is about what is spermiation means, detachment of sperm head from Sertoli cell, and sperms at that time fall into the lumen of seminiferous tubule. Obviously, no, it is suppose here something is detached, it will fall into the empty space only, and later it is pushed out of that particular tube. So that is only like falling into the lumen, moving out of the seminiferous tubule altogether. This is all called as spermiation. So detachment of sperm from Sertoli cell fall into the seminiferous tubule and moving out of the tubule, that is called as spermiation process. Okay, so that is next. I told you the hormones LH role, I told you FSH role, FSH on Sertoli cells I told you, and one more, Sertoli cell can produce inhibin also to give a negative feedback mechanism on FSH. Okay, that inhibin Sertoli cell can produce inhibin, that also you remember. Okay, next about oogenesis. So clarity is there now. One sperm, one spermatogonium. Finally, how many sperms it can produce? 4 sperms. Okay. Oogenesis means formation of oogenesis is about formation of ovum from oogonium. Simple to understand actually. So one oogonium which is diploid with 46 chromosomes. Okay, first even this oogonia also, like it begins in the fetal life and mitosis happen and multiplication is there here also, and each oogonium is converted into one primary oocyte. This is actually not continuous. A lot of concepts we need to recall here. Primary oocyte, and it undergoes meiosis 1. From there itself, array, important topic. He will connect. In the meanwhile, I'll. Very black bottle is there outside gate, no? Black bottle outside. Ouch. Shall I start? Everywhere it is delivered. Now check no. Whether in classes it's coming or not. Sure. Okay. From where it was not there. Chart oogenesis process. Okay. Okay. In certain campuses it was not delivered. It seems. Yeah. See here. So oogenesis process begins in fetal life. Okay. So first in each ovary. In fetal life, in the embryonic stage itself, oogenesis, like when it began, like in each ovary, a couple of million oogonia are formed. Later on in the life, it will not be newly formed or newly added. That's all the final number. Okay. Each oogonium involves in growth phase and converted into primary oocyte. So each one is now grown. Like each oogonium is changed into primary oocyte, one to one like that. And this primary oocyte immediately starts its meiosis 1 and arrested in the prophase 1 stage only, because certain nutrients factors are not available to continue the division. So arrested in the prophase 1 itself. itself. And each this primary oocyte surrounded by the protective layer, like follicular cells, I can say, or granulosa layer, like that, a thin layer is formed. So now this entire structure, like inside primary oocyte is there, na, like covered by follicular cells, it is now termed as primary ovarian follicle. This is all. Like now 9 months are over, like delivery happens and baby is born, and when baby is born, in her ovaries she has all the primary ovarian follicles. In each follicle, what stage is there? Prophase 1 arrested primary oocyte. This is one of the important points in the oogenesis. Like when baby is delivered, what is the stage of the cell present in her ovary? Means primary follicle with prophase 1 arrested primary oocyte. Okay, prophase 1 arrested primary oocyte is present. Okay, next, childhood, same thing will continue, no change. Okay, so same stage will be there, and she enters into her puberty, like you can consider some 12 years, 14 years or something. So puberty, at that time also till puberty, till 12, 14 years, not much change. Okay, puberty, why change will be there? Means more FSH, LH secretion begins, because first GnRH is produced by hypothalamus, and which stimulate our anterior pituitary, so that pituitary gland starts producing the gonadotropins. Okay, this because of the influence of FSH, majorly FSH, and even LH is also present. I can't say it is often. Majorly FSH is important for the follicular development. So from puberty stage onwards, see the difference. See, now one more point also I have to tell you. When baby reaches to puberty, so her primary follicle number is reduced highly, and only 60,000 to, when she reaches to puberty, 60,000 to 80,000 ovarian follicles only remained. Follicles remained in each ovary. Each ovary remained. That's the important step. Okay, the rest all, because each earlier in each 2 million were there, like each a couple of million, I said, 2 million oogonia converted into primary follicles and all that. Okay, so finally only 60,000 to 80,000 in each ovary it is remained. The rest all degenerated. That process of degeneration of ovarian follicles is called as follicular atresia. Follicular atresia, and those degenerated follicles called as atretic follicles. Okay, now coming to the actual process, those 60,000 to 80,000 are there, no? Each month, suppose if I take my own life, like from puberty, every month from those, like in each ovary 60,000 to 80,000 follicles are left, right? So every month from that puberty stage in my life, like 10 or 8 to 10 follicles, no, will be undergoing the developing stage. Okay, so because of the influence of FSH, LH and all that, every time like only one follicle finally produces ovum, but even the other some 7, 8 also extra will be undergoing the procedure. Okay, so later on everything will be delivered. Once after ovum is released, the rest 7, 8 whichever would have followed the procedure get degenerated. Like that only, till like all the 60,000, 80,000 follicles get over, only we will be having the fertile period. That's the reason why menopause occur, means because of this reason only, because follicles in the ovary get over. That's the reason, though FSH availability, all this is there also, as follicles are not available in the ovary, female this menstrual cycle will be ceased. Okay. So now in puberty period, because of FSH, what happened? Primary follicle. Now primary oocyte arrested in prophase, surrounded by what? Granulosa layer. This is primary follicle. Primary follicle changed to, because of what? FSH, influence of FSH, it is changed to secondary follicle. Secondary follicle little more bigger, and one outer covering, this granulosa, thick granulosa layer is there. Thick granulosa layer is called as theca. It is called as theca. Okay, in this primary oocyte only, primary oocyte arrested in prophase 1 is only present, no change. See, gamete primary oocyte is not changed from fetal life to even till the puberty stage and all this. Puberty stage also among 60,000 follicles, every month only 7-8 only will be undergoing the procedure. The remaining will be arrested always in this stage only. Okay. So secondary follicle, again FSH influence this and change this one into tertiary follicle. Tertiary follicle here, the theca is differentiated into theca externa and one more is theca interna. Theca externa, theca interna. Among these two layers, theca externa, theca interna, theca interna is secretory. It secretes estrogen. Okay, that's important. That's why whenever follicles are undergoing development process, no, even in the body, the female body, estrogen levels also gradually will be increasing. Okay, and here and now this primary oocyte, the arrested in prophase 1. It has to complete its divisions. That's why properly see, like it's holded properly, cumulus oophorus, this hill-like structure. It fixes properly in the position. Okay, and this tertiary follicle is having a unique feature to have it. What is this? Fluid-filled cavity present in tertiary follicle is called as antrum. Fluid-filled cavity present in tertiary follicle is called as antrum. The fluid name also liquor folliculi, we say, all nutrients are there. The support of all the factors and all that. This primary oocyte completes its meiosis 1 in the tertiary follicle. Okay, when primary oocyte completes meiosis 1, what will be produced? Secondary oocyte. Secondary oocyte, larger cell and one tinier cell, first polar body. So when the meiosis 2 is almost continuously happening, I mean secondary oocyte again continues its meiosis 2. So that time we consider this tertiary follicle as a mature tertiary follicle or Graafian follicle. Okay, little later stage, that's all. In the tertiary follicle only meiosis 1 is completed. That somewhere you write it down. Meiosis 1 is completed in tertiary follicle. Meiosis 1 is completed in tertiary follicle. Okay, and again meiosis 2 begins. Meiosis 2 begins in tertiary follicle. Okay, so once after it is arrested in metaphase 2, secondary oocyte is arrested in metaphase 2. Now we consider this as Graafian follicle, and in the meanwhile LH surge would have happened. So because of that, Graafian follicle ruptures. So Graafian follicle ruptures and releases the secondary oocyte, which is arrested in metaphase 2, arrested in metaphase 2. So this is secondary oocyte arrested in metaphase 2, and it is surrounded by a new layer that is called as zona pellucida. This point also important. Zona pellucida is a non-cellular layer, no living cells present. Okay, it's made up of glycoproteins. Zona pellucida. That ovum itself can secrete that. Okay, then on top of it, loosely arranged this granulosa cells are present, and this is called as corona radiata. Okay, so corona radiata, zona pellucida additionally present around the secondary oocyte. So this process, like because of LH surge, Graafian follicle is ruptured and releases the secondary oocyte, arrested in metaphase 2. Metaphase 2. This process is called as ovulation. Generally in the mid of the menstrual cycle and 14th day it happens. Okay, and one more, and inside the that time in this, because of the presence of LH only inside the ovary, this ruptured Graafian follicle is converted into corpus luteum. This is called as corpus luteum. Corpus luteum, and after 12 days, this if she is not pregnant, fertilization is failed, then corpus luteum is changed to corpus albicans. This is the process. Okay, now we will because oogenesis, right? Finally, functional ovum has to be released. So in most of the females, unmarried females, okay, so generally the ovum will get degenerated in this stage only. Why? Because oogenesis will be completed. See now, Now, in that particular girl, okay, so wherever this process is happening, every menstrual cycle 14th day, ovum will be released. Where does that ovum goes? The ovum is picked up. This is uterus. So this is the ovary. So now secondary oocyte is released, no? So that goes to the ampulla region and it will be here. So ampulla, this is the ovum. And now monospermy is allowed in our body. So only one sperm will be entered. And if suppose two sperms or something enters, then fertilization is failed. Okay, only one sperm enters here. So whenever sperm enters in the ampulla region, after sperm entry, sperm also can enter into this oocyte waiting over in the ampulla region after capacitation process. Capacitation is the phenomenon in which sperm is again becomes more active and eligible to enter into secondary oocyte. This was also PYQ. Capacitation process means activation of sperm, like to enter into secondary oocyte so that it is eligible for fertilization. This happens in the for sperm. This type of activation capacitation occurs in the female genital tract only. Okay, at that capacitated sperm enters into secondary oocyte. After sperm enters into secondary oocyte, that metaphase 2 is broken, promoted to anaphase 2, and after that it is promoted to telophase 2. Like that, this particular the nucleus becomes like ooted nucleus, and second polar body is produced, and that comes out into enter into the perivitelline space between this zona pellucida and ovum membrane. Perivitelline space is there. So during fertilization, that second polar body is produced and get degenerated also later on. Okay, so now question will be asked, when exactly functional ovum will be produced means after sperm enters during fertilization. Okay, so this fusion of ooted nucleus and sperm nucleus is called as fertilization. Like that, after, so when actually functional ovum is formed means after sperm enters only. So whenever sperm is available only, oogenesis will be completed in the ampulla region. If sperm availability is not there, the secondary oocyte, it waits for 24 hours. That's all, 1 day lifespan. So that's all in the ampulla, and later it gets degenerated. Okay, if sperm availability is there only, after sperm enters, that secondary oocyte is divided to ooted and second polar body. So finally, so here what points you have to recall is oogenesis begins in fetal life. Okay, when baby is delivered in her ovaries, prophase 1 arrested primary oocyte is present. Okay, then by the time puberty she reaches, due to follicular atresia, okay, many follicles degenerate and only 60,000 to 80,000 follicles remain. Okay, then after that, then gradually due to this hormonal FSH, LH influence, the development of follicles occur. Then in tertiary follicle, meiosis 1 is completed. Meiosis 1 began in fetal life, completed in tertiary follicle. Every month it's continuous. Okay, so meiosis 2 also begins in tertiary follicle only. Okay, when the Graphene follicle is ruptured and ovum is released, that time secondary oocyte is arrested in metaphase 2. Okay, so that is only ovulation. Like that reaches to ampulla region. Then if sperm enters into that secondary oocyte only, then secondary oocyte is divided into ooted and second polar body. Children, okay, this is oogenesis. Okay, so when meiosis 2 is, where meiosis 2 begins, tertiary follicle, where it is getting completed in the ampulla region after sperm enters. Okay, then menstrual cycle, some children asked. Only hormonal condition I'll highlight here. And parturition also hormonal control I have to highlight. Menstrual cycle, 28 days cycle. I think you might be hungry. Children, you might be hungry. If you are hungry, go have food and again settle down. 28 or 29 days period. In female primates it happens, monkeys, apes and human beings, because non-primates generally undergo estrous cycle. Okay, and first menstruation is called menarche and last one is called as menopause. Okay, and now first in the stages are menses, menstruation phase, next
follicular phase. Follicular phase, otherwise called as proliferative phase. Proliferative phase. Then ovulation phase. Ovulation phase. Then last, luteal phase, otherwise called as secretory phase or secretory phase. Okay, so menstruation phase. When exactly menstrual cycle begins in the female, menstrual cycle actually happens in the female is due to withdrawal of progesterone. Remember that point, very, very important. Due to withdrawal of progesterone only menses occur. The next menstrual cycle begins. Okay, because remember here totally 4 hormones are involved. What are those? FSH, LH, estrogen, progesterone. What are the hormones involved for regulation of menstrual cycle? FSH, LH, estrogen, progesterone. Estrogen, progesterone. See, FSH, LH actually pituitary hormones. Okay, estrogen, progesterone actually ovarian hormones. See the statement here. Pituitary hormones control ovarian events in the menstrual cycle. Ovary. Okay, and ovary based hormones means estrogen, progesterone, ovary based hormones control uterine events. So only 2 types of events you are seeing, situations you are seeing, ovary based, uterus based in the menstrual cycle. Okay, so ovary based changes are because of pituitary hormones, uterus based changes are because of ovarian hormones. Okay, that statement you remember here. So first I told you that the menstrual cycle begins because of the withdrawal of progesterone. Okay, because among this, estrogen increases the thickness of endometrium. Progesterone is important for maintenance of endometrium. Okay, when progesterone is very less, then endometrium is not maintained, and that time like endometrial ruptures of blood vessel, everything like the pieces it becomes, and it will be leaked. So when fertilization is not happening, what will happen? This all this endometrium, the ruptures of blood vessel, everything like pieces, it will be shed actually through vagina. This is only menses. Okay, and next one is like generally 3 to 5 days this phase lasts about. Okay, 3 to 5 days. At that time, you can consider like progesterone is less at that time. Okay, I think it will not overlap, no? So estrogen is less. Okay, then FSH is less, LH also is less. So the during menstrual phase, all these hormones are less in amount only. The main reason for the menstrual cycle to begin is like withdrawal of progesterone only, but all the other hormones are also less only. And next, follicular phase. Follicular phase is happening in the ovary. Proliferative phase is happening in the uterus. Endometrium thickness increases. Okay, so in the follicular phase, actually, gradually, like primary follicle is changed to secondary follicle, secondary is changed to tertiary, graphene follicle, like that. Developing follicles, like see, FSH, LH are important for development of the follicles in the ovary. This is happening in the ovary. Clear? The same time, parallelly, as developing follicles produce what? Estrogen. So because of the increased amount of estrogen, endometrium thickness also increases. Thickness of endometrium also increases. Okay, so only FSH is more, LH is more here, estrogen is more in this phase, follicular phase and or proliferative phase. Follicular phase is follicular development is happening here. The developing follicles are producing estrogen, so gradually estrogen amount also increases. That goes to the uterus and increases the thickness of the endometrium by proliferation of the cells. That's why name is given as proliferative phase. Clear? Then now here forget about progesterone because source is not there to produce it. It was already less and the same thing continues here. Okay, next ovulation. Like
LH surge, suddenly LH surge occurs because of, some says that because of increased estrogen or something, but suddenly LH surge occurs here because of that, because the wavelengths vary actually. That's why sometimes positive feedback mechanism, sometimes negative feedback mechanism, all this will be operated. Okay, due to LH surge, Graafian follicle is ruptured and secondary oocyte is released, which is arrested in secondary oocyte arrested in metaphase 2. Okay, this is only ovulation. How to find out? Generally, it happens on 14th day of mid of the menstrual cycle, and sometimes it varies in certain females. It's not like in every female on 14th day only it happens. How to find out the day of ovulation? Like suppose the duration of menstrual cycle in certain female is like 32 days or 36 days, something like that. Then how to find out the day of ovulation in that particular female? So day of ovulation is equal to number of days in menstrual cycle, minus 14. This 14 related to luteal phase. Actually, in the menstrual cycle, longest phase is luteal phase only. Okay, so 14 is standard. Okay, now number of days in menstrual cycle, generally 28 days. Generally 28 minus 14. Normal condition 14th day is the ovulation day. Now I asked you the female menstrual cycle duration. I said that 32 minus 14. Find out the ovulation day then. 32 I said her duration, and minus 14. Which day she ovulates? 18th day. Okay. Is it? Shall I believe? Yeah. Okay, next about, so like that you can find out. Then after that, in the presence of LH, the ruptures of Graafian follicle. So the Graafian follicle converted into corpus luteum. Ruptures of Graafian follicle converted into corpus luteum. So that's why it is luteal phase. And this corpus luteum is more secretory. It secretes progesterone and all that. So this produces progesterone, high amount of progesterone. Okay, and even little amount of estrogen also it can produce. Inhibin, relaxin, all these hormones it can produce based on the situations. Okay, so not only progesterone, estrogen, relaxin, inhibin, all these hormones also corpus luteum can produce. Corpus luteum is considered as temporary endocrine gland in ovary. Okay, so if that released ovum undergoes fertilization in the ampulla region, then this corpus luteum maintains the pregnancy. Okay, because progesterone is important for conception, right? Implantation and all that. So this corpus luteum is maintained for 3 months. Actually, first trimester of pregnancy is totally maintained by corpus luteum based progesterone. Okay, if fertilization is failed, then within 12 days, actually 12 to 14 days, actually this will be degenerated and becomes like corpus albicans. Children, okay? And now one more point, like I'll show you this graph given in the textbook. Did I open the textbook? Array. The graph I have to show. And in this graph, you can see you will be having textbook. You can see FSH, LH. You notice exactly whichever is at peak. So highest amount, that is LH. Little lesser one is FSH. Among estrogen, progesterone, 2 peaks are there for estrogen before ovulation and after ovulation. 2 peaks are there for estrogen graphical representation, and only after ovulation the peak is present in progesterone. After ovulation, peak is there for estrogen and progesterone, but 2 peaks like before ovulation and after ovulation for estrogen you consider, and after ovulation for progesterone you can consider. Okay, so how exactly next menstrual cycle will happen? Yeah, this is what I was expecting. Array, now again what is the problem? Move. Ayyo. I would not have opened this.
Food will get over. Children, you go have hostelers, you can go have food and come back. No problem. My God. Ayyo, it's hurry. Again, it will take time, I think. I don't know. So what I want to tell you is like when it comes to, on graph you can see, when it comes to 22nd day like that, 23rd, high amount of progesterone, high amount of estrogen is there in the female body, in female circulation. So because of that, good, at least this is supporting now. So high amount of progesterone, high amount of estrogen have negative feedback mechanism on FSH and LH. High amount of progesterone, mostly it inhibits the production of LH. Okay, when LH is not there, because of high amount of progesterone, LH is not there. When LH is not there, corpus luteum is not maintained. So important point you have to consider, corpus luteum is formed because of LH, maintained because of LH. If LH only not there, it can't be maintained. So because of high amount, LH is not there. So gradually corpus luteum size will be decreased, and so progesterone amount also will be decreased. And because of that, endometrium is not maintained, and it will be like ruptures again because of the less amount of progesterone only, the menses occur. Okay, next menstrual cycle begin. Okay, next about. Yeah, fertilization occur at ampulla and monospermy is allowed. And one more point here, to produce identical twins, one fertilization only is the reason. Okay, identical twins are produced because of the result of one fertilization. For non-identical twins to produce, two separate fertilizations have to happen. Okay, two sperms, two different ova, like that. Then fraternal twins or non-identical twins are because of the result of two different fertilizations. Acrosomal reaction, cortical reaction, all that we'll say. Okay. Sex determination occurs when fertilization occurs, zygote is formed only. And in this picture, children, zygote undergoes repeated mitotic divisions actually. Those are called as cleavages, and 2-celled stage, 4-celled stage. Where exactly it is situated in the female genital tract, that also you notice. Okay, so because in reproductive health chapter, ARTs are there, no, assisted reproductive technology, ZIFT, IUT, where which one has to be injected to the fallopian tube uterus, that can be understood clearly if you know this particular picture. Okay, so 8 to 16 blastomeres are there in the embryo, that's called as actually morula. And when it becomes like more than 16 blastomeres, then we can term that as the blastocyst. Blastocyst outer layer is called as trophoblast. Trophoblast gradually it secretes HCG also. In pregnant women, actually pregnancy test also confirmed because of the presence of HCG only. Okay, and one more, the inner cell mass present in the blastocyst, that is important for organogenesis. Okay, from this trophoblast only chorionic villi appear, and that will be interdigitated with the endometrium. And after fertilization, exactly 6th day or 7th day, implantation begins. Okay, once implantation is over, then only we consider the women as conceived. She's like from that time, like she's like pregnant women, like that. Okay, and one more, and once implantation is over, after gastrulation, gastrulation actually occurs. During gastrulation, only 3 germ layers are produced. First of all, this inner cell mass actually arranged as 2 layers, epiblast, hypoblast. Epiblast is the source for 3 germ layers, ectoderm,
Endoderm, mesoderm. First formed germ layer is endoderm. Second, mesoderm. Last formed germ layer is ectoderm. But while writing and in textbook, actually little different way they mentioned. But the fact is, the first formed germ layer is endoderm. Next one, mesoderm. The last formed germ layer is ectoderm. Okay. And 4 extra embryonic membranes are also produced. Chorion, amnion, allantois, yolk sac. They labeled it actually here. You see these labelings, or yolk sac and all that they labeled. What is the problem with this? Okay, anyways. So chorion is ectoderm and mesodermally derived. Even amnion also ectoderm and mesodermally derived. Allantois, yolk sac are mesoderm and endodermally derived. Why I am saying this one is in 2017 or 18 paper they asked about chorion, amnion and all. That's why I'm saying these are the 4 extra embryonic layers: chorion, amnion, allantois, yolk sac. Chorion and amnion are derived from ectoderm and mesoderm, and allantois and yolk sac are derived from mesoderm and endoderm. Okay, yolk sac is the first hemopoietic organ in our life. Fine. So go through this one, the events which happen in the gestation period. The first sign of growing fetus is noticed by the listening of heart sound. Okay, and by the end of the 2nd month only, all the digits and limbs, everything is formed. And actually major organ systems, everything formed in the 1st trimester only. 2nd trimester is actually the main growth is maintained, and first movement of the fetus is noticed in the 5th month, during 5th month only. And by the end of the hair appears also in the during 5th month only. By the end of 24 weeks, actually totally body will be covered by fine hair, eyelids are separated, lashes are formed. And 9th month, the myelination of neurons, this type of nervous system based changes majorly take place here. And this is about the embryonic development. And once the fetus is matured, 9 months are over, then signals, the certain factors are released by fetus and also placenta, and that triggers mother's hypothalamus, and that produces oxytocin. Okay, so because of that, myometrium of the uterus will be triggered, and labor pains begin. Okay, and oxytocin has actually positive feedback mechanism, so that it's a fetal ejection reflex, we call this. Because of that only, gradually oxytocin production will be there. Oxytocin related, again I repeat, produced by hypothalamus, released by neurohypophysis. Okay, and gradually the labor pains will increase and delivery of the fetus occurs. Write down clearly, parturition is controlled by. Shall I move little? Parturition is controlled by oxytocin. Not only oxytocin, dear. You might think that only oxytocin is involved in parturition. Not only that, and in PQ also they mention oxytocin, prostaglandins. Prostaglandins are produced from these placenta, fetal membranes. Okay, then uterine. That's for uterine contractions. And next one, cortisol, estrogen, relaxin. Last, after delivery, endorphins are also produced to relieve the pain, actually. Okay. So this is the concept, like cortisol and all, important to make the prostaglandin secretion to trigger for uterine contractions and all, glucose level to maintain in the body. So a lot of lung maturation, like because in the fetal life lungs are not matured, no. For lung maturation also cortisol plays very important role. Okay, later on only after delivery, baby starts breathing with the help of lungs. Then estrogen, like lot of help it will do. Progesterone, don't consider. Progesterone is for pregnancy only. During labor time, it means parturition time, progesterone is very low. Okay, because of that only parturition is happening. So estrogen is more, progesterone is lesser that time. Okay, and relaxes the cervical region and pelvis region. And parturition is followed by lactation, colostrum, IgA rich, so passive immunity is triggered. Okay, this is about human reproduction, reproductive health. ART, shall I say? ARTs, contraceptives. Ayyo.
Yeah. Family planning program was initiated in 1951. RCH, like all the reproductive health related awareness programs, everything is categorized under RCH only. Okay, and reproductive and child health care program is RCH. And amniocentesis, very, very important concept here for asking questions. Amniocentesis actually, it is a fetal sex determination test actually, and this is useful to know to find out the genetic disorders like Down syndrome, hemophilia, sickle cell anemia. Generally, write down clearly. It is like amniocentesis is suggested by the doctor from 15th week to 20th week. Something at that time only they will around one plus or minus 1 or 2 weeks it may be there, but around that period only 15th to 20th week, that time only they advise them to undergo with amniocentesis. Okay, they collect the micro needle will be there, doesn't rupture much, and with that amniotic fluid is picked up, and in that fetal cells will be there, some chemicals also will be there. Okay, the nucleus is absorbed properly here, the genetic material will be absorbed properly for the abnormalities and all that. So anything like which follow Mendelian inheritance can be identified, and even chromosomal disorders also can be identified, but which we can't identify is cleft palate, cleft lip and all this type of cleft palate, splitting of lip and all that we can't identify based on amniocentesis. Okay, and Saheli is once a weak contraceptive pill, and discovered by what? Central Drug Research Institute situated in Lucknow, and Saheli is non-steroidal. There is a chemical in that is centchroman. What's the name of the chemical present in Saheli? Centchroman, which can destroy the estrogen receptors on endometrium. That's why implantation is inhibited. Okay, no hormonal role, nothing, only because of centchroman, estrogen receptors on endometrium are destroyed, so implantation is inhibited. Okay, and this data, case study based data, you just go through here. Contraceptives. Natural method has more failure rate, and more effective contraceptives are implants. More effective contraceptives are implants. Okay, natural method here, periodic abstinence, certain period, 10th day to 17th day, normal like 28-day cycle means that particular period is avoided to undergo coitus. Okay, then withdrawal and coitus interruptus, penis is withdrawn before ejaculation. Then lactational amenorrhea. Here during lactation time, you know, maximum of 6 months actually she can't ovulate, the mother can't ovulate. The reason behind it, like lactating women have high amount of prolactin. High amount of prolactin will be there, so inhibition on gonadotropins, FSH, LH will not be produced. So because of that, she can't ovulate. Okay, and coming to barrier methods, females and males have condoms. Condoms actually not only to avoid the physical meeting of woman's sperm, they are also useful to avoid sexually transmitted diseases. Okay, so protection will be given from the transmission of STDs. Then diaphragm, cervical caps, walls, if you take it, they are reusable. Like condoms are disposable, and these are actually for female only, and these are reusable, and they can't give protection from STDs transmission. Why? Because they only cover cervix region, vagina will be still open only. So when intercourse happen, you know, the virus and all can enter into the body through that vaginal ruptures or something like that. So that's why it can't give a protection from STDs. Okay, coming to IUDs, intrauterine devices, 3 types of IUDs are there. Non-medicated IUDs, example, lips loop, copper releasing IUDs, hormone releasing IUDs. Okay, non-medicated lips loop and copper releasing copper T, copper 7, multi-load 375. The role of function is important. Copper releasing IUDs are for preventing the fertility and motility of the sperm. Okay, so and hormone releasing IUDs, example, progestazat and LNG20. Progestazat and LNG20. See dear, one more point I'll tell. Emergency contraceptive, in our weekend paper also one question was there, right? Emergency contraceptive purpose also IUDs are useful. Okay, which IUDs? Copper releasing IUDs, anything can be used. Okay, for emergency contraception I'm saying. And among hormonal releasing, levonorgestrel based anything can be used for emergency contraception. Are you listening? So emergency contraception purpose, copper releasing IUDs can be used, no doubt. Okay, among hormone releasing IUDs, progestazat will not be used.
Okay, so levonorgestrel based any IUD can be used. Okay, the role of hormone releasing IUD is to inhibit the implantation and make the mucus plug, cervical mucus plug. Okay, and oral contraceptive pills, 21 days pills. These are start in like first 5 days, any one day they have to start, 21 days continuously they have to take, and later 7 days gap they have to give, again they have to start the course. Like that, these oral contraceptive pills maybe in combination like progesterone and estrogen, or else only progesterone pills also can be there. Okay, and the main function of these pills inhibit ovulation, inhibit implantation, and make the cervix cervical region more thick with a mucus plug and all that. Okay, and implants injections also have the same mechanism like pills only, hormonal rich only. Okay, and mainly implants are progesterone rich actually, so long term contraception can be provided because of this. Okay, Narplant, Implanon, like that we'll say. Okay, and this is about contraceptive methods. Pills, IUDs can be used, and after coitus, the emergency contraceptives are used after coitus, so it will be there, not before that. No, I don't want. No problem. I am still student is, wow, so nice. Students are giving me juice and all that. No problem. Thank you so much. God bless you. Arey, wait, Ra. Thank you so much. God bless you. What's the time? My God, it's 2 o'clock. Okay, 1 second. Vasectomy and tubectomy. Okay, this is the surgical method, a sterilization method. And here vasectomy, vas difference is cut and ligated. Okay, tubectomy, fallopian tubes are cut and ligated. Okay, fine. Among these two, vasectomy is easier. Okay, healing time also lesser than the tubectomy. But in India, tubectomized females only you find more than the vasectomized males. Okay, so coming to here, in both the cases, because of this surgery, transport of gametes is blocked. Okay, transport of gametes is blocked. And one more, after this, like because of this surgery, they are not disturbing the testis function or ovaries function. So any like application level questions they can frame. Okay. So spermatogenesis or oogenesis is not at all disturbed here. Uterus functions are nothing else. Only the tubes are blocked to prevent the transportation of the gametes. That's all. Okay. So next one is one more. So after vasectomy, within like 1 or 2 months, like I think we consider certain ejaculations, actually it's not like months or something, few days in the semen, sperms can be there. After surgery immediately it's sperms can't be avoided, so semen is released without sperms after few days of the surgery only. Okay, and this person can ejaculate semen without sperms, and he can have his sexual life also normal only, no problem. Okay, coming to tubectomized female, her ovaries are functioning properly, uterus is functioning properly. So, general doubt is like whether she can menstruate or something like that. She can menstruate, no problem. Okay, only the tube is cut and ligated. So only ovum will not be there. Endometrial ruptures and all can be shed, no problem. Okay, and she can act as pregnant, like in IVF, in vitro technology. She can act like surrogate mother also. Okay, IUT can be done on her, and she can act like surrogate mother also. Okay. Side effects are there because of certain pills and all that, and nausea, abdominal pain, breakthrough, bleeding. MTP is legalized in 1971. 2007 amendment actually says very safe to terminate the pregnancy in the first trimester. Till 20 weeks also they can do. By considering certain health risk of the mother or fetus and all that, then 24 weeks pregnancy also can be terminated with the permission of 2 doctors. Okay. Here I have to highlight the diseases because here pathogen name is not given, but in the exam there is a chance of asking pathogen name, everybody. Okay, gonorrhea, See the list of STDs: gonorrhea, syphilis, herpes, chlamydiasis, genital warts, trichomoniasis, hepatitis B, like this. Okay, gonorrhea caused by Neisseria gonorrhoeae. Neisseria gonorrhoeae. In my classes, I have the list, and everybody would have had the information. Gonorrhea caused by Neisseria gonorrhoeae. Syphilis caused by Treponema pallidum. Okay, genital herpes caused by HSV2, herpes simplex virus 2, not 1, 2. You have to mention. Then chlamydiasis caused by Chlamydia trachomatis. Genital warts caused by human papilloma virus. Human papilloma virus. Trichomoniasis caused by Trichomonas vaginalis. Trichomonas vaginalis. Okay. Hepatitis B caused by Hepatitis B virus. Hepatitis B virus. Among these, let us separate bacterial viral infections like that. Gonorrhea, syphilis, chlamydiasis. These are the examples of bacterial diseases. Okay, genital herpes, genital warts, hepatitis B, these are the viral infections. Okay, and trichomoniasis here, this is protozoan disease. Once again, I will tell, mark correctly. Gonorrhea, syphilis, chlamydiasis are bacterial diseases. Herpes, warts, hepatitis B are viral diseases. Trichomoniasis is a protozoan disease. Okay, pathogen names, I think you are clear. Then one more point here. AIDS, hepatitis B can be transmitted by blood transfusions and also through sexual intercourse. And these two diseases, though they are STDs, they never disturb the reproductive system functions. Which one? AIDS and hepatitis B. They don't disturb reproductive system functions. They disturb the, like AIDS means immune system. Okay. Then hepatitis B, liver related functions are damaged. Okay. The next one. See children, when you go through symptoms, you early symptoms and if the complications and all, you just separately you learn it. Okay. And next, which age group people are more vulnerable to get affected? 15 to 24 years. Yeah, coming to this last concept of reproductive health, ARTs, assisted reproductive technologies. Okay, so here for infertile couple, like though they are not using any contraceptives and all that, unprotected sex is there, but even they are not able to produce children, that case is considered as infertile case. Okay, for them initially they advise tablets and all that, injections, hormonal supplements and all that. Even then the body is not responding, fertility is not gained. At that time, doctor finally advised the couple to undergo assisted reproductive technologies, ARTs. Okay, here in vitro technologies are there, in vivo technologies are there. In vitro, in vivo. In vitro technologies, example, many times we discuss about it, but still children will be having the confusion. In vitro means fertilization happens in the laboratory. Okay, so in vitro technology only test tube baby program. Okay, and after fertilization in the laboratory, we follow like embryo transfer technology will be. Embryo has to be transferred to the female, otherwise biological mother or the surrogate mother, like that. Okay, this is only test tube baby program. In vitro technologies are ZIFT, okay, IUT, intrauterine transfer. Okay, ZIFT, IUT, ICSI. It's there in next paragraph, so don't get confused thinking that is in vivo. It is in vitro only. ICSI. Okay, intracytoplasmic sperm injection. More commonly nowadays ICSI is used, and that success rate is also more for that. Okay, ZIFT. When ZIFT is allowed means like couple related gametes they can take whenever her fallopian tube ampulla region is not supporting for successful fertilization. Doctor advised the couple to donate their gametes, and at that time in the artificial environment in the laboratory only fertilization will be successfully conducted. Zygote are the embryo till 8 blastome year stage. Okay, that will be injected to fallopian tube. Zygote, see ZIFT, remember thoroughly in case of ZIFT. Zygote are the embryo till the 8 blastomeres can be injected to fallopian tube. Okay, and IUT. So here embryo with more than 8 blastomeres has to be injected into uterus. Okay, embryo with more than 8 blastomeres has to go to uterus. Okay, that is IUT, intrauterine transfer. Okay, so after that, ICSI is like the doctor only will take the active sperm and give the injection to the secondary oocyte. So in front of them only, like on the slide only, fertilization will be successful outside. Okay, that is ICSI. After that, he can follow either ZIFT or IUT to transfer the embryo to the body. To the fallopian tube means ZIFT. Uterus means IUT. Okay, like that. And here, even in vivo, like sometimes the embryo transfer can be done. Suppose, see, one biological mother is there. Her fallopian tube is healthy only, but uterus is very small. Uterus is not supporting for the gestation. Every time abortion. Like that time, no doctor, what he'll say, okay, let morula stays and all, like zygote, like this, 8 blastomeres and all, continue in her body only. Normally, her ova, her husband's sperms fertilize, and the morula stage is also there in her fallopian tube. Okay, so after that, her uterus is not supporting for the implantation. Na? So doctor will take out that morula stage and he can place in the surrogate mother's surrogate mother's uterus actually. So that's how IUT can be done. So embryo transfer can be done in even the in vivo technologies also. Like biological mother's body, suppose till morula stage. Later on, surrogacy can be done. Okay, so no, I don't want. Okay, next one, in vivo technologies. In vivo technologies like here, 1 second. Coconut juice, what is this? No dear, no, time is not there. No, it's okay, let me complete. The health and disease also immunity I have to complete. No, I am fine, I'm really. Yeah, everything here and okay, thank you so much. Love on Zoom on me, okay. Okay, in vivo fertilization. Here, fertilization happen in the female body actually, in ampulla region. See, what is that? So, shh, don't disturb. Okay, in vivo means fertilization happens in the female body itself. Okay, so here in this technology, first we have to focus GIFT, gamete intrafallopian transfer. Then after that, AIUI, artificial insemination, IUI. These are the technologies we will consider under in vivo fertilization. When doctor advises the couple to undergo GIFT means anovulation condition. Anovulation means she is not able to produce ovum. Amenorrhea means no menstrual cycle, probably some disorder or something. Okay, birth defects or something. So contribution of ovum is not there. But the rest all uterus, everything is fine or something. Ovary is the issue. Fallopian tube is fine, uterus is also fine. At that time, doctor advised the couple to undergo GIFT. Okay, normally her husband's sperms only will be introduced and only gamete is taken from the donor. Okay, and normal fertilization happens in her ampulla, only fertilization will get over. So in the question they will explain thoroughly, like the female is healthy, her fallopian tube, uterus, everything is healthy. Only ovaries are the problem, like anovulation or something is the problem. At that time, doctors advise her to undergo GIFT. Okay, then this is about in vivo fertilization. And one more, AIUI. When doctor advises to undergo like artificial insemination means husband is not able to inseminate. That's a problem. Maybe penis erection problem or something, ejaculation issues or something will be there. And one more is like sperm count is less. Okay, maybe alcohol addicted or some other reason, drug addicted or something, maybe health issues or hormonal related issues or something. That time no sperm's count also will be very less. At that time our insemination issues are there. At that time doctors advise the couple to undergo artificial insemination or intrauterine insemination. So artificial insemination in general, no, the semen from donor, like our husband, that will be injected into either vagina or into the uterus. If it is injected into the uterus, it is called as intrauterine insemination. Children, okay? This is about
ARTs. Next, health and diseases, immunity. You'll study. We'll stop with this. Your biodiversity problems and all. 1 second. Okay, calculations and all you can follow, right? Yeah. Then we'll stop with this. Our immunity, somebody messaged dear here. Okay, shall we close the session then? All the best, everybody. Study properly. Okay, due to time problem, I couldn't complete certain chapters. Go to the self-study. Okay, all the best, everybody. Okay.